Learning process of bachelor degree. In the pandemic, learning is generally carried out by Zoom. Even though it is online, various learning methods in delivering courses can still be done, such as providing one-way material, discussions, group discussions, and doing practice questions. Delivery of lecture material is carried out using either a PowerPoint.
Ya. Jadi kalau kita makan lemak ya, misalkan gajih gitu ya.
Muncul persatuan volume dikalikan.
Explaining from textbook sources.
Konsentrasi A dikali HR ini kan. Tetapi ini berbeda karena ada yang mengurangi sejumlah HR absorpsi tadi ya.
As well as adding other sources that can enrich the subject. For example, in the subject of atomic and molecular structure, students are shown the website chemtube.com, which visualize several types of molecular orbital to make students become more aware of symmetry that are difficult to imagine. In the subject Modern Physics, the lecturer also introduced students to use EduGame in gamelab.mid.edu to help students understand about the relativity concept.
Nah, warnanya kalau kita perhatikan itu kayak ke arah biru.
The subject structures, properties, and reactivity of inorganic compounds. Lecturer asks students to make the molecule model using plasticine when explain about the molecule symmetry concept. Lecturer also developed the web from the medium to deliver the subject content as performed in the subject chemometric. Some subjects also add the external exposure to introduce students the subject application. For example, in the subject marine chemistry, students go to the sea to see various marine species. Then they discuss the relation to the subject. This activity was done in 2019 before COVID pandemic. In the pandemic, the external exposure is replaced by the virtual external exposure. In the subject chemistry membrane, the lecturer invites the industry practitioner to sharing about the problem of membrane application in industry, and the students are invited to discuss the solution. In the subject research methodology, Students are required to be more active in exploring chemistry literatures and writing it down in the form of manuscripts, oral presentation, and posters. Guidance on how to search literatures is done in practice, directly by showing several websites related to searches on chemistry. For subjects that require students to have the ability to calculate, exercises are carried out and discussed carefully. Implementation of assessments are carried out in various ways, such as conducting tests via My ITS Classroom and supervision using Zoom, or giving other tasks that can increase learning outcomes in the form of skills. For example, subjects biochemistry and foodstuff chemistry have assignments that are required to be submitted through the TikTok app. Subject chemical literature gives out assignments of video presentation, example, video, and poster. During this pandemic period, practicum is carried out either with demonstration in the laboratory with the help of lab assistants, or some practicum are developed with an online module. The following is a video footage of the Chemistry 2 Basic Technique Laboratory. Measurement of density.
Dengan ukuran penyitaan data dan cair.
Solution.
Mari kita lakukan percobaan 3.
Thermodynamics. Calorimeter bomb. Binary solution. Freezing point.
Kita akan melakukan praktikum termodinamika yang membuat massa melakukan dengan menggunakan metode
Chemical dynamics. H2O2 decomposition. Acetone iodoform reaction. Synthesis organic reaction practicum. Styrene polymerization. Welcome to the Chemistry Department Faculty of Science and Data Analytics, Institut Teknologi 10 November, Surabaya. The Chemistry Department of ITS has plenty building facilities for students to use. For instance, classrooms with capacity of 40 to 50 persons, each with LCD projector, AC, and Wi-Fi. Trial rooms for final project presentation and thesis. And discussion space at the corridor. The chemistry office for administration is placed on the second floor. There is a waiting
2023, with General Importer as core of its business, PT Fajar Mas Murni has marked the beginning of its glorious journey. In its first office in Menara Hijau, PT Fajar Mas Murni has morphing into a trading company that actively contributing to Indonesia's national development up to this very moment. It has also becoming a preferred and trusted company for its business partners. On the 11th of May 1978, PT Fajar Mas Murni has accepted the challenge to enter the global market by establishing international partnerships and represent several prominent brands from numerous countries. To assert the transformation and strengthen the image as a dynamic, competitive, and always keep abreast of technological advances company, on 2021, PT Fajar Mas Murni has rebranded its logo. Becoming an exclusive distributor for several prominent brands, PT Fajar Mas Murni is confident that it can give a high standard support to 4 business groups: industrial technologies equipment, construction and mining equipment, laboratory and inspection equipment, rotating and process equipment. By establishing branch offices in major cities in Indonesia, PT Fajar Mas Murni is ensuring that the service quality align with its market expansion. PT Fajar Mas Murni is committed to establish good relationship with business partners. With complete warehouse and workshop facilities and supported by skilled and professional human resources, PT Fajar Mas Murni is providing the first-rate after-sales service in its field of business. All aligned with PT Fajar Mas Murni vision and mission commitment to provide the excellent services by implementing the good management standard on quality, health, safety, and environment.
learning process of bachelor degree. In the pandemic, learning is generally carried out by Zoom. Even though it is online, various learning methods in delivering courses can still be done, such as providing one-way material.
para ilmuwan, walaupun ada yang offline, ada yang online, sehingga acara ini hibrid, tetapi secara garis besar kita dapat mendapat pencerahan ilmu-ilmu baru.
Audio Jungle.
Before starting the event today, let us give a warm welcome to the honored guests. First, the first guest lecturer Professor PhD, the Dean of ITS Science and Data Analytics Faculty, Professor Mansuri, ITS Chemistry Department, Professor our keynote speaker, Professor Shigeru Okada from University of Tokyo, Professor Yun Hao Ng from City University of Hong Kong, Dr. Aditya Farhan Harif from Mineral Industry Indonesia Company or MINE-ID, Professor Mark Benton from Curtin University Australia, and Professor Fahima Martak from Institute Technology Spona Pember. Also, our invited speaker, Dr. Hoi Chung from City University of Hong Kong, Professor Masato Tominaga from the Saga University of Japan, Association Professor Zahir Jude from Nanyang Technological University, Singapore, Association Professor Satria Zulkarnain Bisri from Tokyo University of Agriculture and Technology, Dr. Ziju from Jiangsu University of China, Professor Dr. Peri Burhan, Professor Ratna Diati, Professor Adi Setio Purnomo PhD, Mrs. Sri Fatmawati PhD, and Mrs. Zeni Rahmawati PhD from Institute Technology Spona Pember. Good morning, ladies and gentlemen. Welcome to Bali, the island of paradise, and welcome to the 6th International Seminar on Chemistry 2024. In this morning event, I, Muhammad Haikal Fazet, you could call me Haikal, will be the master of ceremony for today. And we welcome once again all of our participants to Bali. It is such a pleasure for us to greet you today on our first day plenary sessions on Isoc 2024. This year's Isoc is taking a theme of Elevate Humanity with Chemistry. As a scientist, all of us here, we strive to make great changes, inventions, and new findings in our own respective fields and ideas. We are hoping that through our hard work in the field that each of us will present, most of us here, will present in the next 2 days, we can do our part to elevate humanity itself. I think that's all that I gotta say, since we will have many things to do today. Before we start, let me, as the master of ceremony, read today's rundown event. First, we're gonna have the opening remarks. Then we will continue to our keynote session. We will have 3 keynote sessions by today, and between that, we will gonna have a coffee break. After all the speakers conclude their materials, we will close today's plenary sessions to have lunch and start our parallel session later on. So without further ado, first of all, we're gonna have some of you opening sessions and remarks, as well as welcoming hands from us to all people present in this year's conference. First remarks will be given by the chairman of the 6th International Seminar on Chemistry 2024, Dr. Trianda Gunawan, SSE. To Mr. Trianda, the time is yours.
Baik, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Dear our honored guests, respected speakers, fellow colleagues and participants. It is my distinct honor and pleasure to welcome you to the 6th International Seminar on Chemistry, Isoc, held at the Grand Wish Hotel in the Nusa Dua, Bali, Indonesia. This year, we celebrate a special milestone for Isoc. It marks our 10th anniversary of Isoc, and over the past decade, this seminar has grown into a vital platform for chemists around the world to connect, share, and innovate. We are delighted to have 202 participants, with participants both offline and online, from 7 different countries, including Hong Kong, China, Japan, Thailand, Taiwan, Malaysia, Brunei Darussalam, and Indonesia. Joining us, reflecting the global reach and significance of this event. Your presence here is a testament to our shared commitment and to advance the field of chemistry and its impact on humanity. The theme for this year's seminars is Elevate Humanity with Chemistry, underscore the profound role for chemistry plays in addressing some of the most pressing challenges faced by our society, from developing sustainable technologies to improving health outcomes and protecting our environment. Chemistry is at the heart of many solutions that elevate the quality of the life for people around the world. Our program is packed with insightful presentations, interactive, hopefully, Q&A sessions, and stimulating discussion led by distinguished speakers and experts. I would like to extend my heartfelt thanks to our keynotes and our invited speakers for sparing their time coming to this event and accepting our invitation. Moreover, sharing their invaluable knowledge with us. Additionally, I express my gratitude to all of our participants for their enthusiasm and contribution to this seminar. I am confident that the knowledge and ideas exchanged over the next few days will inspire us all and contribute to meaningful advancement in our fields. I would like to extend my deepest gratitude to the organizing committee team for their tireless effort in making this event possible. Their dedication and meticulous planning has ensured that we have comprehensive and engaging seminar, hopefully. Additionally, I thank our sponsors and partners for their invaluable support as we embark on this journey of discovery and collaboration. I encourage you all to actively participate, share your insight, and build lasting connections. Let us make the most of this opportunity to learn, innovate, and elevate humanity with the power of chemistry. Thank you for being here, and I wish you a very productive and memorable seminar. Thank you.
Thank you so much for the wonderful speech, Mr. Trianda Gunawan. And following after the speech from our chairman, we're going to go to another remarks from the head of ITS Chemistry Department. We invite Professor Renat Freddy Kurniawan to the front. To Professor Freddy, the time is yours.
Good morning, everybody. Assalamualaikum warahmatullahi wabarakatuh. Dear honored guests, respected speakers, fellow colleagues and participants, it is my great honor and pleasure to welcome you all to the 6th International Seminar on Chemistry, Isoc, hosted by Department of Chemistry at Grand Wish Hotel in the beautiful Nusa Dua, Bali, Indonesia. This year, we are celebrating the 10th anniversary of Isoc, a milestone that reflects our ongoing commitment to fostering scientific collaboration and innovation in the field of chemistry. Over the past decade, Isoc has grown into a premier event that brings together chemists from around the world to share their latest research, exchange ideas, and build lasting professional relationships. This year's themes, Elevate Humanity with Chemists, highlight the crucial role that our field plays in addressing global challenges and improving the quality of life for people everywhere. We have an exciting program lined up for you, featuring distinguished speakers, engaging presentation, and interactive workshop. I'm confident that the discussion and collaboration that emerge from this seminar will be both inspiring and productive. I would like to express my deepest gratitude to organizing committee for their hard work and dedication in making this event possible. I also extend my sincere thanks to our sponsor and partners for their generous support. As the host of this seminar, I look forward to the fruitful collaboration and stimulating exchanging that will take place over the next few days. I encourage each of you to take full advantage of this opportunity to learn, share, and connect with your peers. Let us work together to advance the frontiers of chemistry and elevate humanity through our collective effort. Thank you for being here, and I wish you all a successful and enjoyable seminar. Thank you very much.
Thank you so much for the thoughtful remarks from Professor Freddy. Right now we go on to the last speech and remarks, as also the symbolic opening of the 6th International Seminar on Chemistry 2024 will be done and delivered by Professor Nurul Widyastuti, our Vice Rector of Academic and Student Affairs of ITS. To Professor Nurul, the time is yours.
Bismillahirrahmanirrahim. Good morning everyone. Assalamualaikum warahmatullahi wabarakatuh. Dear esteemed guests, distinguished speakers, colleagues and participants, it is with great pride and pleasure that I extend my warm welcome to you all at the 6th International Conference or Seminar on Chemistry hosted at the beautiful Grand Breeze Hotel in Nusa Dua, Bali, Indonesia. As a Vice Rector for Academic and Students Affairs, I'm honored to be part of the prestigious event that brings together some of the finest minds in the field of chemistry from around the globe. The seminar represents the unique opportunity for us to engage with groundbreaking research, innovative methodology, and the latest advantages in our field. The diversity of the topics in the seminar and the expertise of our speakers promise to make this an intellectually stimulating and enriching experience for all participants. We're lucky here to have 6 countries, participants from 6 countries here, from China, Japan, Brunei, Malaysia, Taiwan, Thailand, and also Indonesia. It is time for us to have collaborations, to sharing the idea, to have collaboration and networking in the future as well. So our location here in Bali, renowned for its natural beauty and cultural richness, provided an inspiring backdrop for our discussion and collaboration as well. Enjoy Bali because this island is called the Island of Paradise. We can enjoy the beach, the sunset, the sunrise, and also the cultures mixed with the Hindu's religion. The uniqueness of Bali will inspire us to have a new idea, discussion for our research and for our educations in our institutions. And I would like to express my heartfelt gratitude to the organizing committee for their dedication and hard work in bringing this event to life. Their efforts have ensured that we have a comprehensive programs that caters to a wide range of interests and expertise. Furthermore, I extend my appreciation to our sponsor and partners for their generous support that has been instrumental in making this seminar possible. As we embark on these days of learning and networking, I encourage each of you to actively participate, share your insight, and build connections that will drive the future of chemistry. Let us use this platform to foster collaboration, inspire innovation, and advance our collective knowledge. Thank you for your presence and contributions, and I wish you all a productive and enjoyable seminar. Assalamualaikum warahmatullahi wabarakatuh.
Thank you so much for the wonderful speech from Professor Noro. Before we move on, we're gonna have a photo sessions to commemorate today's event for the handovers of the souvenirs itself. We invite our rector once again, Professor Noro, to the front, our chairman, Mr. Trianda, and our head of department, Professor Freddy, to the front to commemorate these photo sessions. Is the photographer ready? We're gonna wait a second. 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 Okay, thank you so much for that is all for the photo session itself. Thank you so much. And okay, thank you so much for that is all for the photo session itself. Thank you so much. And okay, thank you so much for that is seems to for the photo session itself. Thank you so much. And okay, thank you so much for okay, it seems fixed now. And can we get a round of applause once again for today's event? Okay, that's great. Now, after the keynotes, after the speeches, the opening remarks, once again, we're gonna go, we'll hear a few presentations from great people in these rooms and in their respective areas. We are now continuing to the next agenda, which is the keynote sessions. For the first sessions, it's gonna be moderated by Mrs. Sri Fatmawati, PhD. Is Miss okay? Since Miss Fatma is here, for the sessions, the time and place is yours.
Okay, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. So this is a very good opportunity for us to be here in Bali. I just landed 15 minutes. And feel that Bali is always like a holiday, isn't it? So I think this is a very good opportunity for us to share and also making a new connection, enlarge our network, and also we can build a lot of collaboration during this seminar. And I believe that this is also a good time, a good chance for us to promote our science, our research, and also our group. Yeah. And today, I would like to introduce one of the keynote speaker of this Isoc is Professor Shigeru Okada. Good morning, Sensei. Yeah. Professor Shigeru Okada, or we call Okada Sensei, Sensei. Yeah. He's a professor in Graduate School of Agriculture and Life Sciences in Tokyo University, and his specialty in the fisheries chemistry. And so, for today, his research is about the biofuel production by microalgae, which is the microalgae and also hydrocarbon, and also the terpene, isoprene, and also biofuels will be the key point of his presentation. And I believe this is very, very interesting for us, since we are, we are working for, sustainable development goals, which is we need to promote how we can protect our one Earth. And I believe that Professor Shigeru Okada Sensei will deliver a lot of things, very interesting, and he will present about the let's make petroleum in your pond, hydrocarbon production by microalgae. So, Professor Shigeru Okada, time is yours, and yeah, we will discuss more about this. Thank you.
Okay, good morning, and thank you very much, Dr. Fatmawati, for nice, very kind introduction for me. And first of all, I would like to thank to the organizers for inviting me to such a nice press body, and also giving me an opportunity to talk about my research at this the 6 Isocs. So, sorry, my title is kind of joking. So and maybe you can judge if that's true or not after you listen my talk. So then I have been working on biofuel production from microalgae. But as you know, not only algae, photosynthesis organisms is considered as a renewable energy source. And this is also kind of joking. So it's not drinking beer, but carbon dioxide fixed by photosynthesis makes their bodies, and once it combusted, it does not generate new carbon dioxide in the atmosphere. So it's cycling. So from this viewpoint, this so-called concept is called carbon neutral. So then, I think most of you are chemists, and I'm a biologist and a chemist. I'm not sure what I am, but I want to briefly introduce what algae are. If you are asked what algae are, what do you think? What your answer? Maybe you said maybe this some plant living in water, or some very primitive plants or ancestor of land plants. All answers are correct, but not perfectly correct. I want to explain. Because as you can see, very long time ago, photosynthetic bacteria, like cyanobacteria, was eaten by some other organisms. And he or she should be digested as diet, but it could survive and start to function as a plastid, chloroplast. So that is the origin of the beginning of the chloroplast. So this event is so-called primary symbiosis, made the original plant cell. Then originally, at first, that's a very simple single-cellular organism, like a green algae. But it's developed to be the multicellular organism, like a moss or a fern or flowering plants. So that means that they evolved linearly. They are all a family. But once such a very primitive single-cell green algae was generated, again, other organisms ate it. Then again, this event, that means that 8 eaten organisms started to function as a plastid. Then this organism stopped to eat diet and lived with simply photosynthesis. This event is so-called secondary symbiosis. And algae are composed of both types of organs, narrow meaning plants, and this quite unique one. And this event, the combination of these organisms and eaten organisms is so many. So that means that this organisms result of secondary symbiosis includes many different type of organisms. So that means the definition of algae is very difficult. Just oxygenic phototrophs excluding the moss, fern, and flowering plants. So that's the kind of definition, not so clear. But because of this divergence of the species, I can show several examples of cyanobacteria, prokaryotic photosynthesis organisms, green algae, or Eugurena, diatom, and dinoflagellate. Those are result of secondary symbiosis. So that means the word algae includes divergent biological groups. So then that makes a difference, very big difference of the ecology or biochemistry or chemistry. So I want to show one example. This is a picture in a hot spring in Japan, and the water temperature is very hot, higher than 80 degrees Celsius or more. So then there is a kind of very famous Japanese souvenir at the hot super. It's so-called onsen tamago, is a hard-boiled egg. So that means chicken egg is soaked into hot super. It turned into the boiled egg. That means normal protein quite easily denatured by heat. But you can see something green in it. They are cyanobacteria that has a very high tolerant proteins. So that means this kind of microalgae has a very unique biochemistries. And another example is this is a picture taken at Lake Letova in Senegal in Africa. So this lady is collecting the salt. So that means that the water of this lake is very salty. But you can see very strange color of the water. And this is because of this organism, so-called Dunariera. This is a member of green algae, but the color is not green at all. Because this organism accumulates a lot of beta-carotene, that's a pigment of carrot root. So then water color becomes like pinkish color. And more interestingly, this Dunariera, this is a green algae, so that means green algae generally has a cell wall to protect them. But this species does not have cell wall. It's a naked green algae. So that means once such kind of naked organism is exposed to a very high osmopressure environment, they should die, but they don't. The reason is that they can accumulate a very high level of glycerol to compensate the osmopressures. So that what I want to say by these pictures, some of the microalgae can live in very extreme conditions and have a very unique biochemistries. So then I also want to emphasize, because the efficiency of photosynthesis by microalgae is very effective, very high. Because, for example, to make some oil, sunflower is also can be made, but oil can be obtained from only seeds. That means the leaf or stems are kind of waste. But on the other hand, microalgae has a very simple body systems. So then life cycle, because of this simple system, life cycle is very short. Sometimes some species divides into two within several hours, very quickly. And many of the algae species also known to accumulate a lot of lipids, oils. So that is the idea why the algae, especially microalgae, is promising for the biofuel productions. So I just list up the merits of the microalgae to algae or microalgae to use the biofuel resources. So carbon dioxide fixation is carbon neutral, and there are so many different species. That means a unique chemistry or a unique biochemistry, and some of them can grow in extreme conditions. That means no competition for space against for the food productions. And the simple body structure means a very high efficiency of carbon fixation. Everything's good, but of course there are several demerits. Demerits is that they are so tiny, we cannot see it by naked eyes. So that means that while we try to culture them, we cannot know what happened during the culture. And also, the history of the research on microalgae is still too short if compared to agriculture. So that means the input energy is sometimes much larger than the energy that we can get as biofuels. That's a problem. So I want to show you that what kind of bioenergy, biofuel can be obtained from aquatic species. So there are four types of compound: methane, ethanol, hydrogen, lipids. So then those methane and ethanol are not produced by aquatic organisms, including microalgae, but created by the fermentation by microorganisms. But on the other hand, some microalgae can make hydrogen and lipids by themselves. That means that methane can be produced for anything, including organic compound, and ethanol can be produced by glucose. That means microalgae also have cell walls, and that composed of glucose that can be converted into ethanol. But microalgae can be used for methane production, ethanol production, hydrogen production, and lipids. So that means in terms of the biofuel production, microalgae must be almighty. So then, I want to introduce how to use algae as biofuels. So the first example is, of course, methane fermentation. So, but as I mentioned earlier, methane can be generated anything, including organic compound, by degradation by microorganisms, bacteria. Something strange. Sorry. But mainly, this component is converted, digested into the organic acid like acetate. Then very unique group of microbes, the so-called methanogen, can convert those organic compounds into methane. Then I want to show some example. So there is a very huge research project, so-called Aquatic Species Program in United States. So then they tested many macroalgae, seaweed, and microalgae. Then this is one example. This is a so-called giant kelp, the brown algae. So it's distributed in the west coast of the United States, and it grows very quickly. So that means that once harvested and used for the methane production, but still next day, the material is already ready. So that's the idea. So then actually, this level of methane could be produced by brown algae. Then this is another example carried out in Japan. This green algae, green macroalgae, ulva, is sometimes a problematic species that propagate so huge in inland sea. So then this Tokyo Gas Company used 1,000 kilograms of wet algae and could convert it into methane of 2 kiloliters. And actually, it can be practically used. So that means that methane can be produced by mainly macroalgae. But of course, microalgae can be used. So next example of the biofuel is hydrogen. Of course, as you know, hydrogen is very clean energy that does not emit any carbon dioxide at the combustion. And the very unique group of microalgae, so-called cyanobacteria, can produce hydrogen with nitrogenase. And as you know, the fixation of nitrogen is very, very important for agriculture to make a fertilizer. And this very famous Haber-Bosch process was invented. Because of this, we can get sufficient food now. But the cleavage of this triple bond of molecular nitrogen is very hard because it's so stable. So then it requires a very high temperature and high pressure, and it makes a large consumption of fossil energies to make it. So then generally, biological reaction catalyzed by enzymes is more energetically efficient than simple chemical reactions. So then nitrogenase. So some group, so-called diazotrophs, has this enzyme and can fix the nitrogen. And this is the reaction by nitrogenase. But as you can see, 1 mole of nitrogen is converted to 2 molecules of ammonia. It requires 16 ATPs, so high energy values. So that means that even biological reaction using the biological catalyst and the enzyme still is very difficult to complete these reactions. Then during this process, the one molecule of hydrogen is generated. So the idea is to use this hydrogen as a biofuels. But the problem is the nitrogenase is very sensitive to oxygen. So that means that on the other hand, the cyanobacteria is a photosynthetic organism and it generates oxygens. So that means a normal vegetative cell cannot do this reaction. So that means the cyanobacteria make very unique cells, so-called heterocysts, to prevent the permeation of the oxygen and consent to focus on the only nitrogen fixation. But instead, they cannot make sugar. So that means that normal vegetable cells can get ammonia from here, and instead sugar is transferred to the heterocyst. It's kind of social communications, even among the bacterial cells. So the third example is lipids produced by algae. So then, lipids can be produced by algae themselves. Then, but if you want to start some research on lipid production by microalgae, you need to select very good strains that can accumulate a lot of lipids. So this is So this is a conventional way, so-called the fluorescence dye, so-called body P. So it's permeable in the living cells and can bind to the hydrophobic compound. You can see the green or yellowish fluorescence. This is the lipids. And the red one is the autofluorescence or chlorophylls. So that means if you see this cells species does not accumulate so much lipid, but this is a lot. So that means that maybe if you'd better pick up this cell, this species to start biofuel production by lipids. So then most of the microalgae accumulates the lipid in the form of triglycerides. Then you can see this table. There are many different classes of microalgae, and some of them accumulate a very large amount of triglycerides, the 70% of algal dry weight. And triglyceride itself is not so easily combustible. If it is, I have very dangerous portion. I have accumulated triglyceride here. But once it converted the methyl ester by ester transferication, this fatty acid methyl ester can be used as a biodiesel, and car can drive by this. And also, quality of fuels depends on the property of this alkyl chain, of course. But problem, of course, this biodiesel is very good, but one problem is this is including the oxygen atom. That means already oxidized. In other words, oxidized means halfly combusted. So that means the energy combusted by this type of energy is not so high. So that means that to use as a jet fuels, fatty acid methyl ester is not so good at this moment. And another problem is some microalgae can grow, propagate so quickly, and also accumulate a lot of lipids in the form of triglycerides. But the problem is while they are propagating actively, they are so skinny. They don't accumulate lipids. Then once their growth is stopped by some environmental stress, then they start accumulation of oils. So that means the growth rate does not always mean the rate of the production, oil production rate. You have to be careful. Okay, then second type of lipids is hydrocarbon. And sorry, this very long introduction, but this is from now I want to focus on my own research. So hydrocarbon is of course composed of only hydrogen and carbon. That means a quite big difference from the fatty acid methyl esters. And then very good for jet fuels. But many microalgae can produce the hydrocarbon themselves, but the contents is generally very, very low. The lower than 0.01% or so on per dry weight base. So, but there are some exceptional algae. This is what I have been studying so long time. that's a Botuliococcus brownie. That is a green microalga. And this is a freshwater alga and unicellular. So this is a cartoon, and this is a single cell. But single cells are connected together by the biopolymer synthesized by themselves, then makes this kind of colonies. And the name, the genus name of Botuliococcus means Botulio means like a grape. So it looks like a grape, the fruits. So then, the quite unique point of this alga is the hydrocarbon content is very high. The maximum contents is 76% of dry weight. It's quite good there. But even algae cultured in the lab, still 40 to 50% of the hydrocarbons. And those hydrocarbons are synthesized inside the cell, but secreted outside. Then accumulate, but unfortunately not directly released to the environment, but trapped in biopolymers by making these colonies. So then once we observe this microalga under the microscope, and by pressing the cover glass, you can see secreted hydrocarbons is extruded from colonies. So then another interesting aspect of this alga is the secreted hydrocarbon shouldn't go back to the inside cells, and the algae does not use as kind of this hydrocarbon as a nutrition anymore. So that means still it's mysterious why this algae need to produce so large amount of hydrocarbon and secret and never use it. And researcher knew that this algae actually biosynthesized hydrocarbon, but it's not so old, just 1968 or so on. But before that, geochemists already knew that some oil shells are produced by this alga. So this is living Botuliococcus, and each cell dies and it may be degraded by bacteria. But hydrocarbon trapped, this biopolymer is trapped, and this biopolymer is so chemically stable. And after a long time, it's become kind of oil shells. So then there are several reasons why this Botuliococcus brownie is considered to be a very promising source of alternative petrol. Because this is a stratum of this oil shell, so-called torbanite, and this is also definitely can be combustible. So that means that the time this stratum is generated, this species was propagated so largely and very so long time. But in the modern time, this is the lake of in Australia. Sometimes this algal species make a blooming or red tide and covers the entire surface of these lakes. So that means that maybe the large-scale culture of this algae can be possible. And still the origin of the petrol is not known, not clearly, but at least the crude oil discovered by this Sumatran area in your countries, this is a so-called Botuliococcus. This is the fully hydrogenated form of the Botuliococcus that I later mentioned. So that means at least the Sumatran crude oil was generated by Botuliococcus in ancient times of Botuliococcus. And the hydrocarbons produced by this alga is just like a heavy oil in terms of the boiling point. But once hydrocracking is done, the hydrocarbon from Botuliococcus can be converted to gasoline, that the yield is more than 70%. So that means that definitely the hydrocarbon generated by the B race, or the one of the Botuliococcus, can be used as an alternative of petroleum. But another interesting of this alga is Botrycoccus browni is recognized as single species. But there are 3 chemical races that produce completely different types of hydrocarbons. The first race, so-called A race, it makes a linear alkane, so-called alkadiene and alkatrienes. And the second group, B race, produce a triterpene hydrocarbons, branched and with double bond. And the Botrycoccus is very specific to this organism, then named by this name. And this is a major component of hydrocarbon fraction, more than 90%. But in addition to Botrycoccus, as a minor component, the methylated squalene is also found. The squalene itself is not so rare. We also synthesize squalene to make stereol. But the methylated squalene is so very unique and maybe specific to these organisms. So this is the B race. And third race, L race, produces exclusively this tetraterpene, C40 hydrocarbon, so-called lycopodienes. So that means that if they need hydrocarbon for some biological reason, but why they produce such a different type of hydrocarbons? That's another question. But as for the biofuels, among those 3 races, the B race is the most promising because the structure of hydrocarbons and also the average hydrocarbon content is rather higher than other 2 races. But the problem, so I just talk about very attractive things about Botrycoccus, but of course there's a demerit. Because Botrycoccus browni grows very, very slowly. So as you can see, this is Botrycoccus, day 1, then this Chlamydomonas is a model organism, model green algae. So even at day 4, the culture of Chlamydomonas becomes denser than Botrycoccus. As I mentioned, the growth rate of microalgae, it's represented by the doubling time. Doubling time is how long time it takes to make a 1 cell division. Then the quicker one make a cell division during several hours. It's more or less like bacteria. But Botrycoccus has a doubling time of 1 week or 2 weeks. 1 week, after 1 week, 1 cell becomes 2 cells, and another 1 week, the 2 cells become 4 cells. So it's very, very slowly. So that means this algae cannot be a winner, cannot become a winner if there's a competition for the nutrition with other microalgae. That's very makes it difficult to make a large-scale culture of this algal species in out open pond. So then why this alga grows so slowly? So this is not my research, but by an American group. So they fed the radio-labeled, carbon-14 labeled carbon dioxide to this algae and chased the what type of compound is generated. Of course, at the beginning of the reaction, most of the radioactivity was observed in water-soluble fraction because the product by photosynthesis is water-soluble. It's quite reasonable. But even after, within 1 hour, 60 minutes, you can see almost 60% of carbon dioxide was converted into hexane-soluble material, some lipids. And actually, the 30% is this hydrocarbon. So that means, again, this organism does a photosynthesis. But the fixed carbon dioxide is actively converted into hydrocarbons and secreted outside and never use it again. So that means that such kind of organism wasting energy cannot grow so fast. That's kind of fate of these organisms. So then this is the picture of my laboratory. So we are taking care of my culture very carefully, preventing any contaminations. But I'm using the fluorescent lamp, and also I'm doing aeration. Once the air containing higher concentration of carbon dioxide, like 1 to 2%, enhances the growth rate of this alga from 1 to 2 weeks to maybe at least maybe up to 2 days still. but it's much better than nothing. But then I'm saying I'm doing the research on energy production. But some people criticize me, you are doing the research on energy consumption. Maybe that's true at this moment. So then most of the hydrocarbons or other related lipids are accumulated outside the cell, as I mentioned. So that means that we don't have to break down the cell to recover the lipids from this alga. Just dried algae is soaked into some organic solvent like hexane. Then most part of the lipids existing in the out of cells can be recovered. Then such a residual amount of lipids staying inside the cell can be recovered by using some organic solvent that are permeable to cells, like a mixture of methanol or chloroform, or just mechanically breaking down the cells. So Botulcoccus researcher defines those are fractions are the outer lipids and those are inner lipids. And let's see how unique lipids are produced by this alga. So this is the hydrocarbon or related compound produced by the A race of Botulcoccus. As I mentioned, it makes a linear alkane, alkadiene, alkatriene, with a carbon number of C23 to C31. It's odd number carbon chain. And also, in addition to hydrocarbon, it makes a kind of epoxide of this. And also this kind of long chain phenol, and it's epoxide too. And as you know, epoxide is very active substitution group, so then it makes a kind of linkage of the condensation of this hydrocarbon to make this higher molecular weight lipids. And also, this is a so-called botulyl. It's a member of aldehyde, but you can see the similarity of structure of the linear alternate chain. And when the radio-labeled uric acid was fed to this alga, the radio-labeled alkane was accumulated. So that means that this could be the precursor. Then this is the kind of hypothesized mechanism. Probably the uric acid or other fatty acids elongated by acetate unit like this up to C32 fatty acid. Then at the final stage, the carboxy group could be eliminated as carbon dioxide. Maybe the beta hydroxy group should be used as eliminate group. Then finally, the hydrocarbons with terminal double bond and one carbon number less hydrocarbons is generated. But still, this mechanism is not clear. And also, this is the biopolymer of this areas. And the French group determined this structure, and it's also composed of the C32 aliphatic aldehyde, the condensed macromolecules, so-called arginine. And this is also derived from uric acid as a precursor. Probably C30 dialdehyde is produced, and this is condensed, make a condensation, and to make this huge molecular weight compounds. But still, the mechanism or enzymes concerned with biosynthesis of these compounds has not been characterized yet. But this long chain aliphatic aldehyde is also a very key molecule because it is made from the carbon dioxide fixed by this alga. So then this is a kind of flow of carbon dioxide fixed by photosynthesis in the A race of Botulcoccus. But the flow is not so complex, simple, rather simple. At least carbon dioxide is to be changed to convert into fatty acid, and the fatty acid further converted alkane and secret outside as a hydrocarbon. Or fatty acid is converted to long chain aldehyde or polyaldehyde and make a condensation to make an extracellular matrix. So that means that as far as fatty acid is produced, this alga is happy. Okay, but it doesn't work. Okay. So, but on the other hand, the B race, the produced triterpene is more complex. because you can see this is a excluded hydrocarbon, but this is not composed of a single component, but a mixture of many variety of homologue of botulocosens. And those homologues are produced by from this parental compound C30 botulocosen, but this is maturated up to maybe C34 compound, or during a maturation process, the carbocation is generated, and at the quenching stages, it makes a cyclization, and some cyclic botulocosen is also produced. One of my students who graduated your institute are now working as a kind of PhD student to characterize this unique methyl transferase now in my laboratory. So he will present his research tomorrow, a kind of online session. Unfortunately, I didn't have money to take him. So that he is attending as an online speaker. But if you are interested, please join his presentation. So then botulocosen is methylated like this. But quite interesting, I mentioned that this botulococcus B race also accumulates small amount of methylated squarings. And the manner of methylation is quite similar to botulocosen, like one methyl group here, or two here, or here, and 3 or 4. So that means that, but this is a major component, 90% of hydrocarbon fraction, and this is less than 10%. So that means maybe those methylated squaring is a minor component, so that means maybe just a byproduct of the production of these botulocosens. But when I did the characterization of new strains that isolated from Japan or anywhere, I realized the color is different. The A race producing linear hydrocarbon is also, this biopolymer part is transparent, colorless. But the B race making triterpenes is a yellowish color. So what this color is, then I did the structure determination of this compound. So then I could identify the new carotenoid and specific to these organisms. One is botulocoxanthine A, and this is branzancin. As you can see, this is a carotenoid part, but you can see this is a derivative from tetramethyl squaring. And also, this is a long-chain phenol. This was considered as a specific compound to the areas of the Botrycoccus. But this discovery of this compound means that the B race also can produce this kind of long-chain lipids too. But anyway, those tetramethyl squaring derivative could be tetramethyl squaring 10, 11 both sides. And the French group also discovered this compound in the B race of Botrycoccus. And the French group also determined the chemical structure of this biopolymer of the B race. And the biopolymer is basically the similar structure as the biopolymer from A race. This is just a conjugated macromolecule of polyaldehyde. But in some part, this tetramethyl squaring diapod side is incorporated. So that means long-chain aliphatic aldehyde and also methyl squaring derivatives is used to make this biopolymers to make colonies in this B race. But on the other hand, botulocosen type triterpene is a major component, but still this is only one example of the kind of derivatives of botulocosens. So that means the botulocosen is just existing as a free hydrocarbon, but not used as other derivatives. So then question is a biology function of botulocosens and methyl squarings are maybe different. So then this is the flow of carbon fixed by photosynthesis in the B race. It's more and more complex if compared to A race. So carbon dioxide should be converted to fatty acid to make a polyaldehyde to make colonies. But on the other hand, B race needs to make a botulocosen and squaring. And as I mentioned earlier, squaring is an essential component to make a membrane sterol. So that means it's necessary as a primary metabolite too. So that means this alga needs to make a botulocosen and squaring. That means the carbon dioxide should be converted into the precursor of isoprenoids. But then botulocosen is a major product, but the squaring is produced but split into primary metabolites or secondary metabolites. So how this carbon flow is regulated by this alga? That's my question. Then I decided I wanted to identify the botulocostin synthase. But nowadays, it's very easy to determine the genome DNA information of one organism. But by reading genome DNA, it doesn't give us answer what kind of protein is encoded because of the presence of exon and intron in the eukaryotic organisms. But if we can know the messenger RNA sequence, that directly suggests as amino acid sequence of protein, then we can predict the function of that proteins. So then I started the cDNA cloning of this botulocostin synthase. Then I started my research about this. The information of squalene synthase was available because squalene synthase is very important for the biosynthesis of sterols. So then squalene synthase is an enzyme that can catalyze two-step reactions by one protein. So the first half reaction is the condensation of two molecules of farnesylate diphosphate to make an intermediate prescreen diphosphate like this. Then second half reaction is elimination of the diphosphate group and reduction by NADPH and rearrangement of carbon-carbon condensation to 1' to 1 linkage to make these squalene molecules. So then if we compare the chemical structure of squalene and botulocostin, as you can see, this molecule is made of the condensation of two farnesyl residues, but the way of linkage is different for squalene. This is a 1' to 3 linkage condensations. But as you can see, the prescreen diphosphate is always has a 1' 3 linkage. So that is once prescreen diphosphate is produced, botulocostin can be produced. So then my hypothesis was if there is a botulocostin synthase, it should be very close enzyme to squalene synthase, but some part that's concerned with the second half reaction should be different. Then I tried to isolate the cDNA code encoding for the squalene synthase-like genes. So this is a kind of schematic diagram of protein amino acid sequence of squalene synthase. This is a translation starting start methionine, and this is the amino acid sequence. And this is the nicotiana tabacum with the tobacco, Arabidopsis corn, rat animal, and Saccharomyces is yeast. So those are very different organisms, but they share some part of the amino acid sequence of squalene synthase. They have very conserved amino acid sequence, so-called functional domains. The domain 1, 2, 3, 4, 5. And the squalene synthase has this part is not so conserved amino acid sequence, but composed of the continuous, very hydrophobic amino acid sequence. So that means that it's a hydrophobic region. Then squalene synthase is tethering in the ER endoplasmic reticulum membrane as a membrane binding proteins. So then, by doing the point mutation analysis, this domain 3 and the domain 4 is important for the first half reaction. If something changed in the amino acid sequence of this part and this part, this first half reaction did not occur. But on the other hand, this domain 5, there is a conserved amino acid sequence, but some amino acid residue is replaced. The first reaction can be processed. But this second half reaction, conversion from PSPP to squalene was stopped, and the accumulation of PSPP was observed. So then we need to return to my hypothesis. If there is a botulocostin synthase, it should be very similar to the squalene synthase in terms of the amino acid sequence. But maybe the some part concerned with the second half reaction should be different. So that means that I tried to find some gene encoding for amino acid similar to squalene synthase, but we're having different amino sequence here. But at that time, there was no good enzyme assay system to detect the botulocostin synthesis from FPP. So this is FPP is a water-soluble compound, and this is a radio-labeled FPP. And the Botuliococcus homogenate or some E. coli expressing recombinant protein, the homogenate, is mixed with this substrate and cofactor. But as I mentioned, squalene synthase is a membrane-binding protein. So that means that it's necessary to add detergent to enhance enzyme activities. So, but then if the botuliococcine or squalene is produced, they are hydrophobic, and this should be recovered in organic solvent like hexane. Then by doing this two-layer partition, those reaction products could be recovered and developed by TLC, separate botuliococcine and squalene. And if there is a radioactivity, researcher can detect enzyme activity. But under this condition, researcher could detect the formation of squalene from FPP, but not botuliococcine. So then, when I teach my student, I ask student to please do the experiment very seriously. Don't be rude. But I was so lazy because if I added detergent, this separation by two layers is very difficult and time consuming. So then I omitted detergent. Then suddenly it shows the botuliococcine activities. So that is a big progress. So and I was quite lucky. So then actually the FPP could be the direct precursor for both of botuliococcine and squalene. Then I could do the enzyme assay. Then at first I could get the first clone cDNA from botuliococcus, and it has a partial amino sequence represented here. But as you can see, this is almost similar amino acid sequence with other squalene synthase. Then actually, I use the data system. Then I could detect only squalene synthase activity, not botuliococcine synthase activity. So that means that this is a conventional squalene synthase from botuliococcus. Then I obtained the second gene that's also encoding for the squalene synthase-like protein. And you can see, at first I named this botuliococcus botuliococcine synthase, BBS, because as you can see, this domain 5 is quite different from other other squalene synthase. So that means, okay, this could be the real candidate for botuliococcine synthase. Then I named this. But unfortunately, or fortunately, I was forced to change its name from botuliococcine synthase to squalene synthase-like protein 1. Then, but unfortunately, when I made the recombinant protein by using E. coli, no enzyme activity, botuliococcine synthase activity, squalene synthase activity was observed. And this is a result, the same assay. When I use the botuliococcus homogenate, time up. Sorry. Okay. Then I just move on the conclusions. Okay. Anyway, I could find new enzymes that make botuliococcines. But it's a kind of a mixture. Two enzymes is a combination. Two enzymes necessary. So that means the botuliococcus has a conventional squalene synthase, and this one that can produce only presqualene pyrophosphate. But this SS2, SS3 can use not FPP, but PSPP as direct substrate and can make a botuliococcines. So then I'll skip, but still biological function is unknown. Then I'll skip some size. But now you can know, still there are some unique unknown enzymes that may be used for the biofuel productions. I want to show another example for tetraterpenoids. But now we could get the gene. So that means maybe we can get some answer why this organism needs a lot of hydrocarbons. And at this moment, this algae grows very slowly because the production of energetically high-value product. But then it interferes the applications. But if we can turn on the switch of the biosynthesis by manual or artificially, maybe we can let them grow much faster, and we can fill the algae in the pond. Then again, maybe we can turn on the switch for the biosynthesis. But at this moment, genetic engineering of this algae is not impossible. So that means this is my expectation to new younger generation to develop such kind of strategy to make a biofuel production by microalgae. So, thank you for your attentions.
Okay, thank you so much, Professor Okada.
Still question time? No? If not, I'll take
Yeah.
question at the coffee break.
Yeah, I know that, but maybe we have one question.
Mhm.
If you have any question for Okada Sensei? There's no question? Okay. So actually I have one, Sensei. Maybe this is the simple things. I still remember when I visited your laboratory in Tokyo, and I saw a lot of, algae. And I believe that Indonesia has also a lot of, extreme microorganism, like in the Kelut Mountains. And can you suggest us, all of us here, we don't have any expertise like you, but I believe that if you can suggest us how we can identify the simple way to identify that the microalgae have potential to produce hydrocarbons.
One answer is just I showed in slides. If you stained with the algae with this fluorescent dye, and if you can see a lot of fluorescence, that's a sign. But proportion is we cannot know if it's a triglyceride or hydrocarbons. So that means that you need to propagate it a little bit and extract and test by GCMS or something. Or maybe just simply develop with silica gel TLC plate, and if the sun spot is moved by solvent with only normal hexane, that's a sign it's not a fatty acid but the hydrocarbons.
Okay,
Yeah.
thank you so much. Okay. Yeah, please, sort. Yeah.
Mm-hmm.
Hybrid catalyst, combining algae with, like, say, electrocatalyst or inorganic chemistry. Is that kind of like the way to modify to get the selectivity as well?
Yeah, that's a good point. Yeah, but unfortunately, this organism grows so slowly, and not so many scientists are interested in. So that means I want to have a collaboration with a different area, field of science. Yeah, and also maybe there's some idea, and quite new idea, by collaborating with a different field of scientists. Maybe new technique to introduce the exogenous gene into cells. Yeah, that's true.
Yeah, thank you so much. And please give applause, a big applause to Professor Okada. And I think this is the end for the first session, right? So the coffee break is waiting for us. Thank you so much. So I give it back to MC. Yeah.
Okay, thank you so much, Okada Sensei, and thank you so much, Mrs. Fatma, for already moderating the session itself. Can I get one more round of applause from the previous sessions, please? Thank you so much. Before we continue to the next keynote sessions, we are going to have a quick break where all of the guests and participants can enjoy snacks and drinks that we have provided outside of this hall itself. Please enjoy the coffee break for the next 10 minutes, and please be back to the hall at 10:30 a.m. Thank you so much. All of you guys could have a coffee break. Thank you.
Audio Jungle.
So I'm from the Advanced Membrane Technology Research Center, at University Technology Malaysia Singapore.
So, like I showed just now, we have the operating temperature of the PMFC up to 120 degrees C. So we try to find out or to determine whether our membrane is capable to withstand with this temperature, right? But in terms of the gas evolved, we never try with this TGA approach in order to determine what type of gas evolved during the operation.
Catalytic conversions. Before we start this process, also prepared previously for the catalyst. In this research, we use 0.08 in molar ratio alkaline catalyst, namely potassium hydroxide.
My name is Heriberto Diaz. I studied in my bachelor and master's of science in Mexico.
Assalamualaikum warahmatullahi wabarakatuh. Terima kasih semua. Alhamdulillah acara Isoc saat ini lancar dengan baik dan mudah-mudahan menghasilkan ilmu-ilmu yang bermanfaat dan dapat dipublikasikan jurnal yang lebih baik atau posting yang lebih baik. Mudah-mudahan pada kesempatan yang akan datang dapat diperbaiki untuk memperbaiki mutu dari penampilan dan kualitas dari acara ini. Terima kasih atas bantuan para pimpinan ITS, kemudian Dikti yang memberikan pendanaan juga, kemudian departemen yang memberi fasilitas dan fakultas, dan teman-teman semua. Saya mengaturkan terima kasih yang sebesar-besarnya sampai terselenggaranya Isoc ini berjalan lancar dan tertib. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Learning process of bachelor degree. In the pandemic, learning is generally carried out by Zoom. Even though it is online, various learning methods in delivering courses can still be done, such as providing one-way material, discussions, group discussions, and doing practice questions. Delivery of lecture material is carried out using either a PowerPoint.
Ya, gitu ya. Jadi kalau kita makan lemak ya, misalkan gajih gitu ya.
Molekul persatuan volum ikan-ikan.
Explaining from textbook sources.
Konsentrasi A dikali AR ini kan, tetapi ini berbeda karena ada yang mengurangi sejumlah KR absorb tadi ya.
As well as adding other sources that can enrich the subject. For example, in the subject of atomic and molecular structure, students are shown the website chemtube.com, which visualize several types of molecular orbital to make students become more aware of symmetry that are difficult to imagine. In the subject Modern Physics, the lecturer also introduced students to use EduGame in gamelab.mid.edu to help students understand about the relativity concept.
Nah, warnanya kalau kita perhatikan itu kayak ke arah
the subject structures, properties, and reactivity of inorganic compounds. Lecturer asks students to make the molecule model using plasticine when explained about the molecule symmetry concept. Lecturer also developed the web from the medium to deliver the subject content as performed in the subject chemometric. Some subjects also add the external exposure to introduce students the subject application. For example, in the subject marine chemistry, students go to the sea to see various marine species. Then they discuss the relation to the subject. This activity was done in 2019 before COVID pandemic. In the pandemic, the external exposure is replaced by the virtual external exposure. In the subject chemistry membrane, the lecturer invite the industry practitioner to sharing about the problem of membrane application in industry, and the students are invited to discuss the solution. In the subject research methodology, students are required to be more active in exploring chemistry literatures and writing it down in the form of manuscripts, oral presentation, and posters. Guidance on how to search literatures is done in practice, directly by showing several websites related to searches on chemistry. For subjects that require students to have the ability to calculate, exercises are carried out and discussed carefully. Implementation of assessments are carried out in various ways, such as conducting tests via My ITS Classroom and supervision using Zoom, or giving other tasks that can increase learning outcomes in the form of skills. For example, subjects biochemistry and foodstuff chemistry have assignments that are required to be submitted through the TikTok app. Subject chemical literature gives out assignments of video presentation, example, video, and poster. During this pandemic period, practicum is carried out either with demonstration in the laboratory with the help of lab assistants, or some practicum are developed with an online module. The following is a video footage of the Chemistry 2 Basic Technique Laboratory. Measurement of density. Solution.
Mari kita lakukan percobaan 3.
Thermodynamics. Calorimeter bomb. Binary solution. Freezing point.
Kita akan melakukan praktikum termodinamika yang membuat massa melakukan dengan menggunakan metode
Chemical dynamics. H2O2 decomposition. Acetone iodoform reaction. And synthesis organic reaction practicum. Styrene polymerization.
ilmuwan, walaupun ada yang offline, ada yang online, sehingga acara ini hybrid. Tetapi secara garis besar kita dapat mendapat pencerahan, ilmu-ilmu baru.
Audio Jungle.
So I'm from the Advanced Memory Technology Research Center. I'm at the University Technology Malaysia Singapore.
So, like I showed just now, we have the operating temperature of the PMFC up to 120 degrees C. So we try to find out or to determine whether our membrane is capable to withstand with this temperature, right? But in terms of the gas evolved, we never try with this TGA approach in order to determine what type of gas evolved during the operation.
Catalytic conversions. Before we start this process, also prepared previously for the catalyst. In this research, we use 0.08 in molar ratio alkaline catalyst, namely potassium hydroxide.
Well, my name is Heriberto Diaz. I studied in my bachelor and master's of science in Mexico.
Assalamualaikum warahmatullahi wabarakatuh. Terima kasih semua. Alhamdulillah acara Isoc saat ini lancar dengan baik dan mudah-mudahan menghasilkan ilmu-ilmu yang bermanfaat dan dapat dipublikasikan jurnal yang lebih baik atau posting yang lebih baik. Mudah-mudahan pada kesempatan yang akan datang dapat diperbaiki untuk memperbaiki mutu dari penampilan dan kualitas dari acara ini. Terima kasih atas bantuan para pimpinan ITS, kemudian Dikti yang memberikan pendanaan juga, kemudian departemen yang memberi fasilitas dan fakultas, dan teman-teman semua. Saya mengaturkan terima kasih yang sebesar-besarnya sampai terselenggaranya Isoc ini berjalan lancar dan tertib. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Learning process of master degree. The teaching methodology in the MOC is conducted by various methods. The conventional explanation by the lecturer followed by the class discussion. The courses that adapt this method are thermal analysis.
Di thermal analysis nanti kita akan belajar tentang yang pertama nanti ada TGA seperti ini sebenarnya gambarnya, kemudian ada TTA seperti yang di atas ini, kemudian yang di tengah ini adalah DSC, yang ini TMA, dan yang bawah di sini adalah DMA.
Research methodology.
Lebih spesifik ya dibandingkan manfaat, karena ada yang tidak tahu juga.
Membrane synthesis.
Yang paling mudah dibuat itu menggunakan inversi fase ini. Metode-metode yang lain ada, nanti kita akan bahas juga metode-metode yang lain, meskipun metode yang lain.
Natural products chemistry.
Gamanya yang dekat A ini alfa, yang di puncak atas itu beta, di bawah gamma di sini. Jadi gamma-gamma, dua gamanya. Dimetil, tadi siapa yang nanya ada dimetil dihidroksi, ini dua metil, dimetil.
Quantum chemistry. For quantum chemistry, students are required to have the deep concept understanding and ability to derive several equations. The video showed that lecturer was direct the students to derive several equations and solve several problems. The study based on the recent article in the journal that has a relationship with the topic. The article-based study as an additional source explained by the lecturers. Discussion topic or presented by the students to develop concept and build the knowledge. Here is the courses that adapt this method. Structure and analysis of surface. The recent research on the application of structure and analysis of surface is studied based on the recent publication. For example, the study of scanning electron microscopy to identify a non-edible oil seed as energy corps. Molecular reaction dynamics. In addition, the explanation based on article-based study. The lecturer also give the assignment for student to study the article.
Telah disajikan 3 artikel mengenai spektroskopi Doppler, yaitu mengenai aplikasi terkini untuk mengamati perkembangan
porous material.
Warna tekstil dihasilkan banyak oleh industri, di antara industri pabrik, industri tekstil, industri pewarna tekstil, dan industri produksi batik, bahwa dimodifikasi dengan reos, kemudian dilakukan
membrane synthesis.
Dimulai dari introduction overview dulu, yaitu teknologi membran yang muncul sebagai teknologi inti di bidang pemisahan. Teknologi membran juga diterapkan secara luas di berbagai bidang, terutama untuk pengolahan air, limbah, industri, farmasi, makanan, bioteknologi, serta lain-lain.
Develop students to have the ability in research design through the course micro
beverages or food that you wanted to eat, you are allowed to do so still. Okay, welcome back to the 6th International Seminar Chemistry 2024 sessions, plenary sessions, or Isoc 2024. This morning, we're still gonna have a few sessions from our amazing speakers, our keynote speakers, and we are hoping that all people here can sit tight and enjoy the session itself. And without further ado, I think we could go to the next keynote sessions, our second and third. And these sessions will be moderated by Mr. Wahyu Prasetya Utomo, PhD. For Mr. Wahyu, the time to moderate is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Let me introduce myself first. I'm Wahyu Prasetya Utomo, the one who always troubling you with all of the emails relating to the Isoc. And thanks for your kind response for our email and information. And finally, we meet here in Isoc. Thank you for your participation in Isoc again. Thank you very much. This time, I would like to serve as a moderator for the second speakers from Professor Yoon Hao Eung. Professor Yoon Hao Eung is a professor in the School of Energy and Environment and the director of Low Carbon and Climate Impact Research Center, or LCCIC, at the City University of Hong Kong. He received his PhD from Osaka University under supervision of Professor Michio Matsumura in 2009. After a brief research to the Radiation Laboratory at the University of Notre Dame under Professor Prasan Kamat's group, he joined the ARC Center of Excellence for Functional Nanomaterials led by Professor Amal Rus at UNSW, Australia. He joined City University of Hong Kong in August 2018. His research is focused on the development of novel photoactive semiconductors, either particles and thin films, for sunlight energy conversion. He was awarded the Honda Fukushima Prize in 2013 by the Electrochemical Society of Japan in recognition of his work in the area of photo-driven water splitting. In 2019, Dr. Eung was awarded as the APEC Science Prize for Innovation, Research and Education, or Aspire Prize in Chile. He is also the recipient of the Japanese Photochemistry Association Kataoka Lectureship Award for Asian and Oceanian Photochemist 2021. And also, additional information, it is an honor for me to have my PhD under his supervision. Thank you, Professor Eung, and the time is yours.
All right. Okay. Thank you, Wahyu, for your very generous introduction and remarks. Good morning, everyone. First of all, thank you, the organizing committee of Isoc. Thank you, ITS. Thank you, Department of Chemistry. Thank you, Professor Freddy. And thank you for everyone to stay here in the room. My name is Yoon Hao. I'm currently working in Hong Kong at City University of Hong Kong, as Wahyu mentioned. And well, I am a Malaysian. I did my, as mentioned by Wahyu, I did my first 2 degrees in Malaysia. Then I moved to Japan and then to Australia and now in Hong Kong. Well, my presentation is on solar fuels, and my presentation or my sharing is will not be as in-depth as Okada Sensei in the previous session. My one is more like an overview to give you an overview what we have been studying, we have been investigating for the past 20 years, around 20 years. And I would like to mention that I would like to highlight Wahyu, Dr. Wahyu Utomo is our latest, not the latest now, but one of the latest PhD students graduated from our team. And he's my first PhD student in Hong Kong. I'm glad to work with Indonesian community for a very, very long time. And my supervisors are Indonesians. And thank you. I would like to keep on a very close touch with Indonesian community. All right, and today, the sharing that I'm going to make today, they are all a collective efforts from our members, either it's current members or past members. So for the current members today, very exciting. A few of our members are very they're very keen in visiting Bali to visit YU as well. We have Dr. Phoebe Chong, Ms. Liu Rui, and Chili. They are all going to make an oral presentation in the parallel session in the afternoon. And the sharing that I'm going to make, part of the results are coming from them. So if you are interested, please stay tuned. All right, my work is on solar fuels. We use light coming from sunlight or artificial light from indoor. And we try to make chemicals that can be used as fuels and through the catalytic conversion. And to be more specific, most of the works are directly associated with the light illumination on the catalyst. It's different from Okada Sensei's domain. We are not working on the biological science, and we are working on more on the inorganic chemistry, involve catalyst development, which is the materials chemistry. And when we study the electron transfer to make the fuels, that would be physical chemistry and electrochemistry. So the content that I'm going to share with you quite align well with the theme of this Isoc seminar. All right, in brief, solar fuels, I would like to sort of like highlight. We are working on hydrogen predominantly. So this is hydrogen molecules, all right, H2 here. In the part that I'm working now in Hong Kong and previously in Australia, hydrogen is something that people talk a lot now. And because both regions, both countries, we have the roadmap for the carbon neutrality. We want to achieve carbon neutral by 2050, by 2060, and so on. We have a target. So everyone is quite excited or quite fascinated by how hydrogen might be potentially contributing to the carbon neutrality effort. But, well, actually, hydrogen is not new, and it's not new at all. We have been using things coming from the hydrogen or process that is coming from the hydrogen, involve hydrogen, for a very long time. For the ammonia synthesis, as what Okada Sensei said, we have the Haber-Bosch process, making ammonia in the process of making fertilizer. And we also use hydrogen to make, let's say, metal and petrochemicals refining. And we even use hydrogen to make food, margarine, for example, the hydrogenation. We use hydrogen. So we have been using that for, well, very long time. It is not new, but we are fascinated by this new role, potential role, which is relatively, or not relatively, is rather underexplored, is that we want to use hydrogen as the energy carrier to be used as the transport, like fuel cell car, fuel cell vehicles. And we want to use hydrogen for heating purpose, to put it in the city gas that you use in your household. And we are exploring to use the hydrogen to make electricity, not natural gas, but combusting hydrogen. The challenges are different from the traditional role of the hydrogen. Usually these kind of functions, we produce the hydrogen and we use it right away. So it's on-site. There is no need or no urgent need for transportation, transporting the fuel, hydrogen. Like for example, if we want to use hydrogen in the vehicles, we need to have the mechanism, we need to have the hydrogen to be transported, to be transferred from the point of generation and then the point of utilization. And for the heat as well, there are many countries, including Australia, including UK, we are trying to have the hydrogen to blend into the natural gas, the gas mixture in the city gas pipeline. So, well, this is a fun fact that is quite new for me. When I joined CTU in Hong Kong in 2018, I realized that in the city gas, we call town gas in Hong Kong, in the town gas that transport the gas mixture to our household, there is 50% of the hydrogen in the gas mixture. And there are only 2 cities in the world that the city gas that contain so high hydrogen concentration. One is Hong Kong, another one is in Singapore. And when we look at the other examples, like most of the cities exploring the city gas to blend in with hydrogen, we are looking at blending it with a 5% of hydrogen, 10%, at most 20% before the pipe that we can use can sustain that content, because hydrogen is so small. It will have the embrittlement issue. When you have more hydrogen, it breaks through the pipeline that you build, and then that's the safety hazard. But in these two cities, they have been using that for many decades. I think that makes these two cities a quite interesting case study scenario for many people working in this area, because the gas mixture itself, they purposely have the carbon monoxide inside, a very tiny volume, so that will do the shielding on the pipeline, and then that prevent embrittlement to occur. All right. I'm aware of time. I spent too much time on the introduction, but still one more page on introduction. So while hydrogen, we want to use that because when we use hydrogen, it is clean. You combust that, it gives you H2O and the energy in the form of heat or in the form of electricity if you use that for fuel cell. However, when we produce that, it is not clean at all as what we are, the technology that we are using. So in our community, we like to name them with the color. We have gray hydrogen, blue hydrogen, turquoise hydrogen, green hydrogen, and in fact, the color scheme go on. We have rainbow color, we have white hydrogen, yellow hydrogen. It is so mouthful, so inconvenient, and we are not going to use that anymore. But for the sake of convenience, for the discussion, I still use the green hydrogen here. So we produce hydrogen mainly from the current practice is from methane, from coal, using the steam methane reforming or coal gasification to get the hydrogen and release a lot of carbon dioxide, very intense concentration or amount of carbon dioxide. So it's well for the application, nothing wrong. But if the purpose is to achieve the carbon neutrality, then that is not helpful. Okay, because we are generating a lot of carbon footprint too. So the community think, well, we can move on if we use the same technology, but then we capture the carbon without releasing that. We call that blue hydrogen. And there's also technology that coming from the methane pyrolysis that turn the methane directly into the carbon deposit and hydrogen. There's no release at all of the carbon dioxide. And then the green hydrogen in general, the meaning is you use the renewable electricity, either it's coming from the from solar panel or from the wind turbine. You get the electricity and you drive the electrolysis, water splitting. So if this one is possible at large scale, so this will give us the hydrogen with zero or very low carbon footprint in principle. And this is what most of the community is working on. So you see a lot of examples in demonstrating this idea, and it's getting larger scale and larger scale. This one is in Fukushima, all right? When it was built, it was the world's largest green hydrogen facilities. You can see a large array of solar photovoltaic here, coupled with the electrolysis facilities, and then with the storage to get the hydrogen we call green hydrogen. But the record is going to be broken from time to time. And now in Saudi Arabia, in Neom City, they are building the much larger one. When it is built, then it will be the world's largest green hydrogen facilities. And then China is also making in the northern part of the China. When it is built, it will be the world's largest. So you see the demonstration of this idea, and seems like green hydrogen we will be seeing this example more and more. However, I would like to, we are working on this as well, but fundamentally, well, at the university, I think we are allowed or we are encouraged to think further for solving problems or creating problems to be solved and so on. All right, so, well, water splitting, we need water. To get 1 kilogram of hydrogen, you need 9 liters of hydrogen, of water. And, well, there's no problem. If you want to produce a kilogram of hydrogen, you have no problem with water for sustaining to power up your one building, your room, a few buildings, a few blocks, probably is no problem. But if we are looking at the power generation for a whole city or whole district, how much of water that we need? And for the electrolysis, we need clean water, not just normal water. They are at the deionized grade, they are at the distilled water grade. Because the catalyst and the electrolysis, they are very sensitive to the pollutants, or especially the organic pollutant. Most of us here, we have chemistry background, so you know that they use the precious metal. When the water is not clean, there will be poisoning the electrocatalyst. So for this demonstration, if we are serious about building a power plant scale of the hydrogen, green hydrogen, the water that we are going to need will be huge. And we also know that water, clean water, is a very precious resources. We use that for agriculture activity to grow crops. We use water for drinking, of course. Then we also want green water or clean water for produce energy. So there's a huge challenge for us to think about. So then our work involved a little bit further, slightly different, is that green hydrogen not necessarily coming from renewable electricity. It can be, as what has been shared earlier by Okada-sensei, artificial photosynthesis. Like this one is just screen cap from the Department of Energy from US. They look at the way of producing hydrogen. There's a roadmap or the timeline for the hydrogen. So the most applicable or most ready to be scaled up is, this is what we have been working on. The industry has been using natural gas reforming to get hydrogen. And move on, there will be electrolysis driven by solar, driven by wind. This is what everyone is in this stage now. We use the electricity to drive the water splitting. Okay, but that is not the ultimate term. This is kind of like the mid-term to long-term, but ultimately, if possible, we get rid of the electricity part. We directly, this one is the STCH, solar to chemical hydrogen. There's no electricity as the intermediate part. And this is what we are trying to learn at the laboratory as well. Of course, we are studying this one. It's more when we look at the translation, when we look at the applications of the technology, we want to demonstrate this one as well. But at the lab, we study this one. All right. So this is a very simple, simple slide to illustrate how the photosynthesis works. When we have green plant, it has the catalytic centers inside, and basically it is doing the redox reaction, reduction and oxidation. So when we have the carbon dioxide, when we have water, the water is reduced, we get these hydrogen. Or when we want to reduce the carbon dioxide, we get hydrocarbon. We get like the green plant, how they make the food and so on. So we are trying to mimic this process. Is using photocatalyst. Photocatalyst is not new. It's not new at all, and it's very active research in Japan and many parts of the world. So imagine that this is a photocatalyst, is a semiconductor. It is defined by two distinct bands. We call this one conduction band and valence band. This is the band gap. This is the energy gap. When we have the energy coming in this case, will be coming from the light. It can be from sunlight, can be from the indoor light. If the energy is sufficient, it will bring up one electron to the conduction band. And when it is in the conduction band, this electron can move. When they are in the valence band, they cannot move. They're quite static there. When they go up with the light, excited electrons go up to a conduction band. Now they can move. If they can move far enough, they may be able to reach the surface of the catalyst. All right, and then if they reach the surface, then if they are able to transfer to the reactant, in this case is water, if they can transfer, then we can have hydrogen. There's also an if there. All right, these electrons, if they can move far away, or if they can move fast enough, if they don't, they will come back. They are not stable. So we call electron lifetime, live how long the electrons can stay. So in the fundamental understanding, not to say manipulation, but just to understand, we are quite interested with this kind of phenomenon, how fast the electron moves, how far it can move, and how long it can stay. All right, so then it's actually the principle of the reactions is just a redox reaction. You have a chemical, they can be reduced, they can be oxidized. In this part of my presentation, mostly would be on water splitting. When we have the reduction of water, we get hydrogen. And when we talk about water splitting, in most cases, we talk about oxygen. H2O, water splitting, you get hydrogen, you get oxygen, which is a very challenging reaction. Water is pretty stable. We want to split them. But even if we do that, this oxygen, sometimes this is the rate determining step because it involves 4 electron transfer here. And even if we have the oxygen here, it may not be the very ideal product because, oxygen is relatively lower value, relatively. And with the presence of hydrogen, oxygen here, they might be mixture. Then we have the safety hazard when the hydrogen and oxygen mix. Then you heard cases of the accidents. They have a explosion hazard here. So we have been thinking, if instead of 4 electron transfer, if we can modify the catalyst, we force 2 electron transfer, not 4 electron transfer. We do not want to oxidize the water completely. We just partially oxidize it. So we can get hydrogen peroxide. Now in hydrogen peroxide, it is chemicals. It can be used in industrial activity. It can be also used as fuel. There is a hydrogen peroxide fuel cell now as well. Of course, it's at the preliminary stage. And another advantage is that now we do not get gas of oxygen. We get the water or liquid state. So we are minimizing the safety hazard, the hazard risk of this process. This is what we have been working on. And Dr. Phoebe Chung in the afternoon will be sharing part of this research in the materials, in the catalyst design as well. And then of course, when we look at the redox reaction, if the reactant is carbon dioxide, then it is quite a fascinating chemistry there. Starting from a carbon dioxide, of course, the possible reactions are very lengthy. And if it involves 2 electron transfer, we get gas phase carbon monoxide, which is in the syngas component, which is quite useful as well if we can selectively get 2 electron transfer and get carbon monoxide. If we somehow can regulate the process to have 8 electron transfer, we get methane. And these 2 products are in gas phase. If not, we can tune if we have a 6 electron transfer, 10 electron, 12 electron transfer, we might be able to get liquid product, like say methanol, ethanol, and so on. So, well, unfortunately, we have, fortunately, we have materials that can do this. Unfortunately, we have the materials that can do a bit of everything. So in your CO2 reduction, you get a bit of everything of these products, which is not ideal because we have to separate the product. For the industrial process, it is unlikely or it's not favorable. So it becomes like the scientifically meaningful if we can have materials that we can tune the selectivity by having just one process and you have a good selectivity. And we are learning that and the community is learning that too. And then the last one is a nitrogen reduction. Is a very stable chemicals. And again, we are interested to produce ammonia because people have been thinking ammonia as the carrier for hydrogen. It is relatively easier to be transported compared with the hydrogen itself. So the community is working on ammonia. And well, same thing, if we can regulate the electron transfer, the number of electrons, we can tune if we want to have the hydrogen. Hydrogen is chemicals, liquid state chemicals, which can be useful as well. So not looking at application, but just looking at the fundamental things, how to regulate these things from the catalytic process, I think is pretty meaningful and very educative as well. All right, so, sorry. Oops. Sorry. Can you please help to click from, thank you. All right, so this is a bit of boasting. Please don't mind. Well, we work on photocatalyst development. We make photocatalyst they have a different color, they have a different functions. One is a highly oxidative. We have a bismuth-based photocatalyst. It is good for oxidizing because of the very deep valence band. Then if we have water, we can oxidize it. We have organic, we can oxidize it. All right, but the conduction band is not energetic enough. Electrons cannot be used to reduce the proton in this case. But on the other hand, we have another photocatalyst that it is very good for the reducing power. It can reduce many things, but unfortunately, the host itself is not very oxidative. So, well, by separating them, they are not very useful because they are short of the reducing power. For this one, it's short of oxidative power. But by combining them, then we are talking about, it's more like the artificial, the photosynthesis process. So these electrons and holes, they can be recombined because they are not very energetic on itself for the redox reaction, but we can maximizing these holes, we can maximizing the utilization of the electrons to give us the meaningful redox reactions. As mentioned, we have spent efforts, we have spent time in understanding how these things happen when they are generated, how far they can move. This is the charge diffusion, the electron diffusion length. If the diffusion length is too short, can we make the catalyst small enough so that the size of the particles, the size of the catalyst is smaller than the diffusion length? That means the electrons can go to the surface. Otherwise, if your particles is very huge, the electrons upon generation, it has no chance to go to the surface, then there's no activity. All right. Another one is electrons, how fast it can move, the electron mobility. We want it to move faster or shorter. And the lifetime, how long they can live. So, well, we spend efforts in understanding them using a combination of spectroscopy technique, time resolve technique to look at the lifetime, and so on. And our team, for the last many years, mostly working on hydrogen, water splitting, but now we spin up to the reactions including the ammonia. And Ms. will share with you in the later part of this in today's program. All right, so for this kind of like the fundamental exploration, I just summarized that in one slide. So while we make catalysts, we want to know how it looks like, and they are crystal, so they have the crystal facet, they have the crystal phase, and every single different crystal facet, different phase, they have a different property for the charges to move around. All right. So with the catalyst, then we can look at how the resistance, the conductivity, when they are illuminated. So when you turn on the light, you see that some are brighter, some are darker, to show that where the electrons are moving to. And for example, this is what we have been looking at. Okay, and then we have a characterization. This is a time-resolved microwave conductivity to look at the lifetime of the charges, how long they can live. These are the meaningful things that we have been working on, we've been studying. So, well then, because this involves some of the electrochemistry, this is semiconductor. In semiconductor chemistry, we look at the band bending as well. This is the potential. We can control the band bending. This is a very steep bending. This is a relatively mild bending, so we can control by having the different bending. We sort of like can control the number of electrons that can be transferred to the reactant, and we use these to regulate the selectivity, and so on. And of course, we spend efforts in developing the detection tools to measure the products. So while this one is hydrogen, this is oxygen. And in many cases, when in the community, when we report a hydrogen from water splitting, we need to report both products, and they should be in stoichiometry. We are chemists here. Most of us are chemists, sorry. So we have H2O. Your product, if we really split water, we should get 2 parts of hydrogen and 1 part of oxygen. Then you get H2O. That's the water. But if we do not report that, or if somehow when we do the experiments, you get a lot more hydrogen, less oxygen, let's say 4:1 ratio, 5:1 ratio, then that is something not right. That means oxygen you do not produce sufficiently. Supposedly you should have a 2:1 ratio, but now you have 4:1 ratio, you have less oxygen. then that might indicate the problem, because water is not oxidized to oxygen. Something else has been oxidized. That can be your catalyst. So that's why the catalyst doesn't last long, because it has been photo-corroded by itself. It has been deactivated by itself, by the holes. Okay, and so on. You can see that sometimes the ratio is not 2 to 1. That needs to be verified, and it's very important in this field of study. All right, in this part of our so-called fundamental exploration, this is a very meaningful work. Meaningful because it's done by Dr. Wayu Utomo over there. All right, this is a published, very good work, published 2 years ago in Small, which is quite a reputable platform for us. And Dr. Utomo, or Wayu, he makes copper particle selectively on a titanium dioxide support. We regulate the defect side. We make some sort of oxygen vacancy and crystal distortion. And we do not randomly put the copper. We selectively control the location of the deposition of the copper. And it helps to give the nitrogen reduction to ammonia. And this is how I want to spend time on his work, on Wayu's work. Wayu did PhD at CTU, Hong Kong. And he is a very good researcher, very good student, and liked by many of our group members, not just my group members, our technical officer, our general office staff like him very much. And that's why when Wayu told me there's a chance to visit Bali, so the members say, oh, well, I would like to go as well. So that's why we have Phoebe, we have Leroy, we have Chili, they want to come here. Of course, Bali is beautiful. But another case is Wayu built a very good friendship. Well done, Wayu. Okay. All right, next, come back to this one. When we make photocatalysts, the science phenomenon could be quite interesting. However, as just now I talked to Professor Hamza, we are working on, like say, uplifting or moving up the TRL, the technology readiness level. We are chemists. We are fascinated by the phenomenon, but we also would like to have real applications to be seen, to be scaled up or processed further. So photocatalyst is powder. Well, the operation is simple. You put the powder. Oops, sorry. You put the powder in, and then you stir them with the light source. It's a very small scale. You produce hydrogen. Hydrogen go to the fuel cell, and the fuel cell generate electricity, and it lit up the device. Can be demonstrated. But when we talk further with the government department, talk further with the industry, no one is interested to adopt that because, well, you use powder in the water. They don't like that. You introduce something to the water stream. This is very troublesome for them if you need to retrieve the powder. And there might be a concern on the secondary pollution. So that's the thing. Scientific exploration, good enough. But for the real adoption, they are not very interested. Sorry, can I have your help for the next? Okay. And maybe the next as well. Next as well, sorry. Okay, so, well, instead of having the suspension, we coat them on the thin film. So now we make the photocatalyst with different color. This is the brown color, we have bright color, white color. Oops, sorry. Oops. Thank you. All right, so we make them onto the thin film. All right, just put it under the beaker, in the beaker. With the light, you see the bubble start to evolve. So things happen. A reaction happen, you get the hydrogen. And, well, this one probably is slightly more practical. However, this kind of a setup is not optimized because water itself will absorb the light. So if we're having the setup like this, most of the light will be absorbed by the liquid before reaching the photocatalyst, which is not ideal. So then we have Chili here. He will be sharing with you later. We start to work on a thin film reactor. You see the thin film over here. This is the photocatalyst thin film here. It has a very, very narrow or very shallow gap that you can fill it in with the water. Very thin layer of water. So, well, as mentioned just now, when we have the water splitting, we use very clean, very high grade, high quality of water, and that is not really something very sustainable. We want to use, we still have 10 minutes, I still have 10 minutes. Okay, thank you. All right, so we want to use something which is not so clean, you don't have to treat it, so we start with the river water, or can be used as lake water. So in natural body, the natural water body, the water is not so clean, you have a lot of organic matters there, coming from the dry leaf, the dead body of animals, small animals, and so on. So this one working with the Hong Kong department, Hong Kong government department. So we go to collect water from two rivers. One is called Shing Mun River in Hong Kong, another one is Yuen Long Creek River for a reason. This Shing Mun River, the water quality, the river water quality is clean. You can see that it's indicated by green color. It's passing through the residential area. This is a relatively better quality water. And then we have the Yuen Long Creek water, which is passing through the industrial zone. The water is relatively not so clean. So in this case, we go to collect, and we want to show if it is possible to use photocatalyst to drive water splitting. And well, we can. If you look at the hydrogen, very low, of course, at this scale, and they all can produce hydrogen. And the ionized water and the tap water, when we look at the production, it's not much of difference. And in fact, of course, this one is in the error bar. In fact, the Yuen Long Creek, which is supposed to be a not so clean water, it gives the higher quality, higher amount of hydrogen production. That's for a reason, because if the organic matter in the water, we have a lot, they are the whole scavenger. They can use the whole very effectively. That makes the redox process happen more easily. But we cannot make the conclusion yet, but this is what we are thinking. In fact, when we look at the total carbon content before and after the elimination, looking at how much carbon left in the water, the carbon content is reduced. That means the water somehow is slightly treated, slightly cleaner. All right, of course, this one involves, if you can see clear, if you can't see clearer, that is Wayu. So we went to the river, and we collect water. So our work include in the lab and sometimes outside the lab as well. Of course, this one is during the COVID time, as you can see. All right. So from this part, I think just 5 minutes here. Not much on chemistry anymore. It's on how we are moving into this direction. We wanted this thin film. We designed a reactor so that we can have the photocatalyst film located there. And with a very thin layer of water, the water can be from clean water, natural body of water, and so on. This is the real reactor that we make, not so huge. We have the 5 by 5 cm, and then we have a different grooving here for testing. And because this one, we need to have the water coming in, water going out, and then sampling of water. So the water is going to have the water flux, and if the thin film itself, the catalyst, is not robustly coated, this might be the region where it is most susceptible for the peeling off and dropping off, and this is not what we want. So then this also involves some of the coating method that we are trying: spin coating, drop casting, spray coating, and then with the binder or without the binder, and so on. And Chili will be sharing with you more on this one later. Okay, so now scaling up from 5 by 5 centimeter, we are interested to go up slightly larger. Of course, for some other chemical engineering applications, this one is very small, but for photocatalysts, this is somehow quite meaningful that we think. So we come up with a slightly, like say, refined reactor. It's very thin, you can see that. The black color here is our photocatalyst after modified, and with a very thin layer to have the water so that it doesn't block the water that much. Okay, so well, this is how we tested that. We can use that in the outdoor illumination, but this kind of a prototype, this kind of things can be used for indoor as well. Thank you. All right, so well, And as we say just now, when the water is not that clean, we have organic, we can see that as a pollutant. After the illumination, the water gets slightly cleaner. So here is where we started to talk to industry in Hong Kong. All right, they have wastewater, pharmaceutical company, or some food waste industry. The water itself contains a lot of organic matter, and for treatment, they need to treat the water, and they have the facilities. So if they can, we are working with them now. So we have this kind of a reactor. You have the photocatalyst underneath, and the wastewater on top of that. You can have the indoor illumination. This one is just a demonstration, but you can consider, you can imagine that we can have a container size of a reactor, that you have one layer of light, one layer of reactor, another layer of light, another layer of reactor, like the sandwich. Okay, so the water coming from the first reactor passing through, the water slightly treated, it can go through the second layer, third layer, and so on. So that then we can kind of like treat water and at the same time extract a bit of hydrogen for achieving some sort of a certain level of self-sustain. All right, this is ongoing. And well, lastly, this is the largest that we have, 1 meter by 1 meter square meter. This is a very small compared with a domain sensei from the work on, they have 100 square meter. But this one, we want to demonstrate that we can use natural water to produce hydrogen, and at the same time sort of like cleaning the water slightly. And this is by far is what we have been working. Well, maybe most of you understand Hong Kong is small, space is limited. So when we want to go further to say 10 to 20 square meter, even larger, the space is an issue that we need to tackle, and this might be something that I have to consider further. All right, so this is the thing. More on the design and the installation and the testing, I think Chili will share with you in the afternoon if you are around. All right, so well, this project is supported by Hong Kong government. The original idea is that we want to build a demonstration unit next to a river with the running park, with the cycling path there, and to have the river water come in, go through our photocatalyst, and then the water can go out, and we have the hydrogen, and to demonstrate that it is self-sustainable. This is the work that we are currently delivering. All right, I think about time. So I just want to make a summary. So we make fuels or chemicals using photocatalyst, and I hope that photocatalyst itself can have a slightly more consideration in the community, well, say for example in the green hydrogen domain. And well, as this part of the work of this sharing, hydrogen can be generated from a non-typical water source, and that can be might be interested to a certain industry as well. All right, that's all for my presentation. Thank you.
Thank you very much, Professor Yun Hao Ang. I don't expect that I will see my face in this slide. And relating to this, I consider it your comment as a comment from the reviewer. Thank you for that.
Thank you, Ayu.
Is there any question from the floor, please? Okay, thank you.
Okay. Good morning. Thank you for the opportunity. Very interesting. First, let's introduce myself. My name is Renanto, doctoral student from University of Indonesia. Thank you for the presentation. It's really good because this is really with my research also, because in our university we are focusing on utilize the perovskite-based material to generate HER and also OER, because we are focusing on that also. We want to make more lower energy that needed to generate some H2 gas and O2 gas. The main concern related to this field, thank you for the presentation. One thing that interested me is related to this water source, because this research field needed water based as a fuel to generate HO gas and or O2 gas. Related to the source of the water, as you see that from the river, it's more or dense hydrogen concentration. And my question is, what is the main reason behind why the river source is more contained of hydrogen elements or something? Because the main competition at the research field, this is compete with human beings, because we also need water, and this research need water as a fuel. About that issue, it's really interested that maybe for we can utilize another water source to gain more, because as from your presentation, I can see that in the more less of water, we can generate more H2 gas. So maybe it's, I'm really interested to the main reason behind it, because if we can solve this, we can make it more sustainable, because we need more less water to generate the same amount of hydrogen or oxygen gas. Thank you for the opportunity. I will give to the...
Okay, thank you for your questions. Well, when we use river water, or in general, not so clean water, we suspect, not suspect, the higher hydrogen amount, the yield, is because of the presence of the organic in the water. Because I don't show back the slide, but for the redox reaction, we need to have the reduction and oxidation happen at the same time. If they do not happen at the same time, there will be no activity, because the charge will be recombined. So that for water splitting, water reduction to get hydrogen, and if the oxidation part is water, water is very difficult to be oxidized. So the overall activity is lower. But if the water has organic is slightly, depends on which type of organic, it can be oxidized more easily. That means it can use the holes more easily. This will give the electrons more chances to be utilized. And that's why the reaction happen easily or more easier with a lower barrier energy. So that's because of the presence of the organic there. So river water is just one example. We are working with the food waste water. In food waste in Hong Kong, we have the facilities to treat food waste. In food waste, they have a lot of organic compound with the nitrogen content, nitrate especially. So Wayu project is is part of that overall objective. We use wastewater with the food waste, specifically from food waste. So we can oxidize the organic one end, and the nitrate itself can be reduced to form ammonia. So we have the ammonia, and then we treat the organic part, and sort of like turning the waste to resources, waste to energy. So we're exploring different water source, like say in addition to the typical source. And on top of that, I'm not sharing here, but we have a very interesting finding. The water source not necessarily from the liquid water. We like the atmospheric humidity. In the humidity, we have a lot of water. It's actually the reserve is pretty high. And we have the project, which is not shared as well, because it's still under some agreement. We can absorb the humid, particularly good for Indonesia, Hong Kong. We have a very high humid, including Japan as well. The humidity is so high, we absorb the moisture from the water, from the moisture, and then at the night time, and then we split it during the daytime. And surprisingly, the hydrogen activity is very high, because the water is not in liquid state. When we have the water in liquid state, it blocks the light from reaching the photocatalyst. But now when they are in the vapor state, we don't have such issue, or we have a minimum challenge on that. So we see, we are working on that. Hopefully, in the next seminar, we will be able to share with you on this outcome. Right, thank you.
Thank you, Professor Yun Hao Ang. Due to the time limitation, I'm afraid that we need to finish this.
Thank you everyone.
Presentation, and thank you. for the next speaker, the next speaker is someone that I consider as an inspiring young practitioners. In Indonesia we call it eksekutif muda gitu. I don't know what is it in English, but I think he's very inspiring because I use him as an example in my class to inspire my students on how to work and how to study. So the third speaker is the Aditya Farhan Arif. He is a practitioner in now he is a department head of the corporate planning and technology development in mining industry Indonesia, or MineID. MineID is one of the biggest holding company concentrating on the mining companies in Indonesia. He was graduated from the ITB, Institut Teknologi Bandung, and he got the PhD degree from the Hiroshima University joint program with ETH Zurich in 2009. In 2016, he also has experience as the visiting researcher and also has the experience as a research assistant professor from 2017 to 2018 in Hiroshima University. And after that, he joined the company, many companies, and now he serves as the department head of the corporate planning and technology. In the scientific background itself, his expertise is in the nanostructure and functionalization of advanced materials, electrochemical energy storage and conversion, and photocatalysis. So the time is yours for the speakers, Dr. Aditya Farhan Arif. Thank
you. Thank you very much, Dr. Wahyu, for the nice introduction. So, ladies and gentlemen, good morning. It's been really a pleasure and an honor for me, of course, to be part of you in this conference. It's really nice to be presenting here. So, and also duly noted that we are approaching the lunchtime. So, my primary job here is actually to keep you awake in this critical time, because I'm going to talk about critical mineral commodities. So my presentation will be much easier to digest, I believe, because it's not really technical. I'm just going to introduce the critical minerals, the strategy for the downstream, and what we as a researcher can do about it. So yeah, I'm representing MineID, Mining Industry Indonesia. So we are a holding company, a state-owned enterprise holding company for mining, mineral, and coal industrial clusters. So in Indonesia, the state-owned enterprises are classified into several industrial clusters, and each cluster is led by one holding company. And MineID is the holding company for mining, mineral, and coal industrial cluster. So we are 100% owned by the government of Indonesia, and we are the shareholder of several mining companies that is pre-existent. Actually, they were founded before us, with various commodities from nickel, gold, silver, bauxite, aluminum, copper, what else? Coal and tin. So yeah, recently we increased our share here in Vale Indonesia from 20% to 34%. Because we want to optimize our nickel portfolio. You know, nickel is really, really important now because of the emerging of EV battery. And also we have a trading arm located in Singapore. And this year we are planning to increase our share in the Indonesia Battery Corporation, or IBC. IBC is a special purpose vehicle founded by the government, so owned by MineID, Pertamina, and PLN, all energy companies in Indonesia. And this year we are going to be the majority shareholder of Indonesia Battery Corporation. This shows you how important nickel battery for the government of Indonesia. And fortunately, all the commodities that we have, that MineID has currently, is categorized as critical minerals. So what are critical minerals? and how we define them, how are they classified. So this figure gives you a list of the minerals, well actually not only minerals, but also the elements, but you know, the terminology that we use in the world, that we commonly use is the critical minerals. But anyway, so this is the list of the critical minerals that were defined by China, European Union, and the US. So we can see here that each country or each region has its own classification or list of the critical minerals, which suggests that they have their own criteria to determine the criticality of a material. So for example, in the US, they focus on the supply risk. So if one material has a potential of supply disruption while they are meaningful for the energy, then this material will be critical for them. And for the EU, same goes for EU, they also see that supply risk is an important parameter to determine the criticality of a material. But they also emphasize the economic importance of one material. While in China, they focus on the importance for economic security, defense security, and strategic emerging industries. But regardless, you know, they classified many minerals, they have their own list, but if we take a look at this list deeper, the materials that are used for EV batteries are considered critical for all regions. So in the intersection, we can see that there are lithium, graphite, nickel, cobalt, copper, and aluminum. These are very important materials for the EV battery ecosystem. What about Indonesia? So Indonesia, earlier this year, the government of Indonesia, through the Ministry of Ministry of Energy and Mineral Resources, MEMR, they stipulated new regulations on the critical material, not only critical actually, but critical and strategic minerals. But basically, the critical minerals and the strategic minerals stipulated in this decree of the ministry have to be able to support the strategic industry. So these are the strategic industry defined by the government of Indonesia. And we can see that one of them being the electric vehicle. And also we see that the transition, the energy transition is also important for our future. And if you see, or if you read many analysis by BCG, for example, or Wood Mackenzie, they see that the energy mix in the future will be dominated by solar power. So that's why photovoltaic becomes one of the strategic industries. Although I personally disagree because, well, from the characteristics of the photovoltaic is not really suitable for Indonesia actually. So Indonesia should find something else actually. But anyway, so we have approximately 47 critical minerals in our list, and 21 minerals for the strategic minerals. And there are also the intersection between critical and strategic minerals, approximately 19 elements. So actually, ladies and gentlemen, the critical materials, I mean the definition of the critical materials is actually a part of what we call protectionism policies. So basically, each nation wants to protect their country for the sovereignty, for the security of the supply, for the goods of their society. So the definition of the critical minerals is also part of the protectionism policy. Like for example, in the United States, recently they released the Inflation Reduction Act, or IRA, which basically prohibits, well not prohibits, but make the goods or the commodities coming from the foreign entity of concern, or FEOC, which are China, Russia, Iran, and North Korea, difficult to enter the US because they will be given a huge tax. So their goods, their commodities will not be competitive inside the US. That's the IRA. So the motivation is to protect the industry in the US. Same goes for European Union, but the EU, the focus of the EU is more on the stockpiling of the commodities. They want to get as much as possible commodities under their control so that they can preserve it for future use. And in Indonesia, one of the protectionism policies that is very familiar to us is the export ban. The export ban of raw materials, of mineral ore. So these actions, these protectionism policies will furthermore lead to the development of downstream industry. Because they want to protect the nation, it means that in order to make a resilient business, then they have to create the value chain within their country or within their region, which leads to downstream initiatives. So this is what happens. One of the implications of the protectionism policy. I'm taking cobalt trade flow as an example. So this is how the cobalt is traded throughout the world, and because of the implementation of the IRA, the cobalt from China will no longer be able to enter the United States because they are incompetitive. So it creates new opportunity for other countries to supply the goods to the United States, which leads to the development of downstream industry in other countries. So that's why I told you that the IRA is one of the triggers that we can create the downstream industry. And well, in the end, this is a part of what we call deglobalization. So the world is now becoming more regional. Well, not really regional actually, but the commodity trade is now reoriented throughout the geopolitical lines. So if you are allies with other countries, then the trade flow will be only between these two countries. So it's no longer that free anymore in order to do the trading. So this is a brief introduction on the critical minerals and the policies behind it. And so, yeah, let's enter to a part that is slightly more chemistry. So earlier, we see that all country agrees that the commodities required for the EV battery are critical. So if you're talking about EV battery, the best proven commercially available EV battery now is the nickel-based battery, the NMC. But actually, the NMC battery does not only contain nickel, but they also contain graphite. In fact, the content of the graphite in nickel-based batteries is even higher than the nickel itself. So we also need manganese, cobalt, lithium, aluminum, and copper. And if you're talking about a battery, then we need to put the battery inside the car. And in the car, the structure requires aluminum. Electric vehicle consumes more aluminum than the ICE vehicle does. Why? Because we want to lower, we want to make the vehicle lighter in order to make the efficiency of the vehicle increase. So that's why the consumption of aluminum is expected to grow very high in the future because of the electric vehicle. And of course, in the electric vehicle, the powertrain requires electrical motor, which mainly constituted by neodymium-based magnet. Well, when I was in Japan, our research group was involved in the development of non-rare earth magnet, but we understand that this is still far from commercialization. So to date, the best permanent magnet is still neodymium-based, which is rare earth. And in the future, we are expecting a huge supply gap for the neodymium. So we are expecting a very high price for the neodymium. Okay, and if you're talking about EV, then you need to charge the EV. The carbon footprint of the EV will be highly determined by where the energy comes from for the charging. So energy transition is an embedded part of EV battery ecosystem. And of course, different from the fossil energy, which consumes only fossil and maybe some other construction materials to build the plant, the renewable energy consumes more critical minerals. So not only carbon, but we will also need tin, neodymium, uranium, thorium, aluminum. These are the materials that will be required in the future. And we also, well, if you read many analyses on the future industry, then you will see that the growth of AI, artificial intelligence, will be very huge. And AI is only the brain. So it's like we as a human, we have the brain, but we also have the hands and also the foot to operate. Then AI also needs infrastructure, which mainly constituted by tin, silicon, copper, and gold. So these commodities will be very, very important in the future. And this is about the renewable energy in the future. So again, as I told you earlier, that solar power will be dominating, while in the solar power, the consumption of aluminum and copper is very high. So the demand of copper will be very high in the future. So in summary, if you analyze the external factors, the global megatrend using the pastel analysis. The pastel analysis is a very common tool to examine the external factors. What I'm going to say is that these factors will give a positive impact to the base metals and critical minerals. So that's why it is really, really important for us to secure the supply of these metals. So now I'm going to talk about the downstream industry. Earlier, Professor Yuan Hao talked a lot about photocatalysts. Photocatalyst is one of the examples of advanced materials. Now, these slides give you a figure on how the photocatalytic materials and other advanced materials were produced from mining. So they are actually originated from mining. So from mining we can get ore, and there will be some midstream processing. Maybe some of you heard about smelters. This is the midstream processing. And then after that we can get the metal, be it in the form of ingot, billet, bullion, anything, and it will be further processed into advanced materials. Although we can also directly synthesize advanced materials from the ore. And one thing that I want to emphasize is that the midstream process releases some byproducts, which actually still contain many critical minerals which are used for downstream industry. One of the examples is rare earth element. The rare earth element, we can find rare earth element in the byproduct processing. So we need a technological solution in order to be able to valorize these materials. So this is to give you a figure on the value chain of the existing commodities for the nickel-based lithium-ion EV battery. So, for example, I'm just giving one example. So from nickel, there are two types of nickel ore, saprolite and limonite. Saprolite is used to, usually we use saprolite ore for stainless steel, so for construction materials. But recent development by China, they can convert the saprolite into ferronickel and then directly forming nickel sulfate without producing nickel matte. And from the limonite, we can further process through the HPAL, the high pressurized acid leaching, to produce mixed hydroxide precipitate, and then we can get nickel sulfate. Nickel sulfate is the main precursor of the NMC cathode. And maybe one other example is bauxite. Bauxite is used to produce aluminum. From bauxite, we do the refinery to produce alumina, and we do reduction to produce aluminum. And then we combine all those materials forming battery cells. And this is another example of the value chain. So in the previous example, the value chain is already complete from mining to the downstream. But there are many cases where the critical minerals have not been monetized. as I mentioned earlier, they are still in the byproducts. Taking tin ecosystem, for example. So from tin mining, we only get cassiterite. Cassiterite is the mineral that contains tin. So basically what we do is just we reduce, we do reduction of cassiterite, and then we can get tin ingot. But together with cassiterite, we will get another 27 associated minerals. And these 5 minerals are the top 5 minerals that we can get from tin mining. First one is monazite, and then we have zircon, we have pyrite, we have ilmenite, we have quartz. From monazite, if we further process the monazite, we can get rare earth elements. And also in Indonesia, this is the case, the composition is coming from the resource that MineID has. In Indonesia, our monazite is also rich in radioactive materials. So we have uranium and also thorium. From zircon, we can get zirconium and silicon. From ilmenite, we can get titanium and iron. From quartz, of course, we can get silicon. These elements are really important for the next for the next industries, for the future industries. And not to mention that from the primary ore, we also have the reserve of tungsten and fluorine. So yeah, the challenge is how to extract these elements. So now, in MineID, we are now processing this one. We collaborate with a Kazakhstan company. We try to extract these materials from monazite, but we have not yet done the beneficiation of the radioactive materials. And we are planning to do so because, later I will discuss about it, because the downstream industry needs a huge amount of energy. And we also have the homework to do the decarbonization at the same time. So we need a huge energy, the electricity should be cheap, and it has to be zero carbon. Then I cannot think any other technology that is very near to the commercialization other than nuclear, frankly speaking. So that's why we want to try to valorize this as well. And this is another example from bauxite ecosystem. When we are processing bauxite into alumina, we will release through the Bayer process, we will release the byproduct or the residue called red mud. Red mud, well, the color is red, hence red mud. It contains mostly approximately 40% of iron, so that's why the color is red. Yeah. So this is what makes it interesting. For every ton of alumina production, there will be 1 to 1.5 tons of red mud produced. So by 2029, MineID Group will produce 2 million tons of alumina per year. So we have to be able to deal with 3 million tons of red mud. So it's a huge challenge for the environment. Whereas the red mud itself still contains many critical minerals. But to the best of my knowledge, I haven't found any proven technology which already commercialized, which close to commercialization, which can extract this from red mud. I'm going to Russia in this September to talk with Rusal. Rusal is one of the biggest aluminum players. They claim that they have a technology to extract scandium from red mud. So this is to give you a figure that this is a huge challenge, not only for Indonesia, but also the aluminum producing countries. So yeah, this is just to give you a figure what we have done. So the downstream industry development has, the protectionism policies have led to the development of downstream industry, and this is what we have done so far. So I'm going to talk a little bit more on the business characteristics of the downstream industry. Why is it a little bit challenging for us to do? So this illustration gives you, a figure on how the trend of the business. So this is the upstream, this is the downstream. So what tend to increase if you go more downstream? yes, we can get revenue and added value, it is clear. But at the same time, the competition in the downstream industry is very, very high. So the bargaining power of buyer becomes high. Means the supplier has no, the bargaining power of supplier will be much lower. And then the threat of new entrants, the product substitute becoming high as well. It means that the competition is very, very intensive in the downstream. Not to mention that we need more capex. So to give you a figure, if we go downstream, like for example in nickel, if we process limonite ore into MHP, into mixed hydroxide precipitate, the value of nickel in the mixed hydroxide precipitate will be 17 times higher than that of limonite ore. And if you further process the MHP into nickel sulfate, then the value of nickel will reach 115 times. So this is a huge money, right? But at the same time, we need to do something with this challenge, because the competition is very high. Means we have to innovate, always innovate. And at the same time, if we go more downstream, the profit margin tend to decrease. So if I compare BHP, BHP is one of the leading mining companies, top global mining companies, and Sumitomo Metal Mining. Sumitomo Metal Mining focuses on the downstream. BHP focuses on the upstream. The profit margin of SMM or Sumitomo Metal Mining is much, much lower than that of BHP. This shows you a figure that downstream industry is really challenging. Although it creates new value, but the margin, the profit margin is low. So that's why, not every country, not every company is willing to do the downstream activity. So this is to give you a figure on how the comparison between upstream and downstream for the from the Porter's 5 forces viewpoint. This is a framework to compare the business environment. So yeah, in the upstream, the thing that everything is low, so except for the bargaining power of supplier, which is good for us. On the contrary, in the downstream, everything is high except for the bargaining power of supplier, which is low. So that's why we need technology innovation. This is what I'm going to discuss a little bit more later on. So these are the key success factors if we are going to build a downstream industry, in my opinion. So the first one, we need to create the value chain. So the value chain should be really complete within the region. For example, we have a joint research with ITS in the development of SNO2 nanopowder from the backlog of tin mining. But then later on, we don't know where to sell this SNO2 nanopowder because the electronic industry is not there. The electronic industry, which is the main consumer of SNO2 tin dioxide nanopowder, is not in Indonesia. So we need to create the value chain. And then we need to understand the market because of the high competition. We need to innovate and we need to really listen to the market, what they really need. So the supplier-buyer relation is different in the downstream. It is based on longer-term contract. And we need a rapid and agile technology development because we need to compete with the market. And because we have to have a value proposition, we need to propose something new to make us different from our competitors. One of the most important things is the price. We need to reduce the price, means we have to have efficient operation. And this also needs a technological solution. So there are many, many homeworks that we have to do. And these 4 factors should be supported by policy support, which allows, an efficient trade, and also the human capital. We need to prepare ourselves for entering the downstream industry. So yeah, this is what I've been talking earlier. So this shows you that how the downstream industry is very, very starving for energy. So we need, in the next 5 years, we need another installed 5 gigawatt of power to supply our downstream strategy, while our downstream initiatives are actually not that much. So even if we want to boost the downstream industry more, then we need much more than this. The figure will be very, very higher. So speaking of the technology excellence, I discussed that earlier, that technology excellence is one of the key strategies for the downstream industry. We need to learn to respond the market needs. So taking an example from the battery, earlier in the battery development stage, the customer's needs are limited in the long driving range for the vehicle, durable and long last or cheap. But recently, they need fast charging and also high power because they don't want to lose the experience of driving the internal combustion engine cars in their electric vehicles. So they need high power. So the battery itself, the battery development has been evolving. If I divided the technology development into four clusters here, so when the market is still premature, is not yet mature, and you have new technology, then you give the new solution to existing problems. Same goes with battery. So earlier, in the earlier stage, battery was in this stage, and then it is evolving towards becoming a complex product where the market and the technology evolve together. I'm sorry, my time is limited. So the battery, many battery developers are now responding the market by creating new types of battery. So this kind of, maybe I will just skip this part. This kind of approach is very, very necessary for us in order to becoming one of the leaders in the downstream industry. And we should not also forget that the technology development should be based on the product maturity. So if the product that we are researching is already mature, then what we have to do is product improvement. If it is in the take-off stage, then what we need to do is lower the price to attract the customers. If it is in the early development, we need to intensify the R&D for first kind of commercial. So this kind of approach should be sticked in our head so that we will not waste our resources to do something that will not be beneficial for the society, for the scientific society and for the industry. So, yeah. How to become an innovation powerhouse? This is what we think as an industry. Well, we need 6M, but what I want to emphasize is, first is the man. We need highly qualified people with strong passion in research, and well, I'm expecting more PhDs are working in industry. So PhD doesn't always work as a scientist or as a lecturer, but we need also PhDs working in industry, because PhDs can forecast the future better than the businessmen, believe me. And for the money, I understand that money is a challenge for research, but money is not everything. I mean, yes, we do need money, but by having men with huge passion in research, we can do better than just having the money. And also for the materials, for the research, I really, really encourage you, because we are all scientists here, and most of you, I believe, are chemistry students. Please focus on what we have. What we have means we need to understand better ourselves. If Indonesia has many resources, if your country have many resources, then try to understand those resources better. For example, in Indonesia, I'm taking the case of Indonesia, we have a lot of resources and many byproducts. We don't know exactly the characteristic of our bauxite, yet we are producing bauxite to produce aluminum, but we don't really understand what is the difference between our bauxite and Chinese bauxite. That is so unfortunate from my viewpoint. That's why if you're talking about red mud, which is the bauxite residue, some people say that our bauxite contains radioactive. They emit alpha radiation. Some other people say they don't. So it makes the confusion in the development and the policymakers. But the basic data is not there. So I'm expecting this kind of research is escalating in the future. So we need to understand our resource better and also focus on value add. So I understand that basic research is important, but in the subsequent stage where the research is about to enter the commercial, then focus on value add. Value add also means we need to reroute the process if it is necessary to make it more economical. So basic research is okay, just focus on the science, but later on when we try to enter the commercial stage, then we need to have better awareness on the commercialization. So for the scientists, for the basic scientists, I encourage you to do more basic research in Indonesia. So there are two beliefs in the technology development for the innovation pipeline. The first one is technology push, and the second one is market pull. Technology push is where R&D drives the market. The best example is Apple with iPad. Before Apple launched iPad, no one thought that they would need iPad. But after Apple launched iPad, then suddenly many other manufacturers create the same thing. So this is one of the example of technology push. While in the market pull, the technology offers solution to existing problem in the market. Which one is better? Nothing is better. Either one is just as good, in my opinion. But what I want to emphasize is, with whichever way we are going to take, it begins with basic research again. So let's not jump into the applied research. We need to strengthen ourselves in the very entrance of the pipeline of the innovation itself, which is basic research. So this is to give you a figure if you want to become a downstream player, a battery player in the world. These are the world's top 4 battery manufacturers in the world, and these are the number of the patents. These patents are, of course, constituted by some basic researchers as well, and most importantly, these patents are productive patents, not only just for file. So. Yeah, this is just a soft and gentle reminder for us as researchers. So, and maybe I have another 2 slides. I'm not a big fan of the term matchmaking. I don't like matchmaking. I want to pick my own girlfriend. I don't want to be match made by my parents. So I don't like the term matchmaking in the research and technology development. Matchmaking means the researcher independently do the research, and then at some points they sell that research to the industry, and the industry continues. Such kind of thing will never work. Why? Because when the researcher eventually meet the industry, they will need to redo everything, because many things have to be reoriented for the commercialization. So what I believe is that the industry and the researchers should begin the research together from the very beginning, but with different proportions. At the beginning, when the technology readiness level is still low, still in basic research, the leader, of course, is a scientist. So the industry just give you, the border, what to do on, just to give the researchers a figure on what is possible to be commercialized or not. For example, if you are using ionic liquid, in 2015, ionic liquid was quite happening. But at the time, many industries screamed that ionic liquid is not possible, it's not feasible for the commercialization. Then we need to find something else. We need to find other routes other than using ionic liquid, for example, for the product development. And at the same time, we need to find another way to make the ionic liquid as cheap as possible. So that's how we work in the technology development, the collaboration between the researchers and industry. In that way, we can move forward and enter the commercialization much smoother. I'm not saying much easier, but much smoother. And this is is just to show you how we upscale our technology. Well, this is the norm that we use in the industry. So we know that from the lab we need to upscale, do the upscale until the commercial. But one thing that we should not forget is that before we upscale our research, we need some simulation on the economic feasibility. Of course, the quality of the simulation will be much better along the way, but at least if we know that our research is not feasible at the early stage, let's just go back to the basic research to optimize it before entering to the commercial stage. In that way, we can do the work more efficiently and we can do the research more with high agility. So, to conclude, developing minerals downstream industry is like solving a complex puzzle. In my view, you might have different view, it's okay. It requires persistence, commitment, and collaboration, but we need to take one step at a time, and we can contribute to the better in accordance with our functions and positions. So I think that concludes my presentation. Thank you, and I will see you next time.
Thank you, Dr. Aditya. It is, for myself, I think it is very interesting to get the insight from the industrial perspective, so we know how we need to do with our research. Because finally, if we do the research, we want to have the products that can be commercialized or can be applied to the peoples. And usually, mostly, or maybe absolutely, we need the industry to do that. Is there any question? Maybe one question from the floor, please. Is there any question? Otherwise, I have a question actually. So, in Indonesia right now, what is the strategy, and what is we have a kind of a strategic on critical minerals, like, and there are so many minerals inside in this kind of in that circle you sign that, and then I guess if we do all of that critical mineral to be developed in this near future, maybe it is very difficult. So in Indonesia, what minerals that being a focus for our development in this current state, especially in the mine ID?
Oh, thank you, Pak Wahyu. So if mine ID represents the state, then I would say that we are focusing on this first. So now we are, the threshold for mineral extraction, for element extraction from minerals, is usually in 1% level. So if the content is beyond 1%, minimum 1%, I can guarantee that it will be able to be commercialized because it will be economical. The process will be economical. So we will try to extract those with the highest concentration first, and also with the higher value in the market, that being rare earth element. So that's why we focus on this first. And then in the future, with the emerging technology of solid state battery, we will need other types of cathode, like for example LTZ, LTZE, which requires titanium, and also zirconium. So our next target is ilmenite and zircon. So in the next 5 years, these developments are already in our horizon. And then after that, I'm sorry, can you please help me clicking the, okay. We are also focusing on this because of this potential disaster in 2029. So we need to do something with the red mud. But we are not thinking of extracting these elements from red mud for now. So our first priority is do something with the red mud. If any of you, we can collaborate to do something to extract these metals, then we are very welcome.
Thank you, Pak Aditya. So the first concern from Indonesian government, or at least from the mine ID, is the extraction of the rare earth elements. And I think it's quite interesting topic, especially for the local researchers in Indonesia to develop extraction, because as I understand, indeed, the percentage of the rare earth element is very low, and it is quite challenging too to extract it from the source itself. Thank you very much, Dr. Aditya. Your talk is always inspiring me, and have an applause to Dr. Aditya. Thank you very much. And
Okay. Okay, thank you so much. And that is concluding our keynote sessions for today. Give a round of applause for the last two speakers also, Professor Ng and Dr. Adit. Right now we are going to be moving on to the giving of these souvenirs and vendors. Okay. Okay, from the first one, we are calling upon Mrs. Yuli Kusumawati and also Mr. Professor Freddy to give the souvenirs to, I invite Mr. Professor Okada Sensei to the front to receive the souvenirs itself. Okay, thank you so much, Professor Okada. And right now we are calling upon and we invite Professor Ng to the front and to receive the vandal itself, the souvenirs. Okay, thank you so much, professors. And right now, one more, we are calling upon Dr. Adit to receive the souvenirs itself. Okay. Thank you so much, Dr. Adit, and thank you so much, Professor Freddy and Mrs. Yuli. Ladies and gentlemen, we are now coming to the end of today's plenary sessions. After the end of the sessions, we will gonna have a lunch break until around 1 p.m. And at 1 p.m. on time, we are encouraging all participants to go to each parallel room that the schedule that has already been given by the committee itself. And in there, you can see others and present your own research watch by all participants. Also, the committee has also sent the links to the group chat itself, the Isoc information, so please do look upon that. And also, the last thing that I want to tell you is that we have 2 spots for lunch. The first one is the Anjani's room downstairs behind the receptions, and one of the others lunch spots is actually in the Aglio restaurant in front of the pool, to alleviate queue and to maximizing the time itself. You can go either to one spot to have a lunch there. I think this is all that I can say for today. Thank you so much for your attention. You guys could have a delicious lunch downstairs. Thank you so much, and see you tomorrow. Oh yeah. Okay, so before that, the okay, so all the invited speakers and all the keynote speakers can go to the front of the stage here to have a photo together with the moderator. So. Di belakangnya udah ada backdropnya. Could the committee help to turn off the projector, please? Professor, let's take a photo. So I'm going to conduct the photo sessions itself. Please take a look at the cameras back there. Once again. Okay, thank you so much. Agus Rianto here? Agus Rianto? No. The other is Wan Nazwani. Is there anyone named Wan Nazwani? No? Okay, thank you so much.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome back to our second session, the parallel session. In this session, we will have the invited speaker. Will talk the invited speaker, Dr. Hoi Ying Chung. Sorry if I get wrong pronunciation. Your name. She come from City University of Hong Kong. Yeah. Her research. This is her CV. Her background. She get PhD from Australia in 2019, and her research is about photoelectrochemistry, photocatalysis, solar water splitting. And we can see her publication. I think more than this publication, right? If we write all, maybe we will have more than this page. Yeah. Okay, before we start, I will introduce. My name is Nikma. I will be a moderator for Mrs. How I call you, Mrs. Hoi or Mrs. Chung? Miss Chung. Sorry, Miss Chung. Okay. We will have time for 20 minutes your presentation, and then we will continue 10 minutes with discussion. Yeah. 20-minute presentation and 10 minutes for discussion. This, I think, not too tight. A little bit is also okay. Without the two, maybe we can start the presentation. Please, time is yours.
So good afternoon, everyone. I'm Hoi Ying Chung from City University of Hong Kong. It is my pleasure today to share my research, which is on the metal oxides in photocatalysis for environmental and energy application. So to start with, I will, as we all know that we are facing one of the global issues, which is the global warming. So, and due to the from the NASA statistics that the global average temperature has been increased by 1 degree Celsius compared to 50 years ago. So researchers has tried to find a way to replace the fossil fuels. So they have found out that hydrogen is one of the powerful renewable energy that can be used to generate electricity and also to replace the fossil fuels, and without any undesirable byproducts is being produced. And what else? Actually, eutrophication is another issue that we're also facing daily. This is due to the excessive of the agricultural and also industrial activities that will lead to a high concentration of the nitrates in the drinking water that will harm our humans' health. So to overcome the environmental contamination, researchers also propose that why we don't convert the nitrates into ammonia, since ammonia is a useful chemical product that can be used to fabricate the fertilizers. And not only that, ammonia can also be breakable to hydrogen gas that can be used as the energy fuel source too. So how we can produce a green hydrogen and ammonia? Among all the renewable energy sources, solar energy is the most abundant energy that can be tailored to the large scale for the long-term needs. And there's a simple and economical method, which is the photocatalysis process. And this process is based on the all-in-one module that the photocatalyst will actually absorb the UV lights from the sun for the photocatalysis and split the contaminated water into hydrogen, or ammonia. And based on the principle of the photocatalysis, then we can construct two different types of photocatalytic system. The first one will be the photoelectrochemical PEC tandem system, and another one with the Seskine system. And how we can, how the PEC operates. So by taking the water splitting application as an example, first, we need two photoelectrodes. And when the sun shines, a higher energy is being absorbed by the photons than the semiconductor band gap. The electrons and holes will be split to the conduction and valence band. Meanwhile, the the holes in the antiphotic anode will actually oxidize the water into oxygen, while the electrons will be transported to the counter electrodes by applied bias and reduce the hydrogen ions into hydrogen gas. And a similar theory is actually applied towards the Seskine system, where it requires an N-type and a P-type photocatalyst, but in a particulate form, and we also requires an electron mediator. So in order to overcome the energy depletion issue, we actually have research on a PEC water splitting process. So this is the water splitting process. And basically, most of our research are mainly focused on half reaction because the water oxidation is and in our research are mainly focused on the water oxidation because it's one of the limiting factor. It requires more energy to withdraw 4 electrons, and thermodynamically, this process has a sluggish kinetics rates due to the lack of the lack of the long-lived photo holes. So in order to achieve efficient photoanode for oxygen generation, researchers has been reported that a photocatalyst should achieve a narrow band gap, a low charge recombination, and also even a low charge resistance. So there are numerous developed photocatalysts, including the binary metal oxides, titanium dioxide, which is earth abundant, but they possesses a large band gap and the poor tunability of their structures. So to overcome this, bismuth-based ternary oxide may help because they consist of hybridized O2p and bismuth 6s orbital that can push up the valence band to achieve a narrower band gap. And among all, bismuth tungstate is one of the photocatalysts can be further developed in the advanced use in the field of photocatalysis. This is because that bismuth tungstate is non-toxic and also achieve the hybridized O2p and bismuth 6s orbitals, which can provide an active site for the oxygen evolution. And in addition, that bismuth tungstate has a strong visible light absorption ability and high stability against photocorrosion. So in most of the studies, they are trying to improve the bismuth tungstate for enhancing the PEC water oxidation performance. And mostly are just modifying the photocatalyst, such as tuning the precursor concentration during the fabrication and forming the heterojunction or addition of co-catalysts, which this allows to change the material's properties, change in their band structure, or even increase their light absorption. So in our research, our motivation is trying to find a simple strategy in order to alter the charge kinetics of the materials to boost the photooxidation reaction. So therefore, in this research, we have simply started by tuning the tungsten concentration precursors of our bismuth tungstate and to try out to figure out how the tungsten will actually affect the PEC performance. So we started by fabricating our bismuth tungstate thin film by a isothermal reaction by starting with a fixed amount of EDTA and a bismuth precursor and varying the concentration of the tungsten precursor with a mole ratio of 0.5, 1, or 1.5 and adding all together for isothermal reaction. And we characterize our sample under SEM, and it shows that all samples has a plate-like morphology, and we also figure out that the dimension of our plates can be controlled by tuning the bismuth and tungsten precursors accordingly. So by comparing their length and thickness, it shows that when a higher tungsten content being used, a smaller size of the plates is being formed. Meanwhile, it's also increased the thickness of our lateral facets. And to easier to follow my presentation, I would like to name my samples as W1, W2, and W3, where the numbers is referring to the ascending order of my tungsten being used. So next, from our XRD patterns and also UV vis spectra, it confirms that an ortho-Hombeck bismuth tungstate is formed, and a similar band gap of 2.9 electron volt is detected for all samples. So after that, we like to measure the PEC performance among all three samples, and we figure out that although that three samples has the same band gap, it shows that the W3 samples has a higher photocurrent density is being recorded from our linear sweep photometric plots. And besides, our apparametric plots, when we apply a constant bias of 0.5 volts, it also shows that the W3 samples exhibit the best performance, which reaches 0.5 milliampere per centimeter square. So, when we're tuning the tungsten concentration will affect the PEC performance. But what are the major factors that actually influence the materials inside that will cause this PEC enhancement? So I would like to talk a few of our observation. So first, we have characterized the conductivity of our samples by the conductive atomic force microscopy. And in principle, that CAFM is a method that can give an overview about the charge transfer efficiency, charge carrier concentration, and the charge mobility of the materials. And when the photocatalyst is under illumination and without any bias, the holes and electrons is actually generated randomly. But when we apply a negative or a positive bias, an electron tunnel is formed, such that the charge kinetics can actually being explained or reflected by the photocurrents being recorded from the measurements. So looking back to our results, it tells us that the W3 samples has a higher conductivity, and this reflects the W3 samples has a low charge transfer resistance, higher charge carrier concentration, and superior charge mobility. So to further verify our results from the CAFM, we also continue to investigate the effects that brings by the tungsten towards our bismuth tungstate thin film. And by looking back to our SEM images, we can tell that actually the crystal facets plays a crucial role towards the PEC performance. And from our HRTM image and also the fast spheroid transformation image, it shows that the 0060 and the 002 planes is actually perpendicular with each other. So this tells us that when a higher tungsten concentration is being used, a higher exposure of the 010 crystal facets is formed. And this higher exposure of the crystal facets will actually influence the charge transportation. So we have conducted a photo deposition method by using the silver nitrate as an electron acceptor. And from the TEM-EDS elemental mapping, it shows clearly that the silver metal is actually deposited on the 010 crystal facet, and this attribute as the electron's dominant facets. Meanwhile, when we use the potassium manganese oxide as the host acceptor, and it shows that the manganese oxide are more preferred to be loaded on the 100 crystal facet, and this is correspond to as the host dominant facets. So when the photocatalyst is under illumination, the electrons and holes are more preferred to be transported to the 010 and 10 crystal facet respectively. So when the electrons and holes are being excited in the W1 samples, they have a longer travel distance that will have a higher possibility that the electrons and holes being recombined. However, in the W3 samples that have a shorter travel distance, that will lead to a low charge transfer resistance. So another factors that will influence the PEC performance will be the charge carrier concentration, where the SEM-EDS shows that the W3 samples has the stoichiometry ratio has been increased in the tungsten, which the tungsten enrichment is called a self-dose tungsten phenomenon that will actually increase the charge carrier concentration. And from the EIS Moshocki plots also shows that the so this equation is actually calculating the total number of the donor density, but we can simplify it to donor density is actually inversely proportional towards the gradients from our Moshaki plots. So from the results, it tells us the W3 samples have a less steep slope, so it reflects the charge transfer at the interface of the bismuth tungstate and the FTO substrate is it has been enhanced in our W3 sample. And upon we also use the time-resolved microwave conductance to figure out the charge mobility of our samples, and it shows no significant difference among the three materials. So therefore, it's inferred that PEC performance does not rely on the mobility. So to summarize this project is that by tuning the tungsten concentration will actually lead to a better PEC water oxidation performance, and this is due to the high exposure of the 010 crystal facet, and because of it consists of self-doped tungsten that will also lead to a higher charge carrier concentration and improves the water oxidation process. So as discussed that by tuning the precursor concentration during the fabrication will helps the water splitting process. And next, how we actually exploit this photocatalysis technology for the for removing the nitrates and also producing ammonia. So in one of our other research, we utilized the Seskin system and used for the photocatalytic nitrate reduction. And in this process, nitrate reduction is more challenging because it requires 8 electrons transfer. And again, that we are mainly focused on half reaction and combines them in the later stage, such that we can maybe improve our photocatalyst and for producing more ammonia in our reaction. So as just now when I mentioned that the bismuth tungstate are actually more preferable for the photooxidation process, but there's lack of research or that talking about their photoreduction ability. So therefore, we are very interested to further develop the potential of our bismuth tungstate for the photoreduction process, and by using the bismuth tungstate that we modified in our previous work. So what we understand. First, our bismuth tungstate has a potential for photoreduction because the conduction band is more negative than the redox potential of the nitrate reduction. And also, secondly, our bismuth tungstate possesses a sheet-like morphology that brings a superior charge diffusion and charge transportation, but they have a poor charge mobility and also charge separation ability. So, in this case, how we can improve? And so to overcome this, in this research, we're trying to addition of our co-catalyst, the hole trapping cobalt oxide and also electron trapping copper metal by the photodeposition method. So by looking through our SEM EDS elemental results, it clearly shows that the copper has successfully loaded on the 010 electron dominating facet, while the cobalt oxide are more preferred to be loaded on the 100 host dominating facets. And besides, our XRD and also UV spectra also confirms that with the addition of the co-catalyst does not affect their structures and also their band gap. So after that, we again want to compare their photocatalytic nitrate reduction performance. And from the results, it clearly shows that the bismuth tungstate with a dual co-catalyst will produce about 160 micromole per gram of the ammonia under the 10 hours of illumination, and which is the best among the others. And furthermore, to prove that the ammonia that are produced are originated from the catalyst itself instead of other nitrogen sources, we also conducted categorization by discrimination between the isotopes. And from the NMR results from our ammonia products, it clearly tells us that the ammonia indeed occurs from the reduction process from the nitrates feedstock that we have provided. So here comes the question. Why our bismuth tungstate can actually use for photoreduction, and how the addition of the co-catalyst will lead to a higher production of the ammonia. So first, we have predicted that one of the reasons is because of the presence of the oxygen vacancy. So there are research has been reported that with ultra thin nanosheets morphology with a thickness that below 6 nanometers has a higher chance that have abundant vacancy pairs. So by looking at our AFM and also their corresponding height histogram, it tells us that our bismuth tungstate nanosheets actually has a thickness about 5 nanometers. And then we would like to further quantify the numbers of the unpaired electrons in our samples. We categorize our samples using the EPR. And although that a very weak and also and not a very significant difference in the EPR signals among the light and dark condition, but we still figure out that the bismuth tungstate with dual coil catalyst has a slightly increase in the spin concentration under light illumination. So these results have suggested that the bismuth tungstate with the ultra-thin nanosheets can shorten the transmission path of the charge carrier from the interior to the surface, and the bismuth tungstate with the coil catalyst possesses with more defects that will act as a trapping center for the charge carriers and act as an active site for the nitrate absorption. And the second factors that we figure out will affect the production will be the photochromic effects. Photochromism defines as a reversible change in the color of the chemical species caused by light. And during the photoreduction, it finds out that our bismuth, the photochromic effects of our bismuth tungstate has been triggered because the color has changed from the pale yellow to blue and then to black along the time illumination. And from the transmittance, is also a significant drop is being recorded. So what we can explain here is that when the photocatalyst is under illumination, the photoelectrons has been prompted for a rapid reduction from tungsten 6+ to tungsten 5+ and reach to the saturation point. And the photochromic effects has also occurred at the same time. And this will actually favors the charge separation process. And then when the light is turned off, we figure out the color of our bismuth tungstate has changed back to pale yellow color, and this reflects, and also from our transmission spectra, it tells our bismuth tungstate with dual coil catalyst has a fastest bleaching rate. And this is actually telling us that the electrons and holes pair are not deeply trapped in the defects. They're more of electrons and holes are being transported or accumulated at the coil catalyst that allows more electrons and hole pairs to react with the nitrates for the ammonia production. And lastly, the PL quenching with the presence of hole and electron scavengers also demonstrates that our bismuth tungstate with dual coil catalyst can suppress the electrons and hole pairs. So by looking back to our results, it tells us that there's a larger extent of the PL quenching observed in our bare bismuth tungstate. And what does this imply? So during the measurements, when the photocatalyst is under excitation, our electron scavenger, the silver ions, actually will withdraw the electrons, and the total number of the electron hole pairs will actually decrease. So the PL intensity actually will also decrease, and that will lead to a higher PL quenching extent. So this can implies that our bare bismuth tungstate is a electron minority materials with a high charge recombination rate. So to summarize this project, that bismuth tungstate with the dual coil catalyst will actually exhibit a superior nitrate photoreduction performance, and this is mainly caused by the addition of the coil catalyst will improve the charge transportation as well as suppress the charge recombination. And besides the photochromic effects, it also helps to improve the charge separation efficiency in our materials. So to for my presentation is that the photocatalysis is an advanced technology that harnesses the solar energy to convert the water or the nitrates to form the hydrogen gas and ammonia. and either by the photocatalytic or the photoelectrochemical system. And secondly, our sheet-like bismuth tungsten can be used for the photooxidation and photoreduction process. And in order to achieve a better or efficient photocatalyst, we may actually consider to vary the precursor concentration during the fabrication, which we may able to improve the charge transportation and obtain a higher charge carrier density. And last but not least, that if the photocatalyst with a photochromic behavior and also addition of the co-catalyst, we might actually enhance the charge separation efficiency. Therefore, with these strategies, it might actually help to design a superior or efficient photocatalyst for the photooxidation and photoreduction in the future for the energy and environmental application. So at last, I would like to thank my supervisor, Professor Ng, and Professor Abdi, and Professor Rosa Mal, and all the other collaborators who contribute in this work. Thank you.
Okay, thank you very much for your nice presentation, Miss Chung. Okay, yes. Now the next session for discussion session. Please, if you all the audience have question, please raise your hand. Any question? Yes, Pak Satria.
Yeah, thank you very much. So, yeah, I'm Satria Bisri from TUAT Japan. So, yeah, it was quite nice talk. I'm a bit curious about the how did you measure the conductivity of this system? So, in one of your slides, you show IV profile. So, is it come from like 2-terminal conductivity or 4-terminal conductivity?
You mean the CAFM, that one? Or
yeah, the CAFM. So you only measure conductivity through CAFM, or you also like measure conductivity, for example, like 4-terminal measurement of your film?
Maybe it's too far. So the conductivity, I just direct. So because like we have a from the W3 samples, they have a higher PEC performance, so we want to understand why, right? So that's why we use the CFM techniques to just give an overview of what's happening. At that time, we also apply lights actually in that particular categorization. Sorry, maybe here, right? Yeah. Yep. So in this way, so we just directly like use our film and then use the CFM to apply the bias and then with the lights together, and then we can figure out the results here. So we can compare that actually the W3 samples will have a higher conductivity.
I see. So okay, you took the conductivity from conductive AFM. So in the sense that what you measure is, let's say, the conductivity of like one sheet of the materials, or it's the whole ensemble?
The whole sample. So we cut it into like the similar size, and then we measure it. But because the point that the tip is actually like just one single tip. Yeah, so one single point. So we expect it should be like on the plate on itself. Yeah.
Okay.
But of course, the results here is kind of like average because we have measured in several different spots and then taking the average of it.
Yeah, because I think it should be really careful in doing CFM because the conductivity that you are measuring is actually local conductivity, right? In case of conductive AFM.
Local, yeah.
And then also here, as you can see, the voltage is quite large compared for your measurement, and the current is very small. So I don't think this kind of measurement is quite representative in order to understand the conductivity landscape. Because can you get, for example, mobility out of this material, or the charge carrier density out of this material from this measurement?
You mean like because we apply a high voltage in this circumstance?
Yeah. So can you obtain like the at least two important value that you need to know, right? Mobility and also the carrier density.
Yep.
But from this measurement, I don't think you can get it, right? Because it's local conductivity, and also, it's a bit difficult to understand actually from here.
Well, maybe discuss more later with you.
Yeah, maybe I can also help you to measure conductivity.
Lots of theories and mechanism inside.
Okay, yeah, thank you.
Okay, nice discussion. Thank you, Mr. Satria. Maybe another question. Maybe Mr. Satria asked about how to measure how you make sure that the conductivity is all your material. This is the conductivity of your material, because if you use AFM, it's just a local
just a tip,
little part of your sample, right? Not whole your sample. How you sure that this is the conductivity can belong to your sample, all your sample, not only a little part of your sample? Because if you use AFM, is I think
you mean like a single tip?
Yes.
Yeah, because like most of the people use AFM is like scanning the currents and having the topography, that image, right? But for me, it's more on like we really apply the bias and lights together to figure out, and then in different part of point we have like multiple session, and then so that's why this is kind of the average of it. Yeah.
Okay, not only one point, right? Okay.
It's like circle like at least like 10 different multi sessions.
Okay. Yeah. Okay. Any other questions from the other participant? I think the time. We have still 2 minutes. Yeah. No question. Oh, okay. Mr. Satria again.
So, can you show again the absorption spectrum of the materials?
Absorption.
I mean, do you show any absorption spectrum
the
of this material?
UV-vis?
Yeah, the UV-vis spectrum.
I'm sorry, I passed it. This one?
Okay. I see. But then how about the absorption spectrum of the material that you use to reduce the nitrogen oxide?
This one here?
I see. So, yeah, I just want to know some clarification. So, for this reduction process, you only need the redox potential is 0.55 electron volt, right?
Yep.
Yeah, so in this case you use material with relatively very wide band gap, which is 2.95. So I just want to know your opinion. So for example, for doing this kind of nitrogen reduction, so for example, if I have only material which has band gap 1 eV, will it work or not? Or should I also consider, for example, the redox potential matching and also the homolumo, or the valence band and conduction band of my material besides the band gap?
From my opinion, I think you definitely need to consider like the redox potential first, because it needs to lie in between that you have enough energy or like to reduce your nitrogen species things, nitrogen or the nitrate reductions. And then, but 1 EV, of course, you might need to modify and see how well that can withdraw, like have more electrons for electrons transfer in my case. In that case, yeah, I think you still need to consider like
so basically the redox potential should be within my band gap, right? Okay, then another question is about, have you ever do the measurement, for example, like, okay, in the case of solar cell community, then we usually do IPC measurement, right? So the wavelength dependent.
You mean in different wavelength?
Yeah, different wavelength. So for this kind of, photocatalytic effect, is it common to do such kind of measurement? And then if that measurement is common, so for example, if you use this material, which has like 2.95 electron volt, so then your system is only efficient around blue light, right? So how will you deal with, the utilizations of the green, red, and infrared light of the sunlight to do this kind of photocatalysis?
Well, IPC is common, but not in my case actually. Yeah, but I'm not too sure for this because I didn't really measure it in my case. But well, sometimes I'm not sure. Like some they have large band gap, but doesn't mean that when you have activity, they might you tune something normify and then will actually affect the properties interiorly, such as, for example, like my photochromic effects. Maybe if you tune another way, they don't really have, like if a shorter band gap or etc. doesn't have that effects at all. Yeah, so.
So basically, still we don't have clue how to design material for example, relatively narrow band gap, but it worked for photocatalytic effect. Okay. Yeah, I think we need to find such kind of material.
Okay, okay. Thank you very much, Pak Satria. Because due to the time limitation, I think we can finish this session and we can continue for the presenter, the next presenter. Yeah, 10-minute presentation. Please give applause to Miss Chong. Yeah. Okay. I think what's your name? Another moderator will continue the next session. So I give to you. Thank you very much.
Download semua dulu aja. Okay, thank you so much. Due to circumstances, unfortunately, I will be replacing Ms. Nikma to be a moderator in this room. And once again, thank you so much, Dr. Chung, for the presentation itself. And right now we are moving on to the presenter first. So according to the new rundown, the first one to present their paper and their title is Aulia Kartika. Is the person's here? Okay, so I'll wait a second for the. So the time is yours.
Greetings to all participants of Isoc 2024. Good afternoon, my name is Aulia Kartika and I would like to present our team's research with title Cellulose Acetate-based Hollow Fiber Membrane Coated with LDPE Surface Modification for Desalination Application. And the next, we start to introduction. This is the first about global water crisis. Water is essential for life, making its availability critical. While 97% of Earth's water is wastewater, only 3% is freshwater, including glacier, groundwater, and surface water. However, clean water availability is increasing based population growth, environmental degradation, and industrial expansion, leading to a water crisis where demand excess supplies. This crisis is particularly present in countries like Indonesia. To combat this, researchers are exploring seawater desalination and wastewater treatment as alternative solution. And then it's about desalination technology. Desalination technology, especially through reverse osmosis, is widely use method for producing clean water from seawater by removing salt. And then membrane desalination technology hold great promise for seawater desalination due to its lower operation temperature and pressure compared to conventional desalination and reverse osmosis. And we use to material of membrane is cellulose acetate and polyether sulfone (PES) to make this membrane hollow fiber. And then we use to LDPE, low density polyethylene, to use surface coating membrane morphology. And in this study, hydrophobic additive LDPE is used both on additive and coating material for the membrane. LDPE is non-polar surface enhanced hydrophobic, creating strong, durable membrane with high selectivity and permeate flux. The objective of this study is to investigate the effect of LDPE as the surface modifier with cellulose acetate and polyethylene surface. And then this is the purpose of this study. The first is provide information on the use of cellulose acetate, hollow fiber membrane, and polyethylene sulfone with LDPE additive for transforming seawater in clean water. And the second is gaining research of development result of cellulose acetate, hollow fiber membrane, and polyethylene sulfone in the application of membrane desalination for seawater desalination for further seawater desalination research. And then for the preparation of membrane, we use to NMP solvents with cellulose acetate 15 grams and polyethylene sulfone 15 grams, and we used to homogenize with coating plate and jadinya dope solution. And the fabrication with spinning process, this is a spinning process. There is creating operation condition controller, there's a coagulation tank and collecting drum. There is a membrane hollow fiber, and then immersion of hollow fiber membrane is ethanol for 24 hours, and then drying the membrane for 20 hours at room temperature. For the surface modification membrane, this is a process of coating. The first is weighting LDPE, stirring LDPE and toluenes as solvents as on a hot plate, and then a variation of coating solution, and then making a dope coating solution. And then the result is the Fourier transform infrared. This is a graphic of Fourier transformation is lalu morphology structure. There's a hollow fiber for membrane 1, 2, 3, and 4. There is a finger-like structure, and then we use to analysis water contact angle for LDPE 0%, 2%, 4%, and 6%. That's the 1 until 4 is hydrophobic. And this is a porosity analysis. Its grade highest is LDPE 4%. And this is a DCMD application. We use to membrane for this application, the contact membrane. And then the for performance of CPS LDPE hollow fiber membrane for salt rejection. That's highest is membrane 4, and fluxnya there's membrane 4. And then the conclusion, the cellulose acetate and polyethylene based hollow fiber membrane with NMP solvent and LDPE coating exhibits hydrophobic like a finger-like structure with contact angle rating from additional DFT spectrum range membrane. And then the performance of hollow fiber membrane with the optimal addition of LDPE and membrane desalination application additive on the highest permeate flux of 3.7 kilogram and salt rejection rate of 90%. Okay, thank you for attention.
Please give a round of applause. Thank you so much. Before we go into the next one, is there any questions coming from the floor to Mrs. Aulia? I don't think there's any questions on the floor. If that's the case, you can go back and you can close the session itself.
Okay, once again, thank you for attention and good afternoon.
Okay, thank you so much. Next one is Abu Hurairah. Okay, so after this one, there's gonna be Miss Minisha, but we're gonna go into the Abu Hurairah first. So we're gonna wait. It's already there. Okay, the mic and the pointer also.
Test my 1, 2, 3.
Okay, the pointer is there also. So for wait a second, we are gonna prepare the PowerPoint itself. So sorry, or just wait a second. There's a
Okay.
Kalau sini aja nggak bisa. Ini dia. Oke. Terus saja aja.
Bismillahirrahmanirrahim. Assalamualaikum and very good day to the fellow academicians and researchers. Welcome to Bali. My name is Abu Hurairah from University Science Malaysia. I'm currently in my first year of my master's degree and this is my very first time presenting in international conference. Today I will be presenting my project with the title Effects of Star Anise Polysaccharide Structural Properties on Anti-Obesity, Anti-Hypertensive, Anti-Diabetic and Antioxidant Activities. Obesity is a growing epidemic that affecting millions worldwide. Obesity among adults has been increasing exponentially over the past 10 years. According to the World Health Organization, obesity is defined as having a body mass index of 30 or higher. People with obesity have high possibility of developing chronic heart disease such as hypertension. The number of hypertension among adults globally is not a joke. Hypertension is characterized by pressure in your blood vessel is too high. Similarly, people with obesity have high possibility of developing diabetes. Currently, 10% of global adults have either type 1 or type 2 diabetes. Diabetes is a chronic metabolic disease that occurs when the body does not produce enough insulin or cannot use it properly, resulting in high blood sugar levels. All of the mentioned diseases have their respective medicine. However, current commercial drugs come with several side effects such as stomach cramps, dizziness, and worsening of osteoporosis. Hence, I am proposing the utilization of polysaccharide as an alternative to synthetic drugs. What is polysaccharide? Polysaccharides are long-chain complex polymer composed of repeating units of monosaccharides linked together by glycosidic bonds. In this study, the source of polysaccharide was Elysium verum, commonly known as star anise in English, bunga lawang in Malay, or adas bintang in Indonesia. My interest in star anise has grown due to the previous research found that star anise extract show strong bioactivities. However, as of now, there has been no report on the polysaccharides extracted from star anise and their capability to treat obesity, hypertension, and diabetes. In the same time, awareness of the anti-obesity, anti-hypertensive, anti-diabetic, and anti-oxidative effects of star anise is still low globally. In this study, I plan to use, in this study, I will use deep eutectic solvent or DES as the extraction media for polysaccharide from star anise. What is DES? DES is a mixture of at least one hydrogen bond acceptor and at least one hydrogen bond donor. It has a unique property known as eutectic behavior, meaning melting point of DES is significantly lower than their starting materials. DES is usually composed of HBA and HBD. DS mixture exhibit low melting point, high biodegradability, and non-flammability. They offer advantages such as low toxicity, low volatility, high versatility, and affordability, making them far superior to conventional solvents. In this study, I will use citric acid monohydrate as the HBA and propane alcohol as the HBD. Both of these compounds are certified by the Food and Drug Administration as generally recognized as safe, GRAS. Therefore, these compounds are favored in the preparation of DS due to their safety profiles. So it comes to my first objective to prepare DS using citric acid monohydrate and different propane alcohol as extraction media for polysaccharide from star anise. First, the HBA was mixed with the HBD, and the DS mixture was treated with the ultrasonic bath at 80 degrees Celsius, usually up to 3 hours. Then the DS mixture, the process was considered complete when a homogeneous and transparent mixture was formed. All the prepared DS were observed to be a stable homogeneous mixture without recrystallizing after 24 hours. Therefore, the DS mixture were ready to use as extraction media for polysaccharides. After preparing the DS, my second objective was to optimize the microwave DS extraction parameters for the extraction of polysaccharides. First, star anise, for the extraction procedure, first star anise samples that purchased from supermarket were ground into powder. The star anise powder was mixed with the DS prepared in phase 1. Then the mixture was placed in a microwave under specific parameters. After the extraction, the samples were centrifuged and the supernatant was collected. This filtrate will precipitate by adding 4-fold of ethanol, and the precipitate which accrued polysaccharide was centrifuged, collected, and lyophilized. Suitable range for the extraction factors in the study were established using a single factor experimental design. After that, the Bose-Benken design was used to optimize the effects of the single factor experiment, the single factor variables on the response, which is extraction yield, using the design expert software. In this study, the obtained data were analyzed using one-way analysis of variance and Duncan tests using SPSS software. This is the optimized extraction condition, resulting in 5.14% of extraction yield. In comparison, in the single factor experiment, the extraction yield was 4.86%. After obtaining the optimized extraction condition in phase 2, the bioactivity properties of the extracted polysaccharide were evaluated. These properties will involve physicochemical properties, functional properties, and structural characteristics that combine to their bioactivities. The bioactivity analysis of polysaccharides was involved several assays, followed by analysis for their physicochemical properties, functional properties, and structural characteristics. This is important to understand how these characteristics contribute to their bioactivities. The bioactivity of star anise polysaccharide or SAP are quite strong, as evidenced by the following. ABTS scavenging activity at 34%, FRAP capacity at 0.56 millimolar, lipase inhibition at 86%, ACE inhibition at 73%, and alpha glucosidase inhibition at 82%. In summary, SAP proved its significant bioactivities. Based on the physicochemical properties, SAP was an acidic polysaccharide. The protein content was very low, indicating that the high purity of extracted polysaccharide. The total phenolic content was 2%. This is found. This is because phenolic content are commonly found conjugated with the polysaccharide structure. Molecular weight of SAP was 26 kilodalton and considered as low molecular weight polysaccharide. Based on the monosaccharide composition, galactronic acid was the predominant sugar. SAP was considered a pectin-like polysaccharide because of their backbone was galactronic acid, but the abundance of the galactronic acid is less than 50%. According to the FTIR spectrum, the C double bond O of ester group was observed at 1700, while the COO minus of an esterified group were observed at 1600. The degree of esterification of SAP was 72%, classified as high DE. The high DE polysaccharide was believed to reduce enzyme activity by forming gel-like structure and preventing the access of enzyme to the substrate. A small diffraction peak was observed in the range of 15 to 25 degree, meaning SAP was in an amorphous state. Previous study found that the polysaccharides extracted from plant sources often exist in amorphous state. SAP exhibited uneven ellipsoid and polygon flake morphology. Previous studies have suggested that the irregular morphological form and amorphous structure of polysaccharides might contribute to the strong bioactivities. The oil holding capacity of SAP was 14.39, while the water holding capacity was 2.99. This significance of OHC indicates that the polysaccharides can attach and immobilize the lipase enzyme, thereby preventing the enzyme from accessing to the fat substrate. In conclusion, first, the propane alcohol base using citric acid monohydrate and propane alcohol were prepared. The extraction parameter of microwave DSS system were optimized for extracting polysaccharide from star anise. And lastly, the bioactivity properties of the extracted polysaccharide were evaluated. These polysaccharides have shown to be the potential to be developed as alternative to synthetic drugs. Lastly, it was suggested that the physicochemical properties, functional properties, and structural characteristics significantly contributed to their bioactivities. That's all from me. Thank you.
Thank you so much for the presentations. Is there any questions from the floor regarding the presentation? Okay. Wait a second.
Yeah, thank you. My name is Araman from Nano Center Indonesia. Interesting talk. Thank you. I learned new information about your research. May I ask, I'm not so familiar with how one can determine the degree of esterification from the FTIR spectra. Could you mind to help me out to understand how you can shed some light about the extracted numbers from your slide? Yeah, thank you.
Thank you for the question. Okay. The degree of esterification have its formula. Okay. The formula is the area for C double bond O ester carbonyl group minus with the COO unesterified carboxylate ion, and then the total will divide by the value of C double bond the surface area for C double bond O ester carbonyl group, and then times by 100, so we get the percentage of the degree of esterifications. Okay, thank you.
Okay, is there answering? Okay. Once another questions.
Thank you so much for that presentation. I like your name, Abu Huraira. It's a nice name. But so, did you analyze your DES after reaction?
Yes, I have analyzed the DES after the.
Are they still DES? Are still the same starting material?
Sorry?
Are they still the same starting material? Because you are extracting polysaccharides, right?
Yes.
Polysaccharides have potential to make DES too, because you are starting from your donors and acceptors.
Okay.
And these polysaccharides can actually react with your DES to replace
your either donor or acceptor based on the reaction.
Okay.
Therefore, you will probably land up with a mixture of this that incorporate your polysaccharide.
Okay. Okay. So, very good question. Actually, yes, DS also can be formed from the polysaccharide structure like glucose and then mannose. But in the here, when we precipitate the polysaccharides, eventually we will use ethanol. Then the ethanol itself will wash all the ethanol will wash off the DS starting material in the polysaccharide. So to confirm this observation, I was conducted the FTIR spectrum. I conducted the FTIR, and there has been no indication or no peak that contribute to the DS starting material. That's why we believe that our DS is totally washed off from the polysaccharides.
Is there any following questions? No? Is it answering enough?
You have shown a table of activities, yeah? You have shown a table of activities, bioactivities. What is the benchmark of these numbers, right? So you have shown...
You mean...
That's the one before that, I think.
Can you help me?
The table before that.
Non-functional.
Sebelumnya.
This one?
Yes. So
Okay.
what is the benchmark? What are you comparing for?
I will comparing with the control positive control. For example, the pancreatic lipase will use the orlistat as the commercial anti-obesity, while ACE with the alpha glucosidase with the acarbose, ABTS with the vitamin C.
I think it's good to show the numbers so that we are able to compare your values with the actual benchmark drugs. Yeah.
Okay. Thank you for your comments.
Okay, thank you so much. I think the time has elapsed, and you can close your presentations.
That's all from me. Thank you.
Thank you so much, Mr. Abu Huraira, for the great speech. Right now, we are moving on to Miss Minisha Sivakumar. I think it's over there. That's totally fine. We're just gonna wait for a while since right now we have still need to set up the PowerPoint. And by the way, the time is gonna be 15 minutes, and I'm gonna be reminding, so please take a look here. Okay. The time is yours.
A very good afternoon to everyone. My name is Minisha Sivakumar and I'm from University Science Malaysia. I would like to present on repurposing agricultural and plastic waste from geopolymer synthesis. Let's look into palm oil. Palm oil is a vegetable oil come from the fruit of palm oil tree. These are the history of palm oil. In 17th century, it's traded by European merchants for industrial use. Followed by 9th century, it became a major export from West Africa. It introduced to Malaysia as an ornamental plant in 1870. Followed by in 1917, it is the first commercial plantation established in Malaysia. Malaysia government launched a major palm oil cultivation scheme in 1960. At present moment, Malaysia and Indonesia account for over 90% of global palm oil production. Palm oil can be used in personal care such as cosmetics, soaps and shampoo. While for food it can be used as a cooking oil and in processed foods, and it also can be used in industrial as a biolubricant and also biofuel. Spent bleaching clay. Spent bleaching clay is a solid waste from palm oil. It produces from the bleaching process of crude palm oil. It removes impurities and color pigments. It also can be used as a base material for geopolymer production. Currently, the major problem is there are tons of plastic waste have been produced globally. Approximately 19.7% of PP has been produced. Although it is good for the recycling, but it environmentally it impact the environment and also the health of humans. Besides, the annual productions of spent bleaching clay reached around 200 million tons. This SBC is normally discarded in landfilling or not in a proper way. So, to overcome this problem, we came out with a geopolymer technology. We use spent bleaching clay as the aluminum silicate material for geopolymer synthesis. And also, due to the high temperature resistance, we use PP plastic as aggregates to the geopolymer system. The advantages of geopolymer, one of them is reuse of industrial waste. It also reduces global warming, reduction in carbon dioxide emissions. When compared to other traditional Portland cement, geopolymer produce less CO2 carbon dioxide, and it also eco-friendly and sustainable. The lifespan of geopolymer is higher when compared to conventional cements. These are the geopolymerization mechanisms. It consists of 3 steps. The first one is dissolution. The aluminum silicate will be mixed. Aluminum silicate material will be mixed with the alkaline activated solutions. During the dissolution process, high concentration of the hydroxide ions will react and the aluminum and silicate ions will be released. The second step will be the condensation, where the alumina and silicate ions will be condensed to form the aluminate silicates oligomers, followed by the hardening and curing, where it will be form the 3D gel geopolymer. These are the flowchart of geopolymerization of spent bleaching clay or PP spent bleaching clay to alkali activation. First we will weigh the sodium hydroxide, sodium silicate and spent bleaching clay in a separate beaker, and the sodium hydroxide and the sodium silicate will be added, and it will be form alkaline activity solution. The solution will be poured into a beaker containing spent bleaching clay. Then the mixture will be stirred for 15 minutes. Then we will be obtain the paste and will be dried in oven for 80 degrees for 24 hours, followed by fumed for 7 days. Then we will be obtain the spent bleaching clay geopolymer. These are the results we obtain. On the right is the PP SBCG and on the left is the spent bleaching clay geopolymer. We use different mass ratio of SBC to PP. Let's look at the characterizations FTIR. As you all can see, after the geopolymerization, the band at the range of 700 for SPCG has been reduced. And also a new band is observed at the range of 1500 for the SPCG. It is due to the OH bond in the geopolymer backbone. And also after the geopolymerization, the bands at the range of 1700 in SBC is disappeared in SPCG. This bond is related to the residual of palm oil, which is triglyceride. It is not present in SBCG due to the OH bonds in the geopolymer backbone. This is the TG analysis. The first decomposition step at the range of 270 degree to 530 degree is due to the decomposition of residual palm oil on the geopolymer, followed by the second decomposition step at range of 620 degree to 70 degree is due to the dehydroxylation of the geopolymer matrix and the formation of new silicon-oxygen bond. And this is the BET analysis. As you all can see, all the samples exhibits type 4 isotope, while SBC and PP plastic exhibit type H3 hysteresis loop, which is due to the presence of non-rigid aggregates of plate-like substances, while SBCG and PBSBCG exhibits type 1 hysteresis loop, which equal to the porous materials. These are some of the applications of the geopolymer. It is used for the durable constructions, high temperature resistant, 3D concrete printing, pavements, and also for the lightweight applications. In conclusion, the clay-based geopolymer was synthesized using SBC with PP plastic added as aggregates. The FTR results indicated the presence of silicon-oxygen aluminum bonds confirming the formation of geopolymer. The BT results show that both the raw and the geopolymer exhibited the type 4 linear isotope, which is H1 and H3 hysteresis loop, and PP plastic as aggregates improve the thermal stability of geopolymer. These are the references. I would like to thank the School of Chemical Sciences, USM, and also my supervisor, Dr. Sangeeta, and KLK Kepong Olimas for providing me the funding. That's all from me. Thank you.
Thank you so much. Is there any questions on the floor to Mr. Shang, to Mrs. Minisha? Is there any questions on the floor? I think no, and just you can conclude your presentations.
That's all from me. Thank you.
Thank you so much. Right now we are moving on to, I'm gonna cross-check this first. Agus Rianto, is there anyone named Agus Rianto here? Okay, I think no. Okay, next up, meaning with the name of Miss Rui Liu. Okay, the times to present is yours. Just wait a second. I'm gonna. Okay, could you try the remote first? Okay, so the time is yours.
To be here to share my work here. My presentation is about incorporating zinc metal sites in aluminum coordinate porphyry metal organic frameworks for enhanced photocatalytic nitrogen reduction to ammonia. As well known, ammonia play important roles in our life and in the industry. First, ammonia is the raw material to produce fertilizer, which could boost world population increasing. And also, ammonia is a critical material for produce chemicals such as nitric acid and polymers. More importantly, ammonia has been considered as an ideal zero-carbon hydrogen carrier. The current ammonia production highly relies on the traditional Haber-Bosch process, which operate under high temperature and pressure, resulting in enormous energy consumption and CO2 emission. To this end, it is highly desired to explore a novel and sustainable ammonia production technology. Photocatalytic nitrogen reduction reaction is a mild nitrogen fixation technology that utilize solar energy to directly convert nitrogen and water into ammonia, assisted by the semiconductor-based photocatalyst. With the light irradiation, the electrons in the valence band could be excited to the conduction band and migrate to the surface of the catalyst and then react with the nitrogen absorbed on the catalyst surface. Compared with the Haber-Bosch process, photocatalytic NRR could be achieved under normal conditions without heat input and carbon emission. Recently, the semiconductor-based photocatalysts, such as metal oxide, metal sulfides, big smooth oxyhalides, and carbonaceous materials, has been investigated for photocatalytic NRR. However, these semiconductor-based photocatalysts afford relatively low solar to chemical conversion efficiency because of their poor light absorption ability, small surface area, and limited active size. The metal organic frameworks could provide an ideal platform to address the above issues. Both possess a similar energy band structure to semiconductor-based photocatalysts. The lowest unoccupied molecular orbital is similar to the conduction band, while the highest occupied molecular orbital is similar to the valence band in the semiconductor-based photocatalyst. The unique, compared with the semiconductor-based photocatalyst, the unique inorganic-organic hybrid structure of MOFs contributes to the improved light absorption ability and enhanced charge separation. So with this similar energy band structure to the semiconductor-based photocatalyst, MOF could be used as photocatalyst for NRR. Especially aluminum-based porphyrin MOFs, namely aluminum PMOF, could be a promising candidate for NRR. Aluminum PMOF is composed of porphyrin ligands and aluminum metal nodes. Thus, it inherits the excellent optical properties of porphyrin and the superior stability of aluminum carboxylate coordination. Moreover, the 4 nitrogen atoms of aluminum PMOF could bond with metal to form unique metal nitrogen 4 sites, which could be potential active sites for NRR. Inspired by the potential roles of ion in the artificial nitrogen fixation, we selected divalent ion with full and incompletely occupied 3D orbitals into aluminum PMOF. We also select zinc with completely occupied 3D orbitals into aluminum PMOF for comparing the influence of different metals on photocatalytic NRR. First, aluminum PMOF was synthesized by a reported hydrothermal method, and then the metal-incorporated aluminum PMOF was synthesized by a post-synthetic method. We then conducted some characterizations to confirm their structures. First, the XRD pattern of aluminum PMOF zinc and aluminum PMOF ion display the similar peaks to Princeton aluminum PMOF, indicates that metal incorporation does not destroy the crystal structure of aluminum PMOF. We further use XPS to see whether metal ions have been incorporated aluminum PMOF successfully. The zinc 2p spectral of aluminum PMOF display 2 peaks assigned to the divalent zinc, and its nitrogen 1s spectral display a peak. The peak corresponding to the zinc nitrogen bond. Similarly, the iron 2p spectral of aluminum PMOF ion display the peak assigned to the divalent ion, and its nitrogen 1s spectral also shows the peak assigned to the iron nitrogen bond. So this XPS analysis verified that metal ions have been incorporated aluminum PMOF successfully, and ICP result further verify this conclusion. Since we have prepared aluminum PMOF zinc and aluminum PMOF ion successfully, we then test their performance for photocatalytic NRR. It can be found that aluminum PMOF zinc and aluminum PMOF ion display superior performance than Princeton aluminum PMOF, and their ammonia yield rate is enhanced by 1.6-fold and 0.9-fold compared to Princeton aluminum PMOF. The stability test of aluminum PMOF also show stable ammonia production yield. So aluminum PMOF zinc possess the best photocatalytic performance and excellent stability. We may be curious about why metal incorporation could enhance the performance of aluminum PMOF, and why inserting different metal into aluminum PMOF could result in different performance. We first explore the nitrogen activation of these samples. The peak at about in the nitrogen TPT spectral, the peak at about 100 Celsius could be assigned to the desorption of the physically absorbed nitrogen on the catalyst. It could be inferred from the peak intensity that metal incorporation could enhance the amount of the absorbed nitrogen. Notably, aluminum PMOF zinc display a higher desorption temperature than aluminum PMOF iron and aluminum PMOF. Because the desorption temperature is highly associated with the interaction between nitrogen and catalyst, the higher desorption temperature of aluminum PMOF zinc than that of aluminum PMOF and aluminum PMOF iron indicates a stronger interaction between nitrogen molecules and the aluminum PMOF zinc. So the superior performance of aluminum PMOF zinc should originate from the easier nitrogen activation induced by the zinc nitrogen size. We further investigate the charge separation ability of these samples. From the steady state PL spectrum, the PL spectrum show two signals, and it's obviously that the peak PL intensity of aluminum PMOF iron and aluminum PMOF zinc is significantly lower than that of aluminum PMOF, indicating a surprise charge recombination. We further get the lifetimes of the charge carriers by fitting the TR-PL spectrum. It can be found that the lifetime of the charge carriers in aluminum PMOF zinc and aluminum PMOF iron are longer than those in aluminum PMOF and aluminum PMOF, both into inspection wavelengths. So this result indicate that inserting metal trapping sites by incorporating metal ice into aluminum PMOF could promote the charge separation and then resulting by a superior performance. We also explore the charge transferability of the samples. The aluminum PMOF possess a highest charge transfer resistance, while aluminum PMOF zinc and aluminum PMOF iron display smaller charge transfer resistance, both in the dark and under light conditions, indicate enhanced charge transfer ability upon metal incorporation into aluminum PMOF. We also get the energy band structure of these samples by Moshoki plots and UV-Vis DRS spectrum. The lumen level of aluminum PMOF zinc is determined to be minus 0.65 volt, which is more negative than that of aluminum PMOF and aluminum PMOF iron. The more negative lumen level could provide more energetic photoelectrons with a stronger reduction ability. So the superior performance of aluminum PMOF zinc than aluminum PMOF iron could be induced by this stronger reduction ability. So in conclusion, we synthesize metal incorporated aluminum PMOF for photocatalytic NR. Metal incorporated aluminum PMOF exhibits superior NR performance than persistent aluminum PMOF. This superior performance may originate from the unique metal nitrogen sites, which could activate nitrogen and improve the charge transfer and separation ability. We also found that incorporating different metals into aluminum PMOF could modulate the energy band structure and result in different NR performance. Thank you.
Thank you so much. Is there any questions on the floor for Miss Liu here? Sorry. Is there any questions? Okay. So, Mr. Satria.
Hello. Thank you very much for a nice talk. So, yeah, I'm not that familiar with this metal MOF. So, I mean, from your measurement, you obtained that the band gap is of this material is around 1.82 electron volt, and it doesn't change by changing the metal core of the MOF, right?
Yeah.
So, this 1.82 electron volt band gap is associated with wavelength in your photoluminescence. Yeah, I mean, yeah, this is your band gap, right? So,
Yeah.
and then on the other hand, you also do the photoluminescence measurement, the PL.
Yeah.
And then you observe two different peaks, right?
Yeah.
And then, so those two different peaks are attributed to what kind of transition? So which one is the band gap? Which one is the, yeah, this one. So 1.83 is the left peak or the right peak?
Oh, yeah, we get the band gap from the right peak.
So, okay, the right peak is 1.83 electron volt.
You mean?
Yeah, I mean, yeah, you have the band gap. So you have one value of the band gap, right? But
Yeah.
here you have two peaks of
Yeah, yeah,
your
yeah.
photoluminescence. So can you explain? I mean, the left peak is from which transition, and on the right peak is from which
We
transition?
think the first peak should originate from some the trapping sites in our photocatalyst in the recombination between the trapping site to the
Mhm.
ground state. Well, the second peak should be the main contribution for the band gap. I mean the charge recombination process occurring in our photocatalyst.
I see. So you mentioned the left side is from the trap states, or is it the other way around?
Yeah.
So the left side is trap states.
Yeah, the trapping states in our MOF
I
photocatalyst.
So, but doesn't it strange that the trap states is above the band edge for transition?
Yeah, because when the electrons are excited from the grounded state to the excited state, we found from the reference that the main peak, I mean the prominent peak, should be induced by the recombination of the single state to the grounded state.
Okay.
Well, the small peak, minor peak, should from the excited carriers, such as in the trapping site, not very as high as that of single light state, to come back to the ground state.
I see. Yeah, because I found it a little bit strange, because usually the trap state should be below the band gap, right?
Yeah.
Okay, so but here you show the trap state is above the band gap, so it's a bit strange that it can be excited in such a way.
Yeah.
Okay,
Yeah.
thank you.
Thank you so much. Since looking at the time, we do have time constraint. So thank you so much. And you can conclude your presentations. Okay. Next up is Adik Roni Setiawan. Okay. We're gonna wait for the PowerPoint first. Okay, the time is yours.
Tes. Okay. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. Please let me introduce myself. My name is Ronnie. In this occasion, I will present to you about my research entitled The Effect of Varying Types of EPDM Curing Agents to IRM 903 Swelling Resistance using Fractional Vectorial and Artificial Neural Network Methods. The background of my research is sourced by the Mordor Intelligence, which predicted the EPDM market will be increased at 6.47%, which mainly affected by the growing of the automotive industry, which turned it back to the polymer material that used to reduce the total car weight to get better fuel consumption and increase engine efficiency. EPDM itself is a monomer of ethylene, propylene, and diene monomer. It is a cheapest elastomer known in the market, but also it has some advantages for having the properties of thermomechanically stable and easy to be processed. But EPDM also have some disadvantages because it has a non-polar bonds which make it to be more susceptible to oil swelling. Commonly, EPDM are used in the hose radiator on car. Commonly, problem on the radiator hose is because it swell if there is some leakage on oil, which make it swell and tear, which can result in fatal engine damage due to overheating. To overcome this problem, we need to do some EPDM compounding by modification of the curing system, which consists of accelerator and curing agent. The development of the EPDM compound commonly used in industry was using the method of trial and error experiment. Also known that the EPDM are classified as a thermoset elastomer. It cannot be recycled and it will make a lot of waste and make a lot of cost. To minimize the cost of development of EPDM compound, it needed to do more controlled experiments such as the design of experiment or DOE. The common DOE is the fractional vectorial design, which commonly correlated to the factor that used in the experiment run, and also it is affected by the variation of material and processing conditions that could make the bigger errors in the result. So to reduce the experiment cost, we can use the DOE data as an input for INN model, the artificial neural network. INN can predict the simulated compounding result using the experiment result of DOE datasets. Here is some literature review. The first one is from Mayasari. This result is resulted in that the accelerator CBS could give better swelling resistance from oil with swelling degree of 3.2%. It is concluded that the accelerator CBS is giving the better swelling resistance. The Carrero, from his research, he resulted that the ANOVA method used in the two-factor factorial design of experiment of developing the self-healing rubber compound can validate that the crossing density was the main factor of the mechanical properties of the rubber compound. And then from the Ruziak, he done research about the artificial neural network that used to predict the mechanical properties of natural rubber compound using RMSE, which at its predicted value are having the R square better than 0.93 or very accurate. So the research purpose of my research is to determine the optimal of oil resistant characteristic of EPDM with various curing system composition using the fractional factorial DOE to identify the combination of curing system that most significantly affect the durability of EPDM compound to oil IRM 903. So, and the data can be used as a data input for the ANN predictive model that is able to predict oil resistant performance based on experimental data. Through this objective, this research is expected to contributing the development of better oil resistant EPDM compound as well as providing an effective method for analyzing and predicting the performance elastomer materials using a combination of experimental design and artificial intelligence. The methodology. Here is some of the tools and materials that I use in the research. The research are done using the PT Pilatus Indonesia Material Research Development Facility, and all material used in this research are industrial grade material. In the first step of this research is the parameter selection. The first one is the constant parameter selection, which is in this case is the master batch formula that's shown in the table. It is processed by using the kneader and open mill. And then the process parameter is the independent variable or free variable. It is a variation of curing system material that's selected during research, which consists of curing agent and accelerator. The formula is shown in the table. It has some min and max value for the research. It is processed using open mill machine. There is some the testing process. The response parameter is a result from various analysis steps used as a simulation input. The test piece is used in this research is using dumbbell and chest box using the standard of GIS K651. It is processed using molding machine. Here is the sum of the test analysis item. that figurized in this DOA matrix. The software used in this result is Minitab for the DOA design and MATLAB to be used in artificial neural modeling. And then this is the step for the DOA creation. It is tested using ANOVA test and main effect plots. And this is the chart for the artificial neural network from the data normalization, INN model creation, denormalization, and analyzing the predicted value. Result and discussion. Here is the result of the main effect plots of the DOE experiment. It is the mechanical properties consist of the hardness, tensile strength, elongation at break, and the specific gravity. It is shown that the curing agent that have significant influence on the mechanical properties of EPDM, especially on variation, is the sulfur and peroxide. These two agents act as a cross-linking agent in volcanicity using a combination of sulfur and peroxide, which can be shown at higher tensile strength and elongation compared to those using only one of the system. For the rheometry, there is a lot of inaccuracy, but there is some significant value, significant effect on the accelerator CDBC and sulfur. On the heat resistance, the main significant parameter is the accelerator, because accelerator in EPDM, which in EPDM can determine the crosslink structure based on the sulfur and accelerator ratio. In EPDM, commonly ratio is the CV, semi-efficient, semi-EV, efficient EV, based on the sulfur and accelerator ratio. And also on the IRM 903 all resistant, the best swelling degree, the lower swelling percentage, the higher the resistance to solvent is by the parameter of sulfur and CBS. Also for the artificial neural network, both in the mechanical properties, we saw that the predicted value for hardness and tensile strength is very accurate, but there is some inaccuracy in elongation. But in the rheometry, the accurate predicted value is only on the ML or the Mooney law, but on the image or the score time and the curing time is not inaccurate. And then for the heat etching resistance, here is the result of the prediction. The etching value obtained from the EB has good MSE, but there is some inaccuracy. For the hardness, there is a lot of accurate. And then the aging, so some inaccuracy. But on the IRM 903, for the main factor of the result is the volume cell. It can predict the value accurately. So the conclusion for the result is that the fractional factorial experiment design method showed that the best oil resin was obtained in the first run order, where in this variable the curing agent was used on the maximum level. This shows that the oil resin is influenced by the cross intensity and voltage, and the INN method can predict the model very accurately. Thank you very much.
Give a round of applause, please. Thank you so much. Is there any questions from the floor to Mr. Ronnie? Is there any questions from the floor? There is none, so you can conclude the presentation.
Okay, thank you very much. That's all for me.
Okay, thank you so much, Mr. Ronnie. Right now we are moving on. Okay, I'm gonna check once again. Mrs. Wan Nazwani Wan Abdullah, is she here or he here? Is the person here? No? Okay, I'm gonna go jump into the next one. Elsa Bella Adiguna, is the person here already? Okay. Please wait a second till we prepare the PowerPoint itself. time is yours.
Okay, good afternoon, everyone. Today I would like to present my research. The title is Addition of Non-Toxic Organic Pigment as Inorganic Cadmium Pigment Replacement in the Denture-Based Production from an Aesthetic Perspective. And these are my team from ITS Surabaya. Edentulous is a condition where the patient has no teeth, lack teeth, or experience tooth loss, and it often happens in the elders, where removable denture have been used for decades to replace function and aesthetic in edentulous patient. Denture base is a part of removable denture, which is defined as the base tissue to which the teeth are attached. The material for commercial denture base is based on acrylic resin with a polymer base of polymethyl methacrylate, PMMA, which is available in the form of a powder liquid system, which is head cured, where the powder and liquid consist of this material, as follows. One of the most important factors in material for denture base to support the success of restorative treatment in edentulous patient is the aesthetic factor. The base gingival color and pigmentation color must be adjusted to the denture base to make it look more natural by adding pigment to the resin composition of denture base. Where inorganic pigments such as cadmium sulfide and sulfoselenite have been used for many years to color denture-based resin, but now the use of cadmium-based pigment is strictly limited due to their potential toxicity. And to overcome this, we can use the organic pigment system such as Chromtal Red BRN or the Pigment Red 144, which is much more safer and has a high color strength. And it is also necessary to pay attention to the concentration of pigment in the denture-based resin, because in some cases, when the proportion of color powder is too large, the acrylic resin will not absorb all the color material, and this excess powder will interfere with resin polymerization and affect its mechanical strength. And the materials and method of this research is the material we use is as follow, and the method is first production of PMMA polymer using suspension polymerization process. We self-product the PMMA polymer in the laboratory using this set of instrument. And then next, the desalinization process, hydroxyapatite treatment with gamma MPS. And then mixing of powder, we mix powder that contain PMME, BPO, silanese HAP, and pigment in the V-blender mixer, and then we mix powder and liquid in the mixed bowl with hand mix. And then the last molding process and head curing for polymerization in a water bath. The operation condition in this research is as follow, and the variable we use is the first type of pigment. We use organic pigment and inorganic pigment. For organic pigment, we use Chromtal Red BRN, and for inorganic, we use cadmium sulfoselenite. And then we also vary the pigment concentration in a powder composition. Result and discussion. First, we analyze silanese hydroxyapatite characterization. This is show the FAR analysis of silanese hydroxyapatite, where the two peaks of silanese and commercial HAP show the hydroxyl and phosphate group, which is the two special group of hydroxyapatite. The difference between these two peaks is in the silanese hydroxyapatite appears silanol group and C double bond group, which means that the silanization process is success. Silane has the ability to bind inorganic material such as hydroxyapatite to PMMA organic resin, resulting in better mixing, better bonding, and increased metric strength. And then next, the aesthetic perspective of denture-based specimen. We use digital photography method like in the FAFI Research 2016 to get the LAB value of each specimen by taking photo of each specimen and then analyze it using Photoshop software. The setting and digital camera we use is as follow. And this is the result of the aesthetic aspect. In this graph, as we can see in this graph, is the x-axis show the pigment concentration. In the above is a bare and red chromoptal, and the y-axis show the LAB scale. As we can see in the bar graph, L value, which is indicates light versus dark, is decreased with increasing red pigment concentration. And according to ASA research, the A and B value should increase with increasing red pigment. But, however, while we add the bare and red chromoptal pigment concentration in the powder composition, the A value decreased slightly and then didn't change in a certain variable. This is a proof that the bare and red chromoptal pigment has a high color strength. And also from this result, we also can conclude the range of the healthy gingiva and alveolar mucosa, which is from Huang and Ho research. In Asia, healthy gingiva has a specific range where the alveolar mucosa color usually darker and redder than the gingiva color. And then from this result, we can conclude that if we use chromoptal red bare and organic pigment, we just need 0.01% of pigment to imitate the darker color of gingiva mucosa, of alveolar mucosa, and then we need less than this to imitate the gingiva color, which is more brighter. But if we use the cadmium sulfoselenide, we need 0.04% of cadmium sulfoselenide pigment to imitate the gingiva color, and we need more than this to imitate the alveolar mucosa which has more darker color. And the conclusion of this research is the result of FTIR analysis of silanese hydroxyapatite show the presence of silanol group, which indicates that the hydroxyapatite silanization process was success. And the result of optical analysis of denture-based specimen show that the addition of organic pigment bare and red chromoptal at just minimum concentration of 0.01% alone can imitate the darker color of alveolar mucosa, so that the use of organic pigment chromoptal red bare and can prevent disturbance in mechanical properties caused by disruption of composite polymerization.
Okay, thank you so much. Give a round of applause to Mrs. Elsabella. Is there any questions that wanted to ask to Ms. Elsa here? I guess none. Just thank you so much, and just conclude your presentation. Right now, we are moving on to Ms. Zela Marni. Is okay. Please wait for a second. That we are. It's okay if you wanted to come front. We're just gonna wait a second until we are preparing the PowerPoint itself.
Okay, thank you. Good afternoon. Now I will present my research title is the influence of silanized zirconia fillers on the impact resistance of artificial tooth composites. My name is Zela Manisa Fitri, and they are my teams, and I am from Institut Teknologi 10 November. Okay, this country, with a population of around 273 million people, unfortunately face serious dental health issues. According to the World Health Organization, 43% of the Indonesian population suffer from tooth decay. The impact of severe tooth decay goes beyond just the loss of teeth. It also leads to various other health problems if missing teeth are not promptly replaced with denture. Fatal consequences such as the shifting surrounding teeth, speech disorder, decreasing chew efficiency, and even jaw displacement can occur. Therefore, dentures are essential to replace missing teeth and prevent various health issues. There are several types of denture available with different material and types to choose from, such as porcelain denture, acrylic denture, metal denture, and nylon denture. However, acrylic denture remain the best choice considering their cost, biocompatibility, and ease of processing. These dentures are generally made of PMMA, who has the drawback of low mechanical strength. To overcome this limitation, it's necessary to add filler materials in this study. Zirconia is used as the filler. Zirconia can enhance impact strength, fracture resistance, hardness, and matrix density. Additionally, its white color does not alter the aesthetic as it closely resembles natural human teeth. However, to successful combine zirconia with PMMA or matrix, it's necessary to consider the surface modification of zirconia nanoparticle by using coupling agent, commonly known as silicization. Okay, here is an illustration of zirconia before and after silicization. Before silicization, this zirconia fillers merely fills the empty space in the matrix without forming a chemical bond. This can cause the filler to easily detach and making the composite prone to cracking. And after modification, the zirconia fillers fills the empty space in the matrix and form a chemical bonds, making it less likely to detach and resulting in a stronger composite. Here, gamma MPS is utilized as the coupling agent, which can reduce zirconia nanoparticle aggregation and enhance its compatibility with the polymer matrix. This helps in achieving a uniform distribution of zirconia nanoparticle within the matrix. Optimal absorption of silane on the zirconia surface is necessary to establish strong bonds. Additionally, the amount of filler added to the composite must be also optimal. The fabrication of denture composite involves mixing powder and liquid component. After achieving a homogeneous mixture, polymerization is initiated with heat associated, resulting in denture composite with favorable mechanical characteristics. In this study, heat cure polymerization is used for denture fabrication, utilizing heat from a water bath to activate the initiator. The following are the stages for denture fabrication that occur outside the mold and inside the mold. Okay, and the methods, number one, PMMA synthesis using suspension polymerization process, and this is a diagram of equipment setup for PMMA production using suspension polymerization process. And then silicization of zirconia fillers with gamma MPS, and mixing powder and liquid, and last curing process with in water bath or bulk polymerization. And the materials, monomer methyl methacrylate, benzoyl peroxide, polyvinyl alcohol, aqueous, nitrogen gas, zirconium, gamma MPS, toluene, dan ethylene glycol dimethacrylate. And the operating condition, mass of BPO 0.5%, ratio of liquid MMA and EGDMA 9:1, ratio of powder liquid 2.5:1, bulk polymerization temperature 74 Celsius and 100 Celsius with two-step heating, and mold polymerization pressure 40 MPa, bulk polymerization time 120 minutes, and concentration of gamma MPS 2.5%, and the variable concentration of silane treated zirconium oxide 1, 2, 3, 4, 5%. And result and discussion. The FTIR analyzed result for the produced denture composite show stretching vibration in 750 per centimeter and 483 per centimeter, indicating the presence of ZRO metal oxygen bonds. And the silicization of zirconia is marked by the presence of ZROSENCSE. Is marked by the as with the hydroxyl group organizing from the hydrolysis of COCH3 groups. Additionally, peaks for CHCC and CO, which come from the gamma MPS monomer and the matrix. The analysis confirmed that the composite successful integrate silantrate zirconia with gamma MPS, resulting in a material with strong and stable chemical bond. And this is a mechanical analysis result. The impact strengths indicate that the higher the concentration of silanized zirconia add to the PMMA denture composite, the higher the resulting impact strengths. The results show that composite with silanized zirconia concentration of 2, 3, 4, and 5% meet the minimum impact strength standard for denture according to ISO 22112 2016, which is 2 kilojoule per square meter. And the conclusion, the heat cure polymerization method has been successfully used to create PMMA composite denture, and this is demonstrated by the composite that has been met, where the silantrate zirconia has been successfully integrated into the denture composite is as evidenced by FTIR analysis. The addition of silantrate zirconia filler has been proven to enhance mechanical characteristics, impact strengths. The research result indicate that the best impact is above at silanized zirconia concentration of 5%. However, the impact value of at concentration 2%, 3%, 4%, and 5% also meet the minimum standard set by ISO 22112 2016 for impact strengths. Thank you.
Okay, give a round of applause, please, to Ms. Zella. Okay, is there any questions on the floor for Ms. Zella regarding her own research in zirconium oxide? I think there are no questions, so please just close the presentations. Okay, thank you so much. Before that, I wanted to make sure if the person is already here. Deva Novelia. Is the person here already? Nope. Vita Fatika. Vita Fatika is not here. Agung Abadi is not here also. So once again, Agus Rianto. Okay. Okay, the next one is our previous moderator that is actually gonna present her own research for Ms. Yatim Lailan Nikmah. The time is yours.
Okay, thank you very much for the time. Today I would like present my, actually not my work, my student work. The title is the synthesis of gold nanoparticles and their stability, and we synthesize to optimize the particle size using CCD design. So we use a design to do experiment. So the experiment become little, and we connect the 3 parameters together and we run together. So we optimize 3 parameters in one running using FCCCD. So the background of this research, next. The outline of the presentation, I will present about the introduction and then methodology, result and discussion, and the conclusion. And first, the introduction. in the recent years, we know that the nanoparticle materials has a lot of attract the researcher to do synthesize nanomaterials because of the unique character of the nanomaterial. And in this research, we do synthesize of aurum nanoparticles. So we specify the nanomaterial using aurum. And the method of the synthesis, we use the chemical reduction method, and we also use the capping agent using tartaric acid because this material have CO and OH group, so it can make the aurum nanoparticle become more stable. And then next, yeah. And like I said before, for the synthesize this nanomaterial, we use CCF RSM. So we have 3 parameter here, and we combine 3 parameter and do this parameter together. So we can see later that 3 parameter is affect the synthesize or not. Next. Yeah, this is the purpose of our research to synthesize the gold nanoparticle that we can get small size of the nanoparticle, and also the morphology become homogeneous, uniform, and also we get more gold nanoparticle that we want. And then we also use the design to synthesize the gold nanoparticle using RSM CCD. We have 3 parameters here. The concentration of tartaric acid as a capping agent, and then we also use the temperature sonication and also the sonication time. So let's later we see that these 3 parameters all affect the result of the gold nanomaterials. Next. Yeah, because we use design of the research design, so we use CCF RSM design. So here we have 3 parameters here, and we give symbol X0, X1, and X2. This is belong to concentration of tartaric acid, and then X1 is sonication temperature, and X2 is sonication time. Its parameter here, we have 3 points. We call -1, 0, and 1. -1 means the lowest concentration, the lowest temperature, and also the lowest time, and +1 means the highest parameter. And 0, we take the 0 from the other researcher that already synthesized the nanomaterials. And by using the CCF design, we can see here we have 22 experiments, and each parameter here we run together, and also we have the last for data. The last is the replication of this experiment. So we also see the statistical analysis that our optimization is correct or not. Next. Yeah, this is the methodology. Okay, next. Yeah, to prove that our nanomaterials already synthesized, we use this 4 characterization. We use UV vis, and then use PSA, and then TEM, and also FTIR. Next. Go to the result and discussion. First, the data here, we have 2 figures here. A figure is only sodium aurum chloride, and the B, the red color, we already mix the capping agent, tartaric acid, and then also the source of cold, and also the reductor we use sodium borohydroxide. And this is from the UV data. We can see here that the red color means that the we have peak at around 525. It means that the cold nanoparticle already success to synthesize. And from the black color, we see here that there is no peak. It means that there is no cold nanoparticle. Next. Yeah, this is from the parameter, 3 parameter. We use temperature and then time and also concentration of tartaric acid. Here we compare 2 data which we give the code AU1 and AU2, means that the AU1 no cold nanoparticle, and from AU2 it means that the cold nanoparticle already success to synthesize. Next. Yeah, this is we also check the stability of the cold nanoparticle that we synthesize. From here we can see until the 15th day, we can see that the cold nanoparticle that we synthesize still have good stability. We can see from the absorbance there still in the high position. Next. This is from the PSA. We can see here that the distribution of our cold nanoparticle here around 100 nanometer. But for the black one, it means that the size is bigger than the red one. Next. And then from the TEM data here, we can see that we make the A feature. We see here that this is no cold nanoparticle. And in B picture, we can see here that the average of the size of our material around 20 nanometer. But without tartaric acid and also without sodium borohydroxide, we can see that the size is bigger than the left side. Next. And from the FTIR here, we can see the peak around 1,700. This is belong to CO double bond. It means that the cold nanoparticle still stable. Next. Next. Yeah. This is the data from our optimization using CCF RSM design. From here we can see that this is 3 parameter we run together, time, the concentration of tartaric acid, and then time sonication and temperature. Here we can see that from here we get the optimization condition from the 3 parameter. Yeah. In here we can see the circle means that this is the optimum condition. Next. Yeah, this is the, we can read that the RSM from this, we can see the optimum time and then temperature and also concentration of tartaric acid. Next. The last slide, this is the conclusion. So from the PSA and from TEM and also from the RSM design, we can see here that our cold nanoparticle success to synthesize, that we can see here by using tartaric acid and also sodium borohydroxide, we can get the particle size around 80 nanometer. And with TEM also, we can compare the result, and from the RSM design, we get the optimum condition here. The temperature is around 57 degrees C, and the time is 30 minutes, and also the concentration of tartaric acid is 0.5 molar. I think that is my presentation. Thank you very much.
Okay, please give a round of applause. Is there any questions on the floor itself? I think no questions. Thank you so much. This is gonna be, wait. Is Mbak Deva already here? Wait, and then Mr. Agus Rianto, still not here. Mau maju sekarang, Mbak? Nggak apa-apa, maju aja sekarang. Oke. Terserah mau di mana, tapi terserah mau di mana aja. Ada. Please do wait a second since we encountered some technical issues. And we do still have just a few more. So, yeah, we're gonna see it first. So we will. Oh, for a second. WA, kasih ke WA ke aku aja, nanti aku kirim. foto, kirim ke WA Mbak, dia kirim aku kirim sini. Satu dua sama Agus Rianto itu belum datang. Sama aja sih Bu. Sama yang paling ada sih yang satu Malaysia tadi. Saya oke. Aku aja gimana yang present?
Kamu dulu aja. Kamu dulu aja deh.
Okay, due to technical issues, we're gonna move into the next one first. Gonna present, which finally that's gonna be me. So yeah, I do it all. I moderate, I being an MC, and now I'm presented as well. So hello, good afternoon everyone. My name is Muhammad Haikal Fazat. You can call me Haikal. And this is actually a project still running, which I run with my friend Atika, which unfortunately cannot be here. So the title of our project or our research is the optimization of acid-activated zeolite as a PES membrane filler for microbial fuel cell applications, which we add zeolite, acid-activated zeolite into the polyether sulfone membrane or polymer to enhance its performance on MFC applications. And this is basically the background. We do have high demand and consumption of electricity currently, and because of that, we cannot keep up due to the previous sources to generate electricity, which mainly are natural gas, oil, and coal, keep decreasing and getting scarce day by day. It's also kind of a movement to move forward into more renewable and green energy sources. Microbial fuel cell, or MFC itself, has some positives in terms of providing bioelectricity from organic matter and wastewater. So sorry, it's still in Indonesian, but to be an electron mediator. And basically, this whole system is using microbes' metabolism to produce electricity. And as you can see, in the microbial fuel cell, you have the anode and the cathode, which gonna be separated by the proton exchange membrane. Usually, we're gonna use Navion membrane, which have a very high proton conductivity, but really much expensive at the cost itself. So because it is far too expensive, and one of our colleague once wanted to try, but in dire need of a membrane that can transport electrons or can transport electricity, we modified it to use a polymer membrane. Since polymer membrane is pretty much easy and cheap to produce, and regarding proton conductivity, it's pretty much average if we compare it to Navion membrane, since Navion is a commercially produced membrane, while polymer membrane is usually the one that we make in a lab. And pretty much it does have average proton conductivity. But since it was not commercialized, it is not going to be as effective as Navion membrane itself. So how do we enhance these polymer membrane? So we incorporate inorganic fillers to the membrane to attain the highest performance in the MFC itself. That is the purpose of the research on the project itself. So we're gonna dope the membrane with acid-activated filler that can increase the proton conductivity and the power density of the MFC itself. So that is basically how the scheme is gonna be used. Next up, so how do we activate the zeolite? So basically we use chloric acid, either like 1 molar, 2 molar, and 3 molar to the zeolite itself, and activating it and stir it for 30 minutes. And then after we stir it for 30 minutes, we filter it and then we dry it until it came into a powder looking like this white powder. And then afterwards, where the powder is going to be dispersed in NMP and added 70% of polyaderosulfone to make a dope solutions, and then we fabricate it onto a piece of glass, and we're gonna make the membrane using phase inversion method to result in a past AZ membrane itself. So how does it work? So basically, this the photo that we took. So as you can see there, oh my god. Okay, as you can see there, here is the bacterial microorganisms, or where is we use the bacteria here, and this one is the electrolyte. And as you can see here, that these two are separated in different chamber, but connected in these parts. But these parts is the part that we attach the membrane itself. So we can see we use also carbons here to measure using the multimeter itself. So this is how we conduct the experiment itself to know how the membrane itself can perform well or not. So based on the XRD itself, as you can see, this is the standard zeolite Y, and this is the activated zeolite, and 1 molar, 2 molar, and 3 molar. You can see that there is a decrease there. So in our conclusion is that the acid activation is successfully carried out because the diffraction peak, and this one is decrease, indicates that a decrease in the crystal phase in the zeolite pore itself. So the prediction is that the natrium here, which are on the zeolite itself, is changed into hydrogen because it's activated by the acid itself, but it still need to be confirmed more through SAM EDX. So this is the water contact results. This is needed to be another measurement, but we could see the trend itself that adding zeolite as a membrane filler shows the improved hydrophilicity of each membrane. So the polymer membrane itself is already hydrophilic enough, but adding zeolite or activated zeolite inside of it still decreases its hydrophilicity, meaning that it's great for water-based application. And so all of this is, all membrane variation itself is actually classified as hydrophilic membranes, as the water contact happen under 90 degrees. But through adding of the zeolite, we make the already hydrophilic membrane became more hydrophilic. And regarding the performance itself, we could see that the data shows that this is like the average performance for the voltage resulted and the current in the system for the first 3 hours. So we could see that 1 molar with 0.3 loading as a filler shows a significant result compared to the need this only zeolite dope, or even like the other activated zeolite, which have a higher acid added to it, 2 molar and 3 molars. The 3 molars shows that these decrease, as you can see it before, that the decrease itself is probably happening because the zeolite itself is not as crystal as it used to be anymore. The acid, 3 molar acid is ruining the crystal phase that the zeolite is already have. So it is impacted in the end in the performance itself. But we could see that 1 molar activation with the 0.3 loading shows that's significant results rather than the other, and shows improvement from the neat and or the usual polymer membrane, and either the only adding zeolite to the mixtures itself. And regarding the power density that we have, because of that, we also shows that 1 molar acid activated zeolite shows a significant power density compared to the other membranes that we have. So in conclusion, it is the best variations in this research. But in fact, these is needed to be researched more, since we have seen, if we compare it to previous or like to the current similar research regarding polymer membrane using in the MFC, we could see that it's pretty much far beyond the other research itself. So in our own opinion, since me and Atika, my friend, we cannot conclude something out of this, the research needs more optimization, especially looking at the potential at the others have. But we do have another conclusions, which adding the acid activated zeolite through 1 molar or like small concentrations and adding it to similar loading could enhance its performance compared to the polymer membrane itself. I think this is all that I could say regarding my research here. Thank you so much for listening, and is there any questions regarding this? I do think that there is no questions, and thank you so much for listening to my presentation itself. I'm go back to moderator mode, so so next up is already okay. This is probably gonna be our last one. Okay, wait a second. Okay, next up is Mrs. Deva Novelia to come to the front and present their research itself. Okay, to Ms. Deva, the time is yours.
Okay, thank you for the stage. Good afternoon for all honorable attendees. I'm Deva Novelia, will present about the research entitled Separation of cerium and neodymium from tin tailing with froth flotation using carbon palmitic acid as collector and sodium chloride as depressant at this International Seminar of Chemistry 2024 with the authors is Owik Dahani, Deva Novelia, Amalia Sidanisi, and the others. This research was conducted with the data of Geological Survey 2024 that mentioned the timah TBK from Indonesia became the representative of Indonesia that have a 5th rank of pure tin producers in the world after China, Peru, and Malaysia, with the total of tin production is 19,000 and 800 tons in 2022 and 15,300 tons in 2023, and it's aligned with the high amount of tin tailing in Indonesia that has containing the monazites, cassiterate, xenotime, zircon, and ilmenite with the concentration in the following below. The tin saline containing the cerium and neodymium in the form of monoxides that can be separated with the Froth flotation process. With the interaction, use differences of wettability where metal particles will interaction with moving froth due to the surface with the froth and leave the impurities below. And the separation of cerium and neodymium can be done with using the activated carbon palmitic acid as a collector and sodium chloride as a depressant. As we know that palmitic acid has a long chain of C16 and carboxyl group in the end of the carbon, and have the non-polar part of the much more than the polar part, so its hydrophobic ability is greater. And palmitic acid has an environmentally friendly and can be used as a collector that will initiate the polymerization reaction identical to hydrophobicity, which will cause flotation. And we have an activated carbon that has a low cost production, very high porosity and high adsorptive capacity, and adjustable pore size, large surface area, and amorphous structure that can be used as an adsorbent of the metal in the Froth flotation process. And sodium chloride that can adjust ionic strength in the pulp flotation, helps the formation of stable air bubbles, and increase the hydrophobicity of the metal. In the experimental chapter, I'll be explain about the chemical activation of bamboo carbon that begin with crushing the bamboo carbon and grinding so it can be sieved in 120 mesh, and the fine carbon can be soaked with 0.1 molar of sodium hydroxide, and also it can be filtered and dried, so it has to be re-soaked with 0.5 molar of hydraulic acid and washed and dried, and then it has to be calcinated so it can be analyzed with SAM, XRD, and BET. The next step is dry impregnation of activated carbon palmitic acid composites. The activated carbon has to be heating in the oven and put in the beaker so it can mix with the palmitic acid with the variety of ratio composites, and put it in the oven and stir it every 8 hours for 1 day, and the composite have to be analyzed by FTR. And the next step is Froth flotation, that we have to prepare 20 gram tin tailing to be put in the beaker of composites and contact them with the adding of 80 milliliters water and the 1 milliliters sodium chloride 1%, and the froth flotation process can be started, and we have the spirited froth that has to be dried and calcinated before it can be analyzed by XRD and XRF. And I'm going to describe about this result and discussion of this research. We're going to start with characterization of tin tailing PT timah TBK with XRD, that we have the information of phase mineral and the structure of the mineral that containing in the samples. There is the cerium phosphate, neodymium phosphate, iron oxide, silicon dioxide, and tin oxide with the angle of the table following below. And we have analysis by SEM-EDX that has an information about the morphology and the distribution of the containing in the sample. The lightest color indicates the most element in the solid, which is oxygen, and the darker color indicates the main elements with smaller atomic concentration in the solid, which is cerium, neodymium, titanium, silicon, and ferrum. And the XRF results of the tintailing sample is shown in the table following, that the cerium has a concentration that of 23.9%, and the neodymium has a concentration of 7.67%. With the other 28.58% are aluminum, sulfur, calcium, cuprum, arsen, yttrium, zircon, ytterbium, tungsten, lead, and the thorium metal with very small concentration. And the next characterization about the carbon bamboo. We have the SEM analysis that at 500 magnification, the surface morphology of activated carbon is in the form of fibers, and at the magnification of 10,000, the surface morphology of activated carbon is clearly in the form of pores that have been opened with various size. And the XRD results of the activated carbon is shown that the two main board peaks at angles of about 2 theta in 23.7 degree and 43 degree. This corresponding to reflection from the 002 and 100 planes. It is known that the activated carbon has an amorphous structure. And then we have a BETSAA from activated carbon that has a surface area from bamboo carbon is 4.7970, and the activated carbon increased to 434.28 meter per gram. And the nitrogen isotherm of carbon is followed the third type, and the nitrogen isotherm activated carbon is followed the first type. And we have a data mass from the flotation prod with 120 mesh, with the highest calcination mass is in the ACPA 2575 composite variation, which is 0.1 grams. This happens because the composite has a large amount of palmitic acid, can help activated carbon interact with the froth and lift to the surface during the flotation process. And the characterization about crystallinity analysis with XRD shown that the product flotation of tintailing 120 mesh has cerium oxide, neodymium oxide, iron oxide, and silicon dioxide with the angle of the table below. And the element content analysis with SRF of the flotation product 120 mesh has cerium by 12% and neodymium by 3.7%, and the high ferrum impurities metal content of 50.5%, which hinders the metal that is target during the flotation process. And the data mass of flotation froth using tin tailing 230 mesh has the highest calcinate mesh, which is 0.5878 grams. The composites with the ratio of activated carbon more than palmitic acid will increase the metal absorption ability of tin tailings, which is polar in nature with the presence of polar sites that do not interact with palmitic acid. And the characterization crystallinity with XRD is shown that the flotation product has cerium oxide, neodymium oxide, iron oxide, and silicon dioxide minerals with the angle in the table following. And the XRF results from the 230 mesh tin tailing has a cerium by 37% and neodymium 9.5%. That known that the finer tin tailing samples will have the largest surface area, resulting in the increased amount adsorbed on the activated carbon and attached to the palmitic acid. And the conclusion of this research is the separation of rare earth metal, cerium and neodymium using physically bound activated carbon palmitic acid composites as collectors in the froth flotation process with the addition of sodium chloride as a depressant was successfully carried out. And separation by froth flotation technique is optimized with smaller sample size of tin tailings. And the flotation with monazite 230 mesh using an activated carbon palmitic acid composite ratio of 75/25 resulted in the increase of cerium metal content from 23.7% to 37% and neodymium content from 7.67% to 9.5%. There's all from me. Thank you for the stage and thank you for the attention.
Please give a round of applause. Thank you so much, Ms. Deva. Is there any questions on the floor coming from the listener and participants? Is there any questions? Okay, I do not think there is any questions. Thank you so much. You can conclude the presentation itself. Once again, please, if you want to give applause. Okay, just for confirmation first, is Vita Fatika is already here? Vita Fatika, okay. Please, you can go to the front first. Did you already? Before that also, is Mr. Agus Rianto is already here? Nope. Agus Rianto, once again? Nope. Okay, that's totally fine. Ms. Vita, you can start the presentations.
Good afternoon, everyone. My name is Vita Fatia Rizkia. I'm from Institut Teknologi 10 November Surabaya under the supervision of Dr. Engineering Eva Octavianingrum. In this session, I will present my comparative analysis of orthophosphoric acid and dipotassium phosphate addition in the synthesis of hydroxyapatite from blue crab shell for bone graft application. Tooth extraction can lead in a change of alveolar ridge dimension. Hence, some clinicians recommend tooth grafting to maintain the alveolar ridge dimension. The material used in tooth grafting need to be osseointegration, osseoconduction, integration and osteogenic. So far, bone graft material are in autograft, allograft, and xenograft. Autogenous bone graft considered to be a golden standard due to their potential of osteoconductivity and the potential of osteoinductive. In Indonesia, crabs are a significantly export commodity and has led higher production level, generating more waste. Material for bone graft are commonly in hydroxyapatite. So hydroxyapatite can be synthesized from natural source like blue crab shell, who has high calcium content. In a post-calcination, the calcium content of blue crab shell is up to 69%, and that's why blue crab shell is a material that promising for synthesis of nanohydroxyapatite. Because nanohydroxyapatite is need to follow the medical standard, it will be testing by several analyses like XRF, FTIR, XRD, crystallinity, and EDX. Previous research has been carried out regarding to the synthesis of hydroxyapatite. In research has been conducted by Eva Octavianingrum in 2022. She varying the concentration of dipotassium phosphate and temperature of calcination, and she found that the maximum or the optimal concentration of dipotassium phosphate for hydroxyapatite synthesis is 1 molar, and the temperature of calcination is 950 degrees Celsius. And then in 2024, she varying the blending ratio of polycaprolactone and hydroxyapatite with the precursor solution is orthophosphoric acid. And she found that the synthesis of HAP or hydroxyapatite meets the medical standard, like CFRP, crystallinity, the presence of phosphate and hydroxyl, and some peak from XRD analysis. That's why from two previous research, in this research, I comparing the hydroxyapatite synthesis from crab shell in various concentration of orthophosphoric acid and dipotassium phosphate solution for bone graft application by following analysis XRF, FTIR, XRD, and EDX. And this is the methodology for synthesis of hydroxyapatite. Step 1 is preparation the material and tools for each stage, and then step 2 is pre-treatment. The blue crab shell was cleaning and then drying and then crush using a blender, and then the crushed blue crab shell is then calcinated at the temperature of 1000 degrees Celsius for 5 hours. To ensure that the calcination is produce gallium oxide, it will be testing by XRF, and then the calcium oxide from calcination is then synthesized with precursor solution, which means the potassium phosphate and orthophosphoric acid to produce hydroxyapatite. Then the hydroxyapatite that has been produced is then sintered at the temperature of 800 degrees Celsius for 4 hours. To ensure that the hydroxyapatite meets medical standard, it will be testing by FTIR, XRD, and EDX. And this is the result of XRF analysis. From XRF analysis, the calcium content in crab shell treated with dipotassium phosphate and orthophosphoric acid is notably high. In comparison, untreated crab shell typically contain 90.55% of calcium, but in this study, the calcium content are in 93%. So that's why it is indicating that the treated blue crab shell are promising calcium source for hydroxyapatite production. And then this is the result for FTIR analysis. So from the graphic, the FTIR result indicate the presence of hydroxyl, and then phosphate, and carbonate functional group at each concentration of orthophosphoric acid and dipotassium phosphate. The presence of functional group suggests that the compound is hydroxyapatite. So, but there are reduction of wave number in functional group of hydroxyl. Nah, this is indicate water loss due to calcination or increased sintering temperature. And this is the result of XRD analysis. So from XRD analysis, we can calculate the crystallinity and the crystal size of hydroxyapatite that was formed. And from the graphic of XRD, we can comparing to the GCPDS data of 090432, and from the GCPDS data, the peaks of hydroxyapatite appear at 2 angel in this number. So from the result of XRD graphic, the number is correct. So the material that has been tested by XRD is fully hydroxyapatite. And then from the crystallinity, from medical grade, hydroxyapatite have crystallinity degree for 62. But in our research, the result of percent crystallinity is above 62, so it will be good. And then in the crystal size, orthophosphoric acid is more effective in producing nano-size crystal, because hydroxyapatite is always in nanocrystal, because if the smaller size or if the nano size of hydroxyapatite, it will be more enhance the resorptibility and bioactivity. And then this is the result of EDX analysis. So from this graphic, we know that the higher of concentration precursor solution, it will lower the CAPP ratio, and also the lower CAPP ratio can lower the pH. And from this, we know that if we compare about ratio CAPP of orthophosphoric acid and dipotassium phosphate, the optimal ratio is in 1 molar of orthophosphoric acid. Why? Because the 1 molar of orthophosphoric acid demonstrating the optimal ratio that closest to the standard value, which is 1.67. So the conclusion is crab shell reacted with dipotassium phosphate and orthophosphoric acid exhibit elevated calcium levels, positioning them as promising sources for hydroxyapatite production. Second, FTIR analysis confirmed the presence of hydroxide, phosphate, and carbonate groups in the synthesis of HAP sample. Third, XRD analysis reveals that both treatment produce crystalline structure. For the crystal size of hydroxyapatite is smaller with orthophosphoric acid, enhancing its effectivity for medical application due to increased resorptibility and bioactivity. And then the last, the CAPP ratio declined with a higher concentration of both reagent, with 1 molar orthophosphoric acid demonstrating the optimal ratio closest to the standard value. And this is the following references as we use for compiling our research. Thank you for your attention.
Give a round of applause, please. Thank you so much. Is there any questions to Ms. Vita that has already presented her research in the front? Okay. Wait a second.
Thank you. My name is Ronnie. I want to ask about the methodology. Which the following steps has the most influence on the formation of the nanomaterial structure of the hydroxyapatite? Is it only because we are using the potassium phosphate, or there is any method that use effect on the nanomaterial structure?
Okay, from the methodology, sorry, there are a typo of in my PowerPoint. So this is the synthesis of hydroxyapatite by orthophosphoric acid and dipotassium phosphate. But we running the orthophosphoric acid first and then dipotassium phosphate in the second.
Okay, is there any questions, another questions for Ms. Vita in the front? Okay, seems none, and thank you so much, Ms. Vita. You can conclude the presentation itself. Okay, it's gonna be either the last one or the second to last one regarding this. For Miss Philia, you can go to the front. Okay, the time is yours.
Okay. Good afternoon, ladies and gentlemen. I'm glad to be here to present our last research about titled Optimization of Hydroxyapatite Synthesis from crab shell via ultrasonic assisted precipitation. I'm Philia Camila from Institute Technology 10 November. We have been working hard on this research below on Dr. Eva's Octavia Ningrum supervisor. Okay, imagine the world with healthy teeth. The smile of every soul will be wonderful. Okay, a significant health problem is tooth loss. It's often caused by tooth decay and gum disease. Report from World Health Organization in 2022, oral diseases occur in 3.5 billion people worldwide, and out of 4 of those people living in the middle income countries. And then from their report, they say that people over 20 years old have lost all their teeth in 7%, and it's even higher, about 22% for people over 60. There are several treatments to cure this tooth loss, like dentures and dental implant. Overall, based on the cost, jawbone stability, eating function, and process appearance, dental implant is good to be chosen rather than denture, even though it's having a high cost, but it's having good eating function, appearance, and stability. And then the dental implant nowadays is mostly used from non-degradable metal-based dental implant because the dental implant is located in the mouth. It's prone to corrosion, and there's bacteria and plaque around the implant. So we doing some research making some biodegradable implant. It makes of ceramic material. There is hydroxyapatite. There is advantage of hydroxyapatite. The first one is it's stable and biological form of calcium phosphate, and it has non-inflammatory and non-immunogenic. It's immensely increased cell attachment and enhance the bioactivity and osteoconductivity. There is the characteristic of hydroxyapatite having the CA per P ratio 1.67, having a spherical morphology, and the standard is ISO 12175/2015. And there are several materials that can be used to produce the hydroxyapatite, like crab shell, snail shell, egg shell, and chicken bone. Okay, these following slides show the previous research. The first one is research conducted by Yeltin Yilmaz in 2018 with precursor orthophosphoric acid. The synthesis temperature are 30, 50, 85 degrees Celsius, and the titration rate are 1.5 and 5.5 ml per minute. Report that the synthesis temperature at the low temperature about 30 degrees Celsius with titration rate 5.5 ml per minute, producing the stable and spherical hydroxyapatite with particle size 0.2 until 0.3 micrometer. And the second one is research conducted by Murtaza in 2014 with precursor orthophosphoric acid, and the calcination temperature 100 to 800 with the multiply of 100 degrees Celsius is report that the best hydroxyapatite produced in calcination temperature of 600 degrees Celsius. The CA per P ratio is 100.67, and their crystal size is 8.47 until 24.47 nanometer. And then the third research is by Rodrigo Lugo in 2018 with precursor ammonium phosphate with solution pH 9, 10, and 11, and the sintering temperature are 300, 500, 700, and 900 degrees Celsius, report that the lower pH will enhance the CA/PrP ratio and reduce the monotite phase. And the best sintering temperature is 900 degrees Celsius with the crystal size is 20 to 56 nanometer. Okay, this following slide shows about our research methodology. The material we use is orthophosphoric acids, aqueous, and the crab shell. In this research, we use ultrasonic assisted precipitation, and the research variable are divided into independent variable and the control variable. Okay, the first step to do is we have to prepare all the material and tools, and then we are doing pre-treatment into blue crab shell, cleaning the blue crab shell from the dirt, and then after that we have to drying the blue crab shell in microwave about an hour at 80 degrees Celsius. After that we have to crush all of the dry crab shell until smooth. And then we got the result smooth crab shell. And then after that, we do calcination on the crab shell with furnace in calcination 1000 degrees Celsius for 5 hours. And we have calcium oxide. In this process, there's obtained a reaction calcium carbonate into calcium oxide plus CO2. And then the fourth step is synthesis hydroxyapatite. Before we use the synthesis using ultrasonic water bath, first of all we have to mix, we have to stir the CaO in the 5 milliliter aqueous for an hour. And then after this, in the synthesis process, we use totally, there are 8 variables. The first one, we synthesize with orthophosphoric acid 1.25 molar, and with the volume titration is 25 milliliter and so on until 100 milliliter. We are doing the same way to synthesize in the orthophosphoric acid 1.4 molar. And then after that, we have to blend the hydroxyapatite solution for 2 hours. And then we have to filter, and filter that we got, we will sinter in furnace about 4 hours in 80 degrees Celsius. And in this process, we've got reaction CaHO2 plus orthophosphoric acid is being hydroxyapatite, and the excess is only water. And the last step is we have to analyze the hydroxyapatite. Is it meet the standard or not? And then the analysis we use in this process, there are FTIR tests and EDX tests. FTIR tests, we aims to know the phosphate and hydroxyl functional groups in the hydroxyapatite that we produce, and the EDX test is to know the CA/PrP ratio. Okay, this following slide shows about effect of orthophosphoric acid volume on hydroxyapatite mesh. As we know that the higher volume we get will be increase the mesh hydroxyapatite produce. It seems linear both on orthophosphoric acid 1.25 molar and also 1.4 molar. And then this is the results of FTIR groups, FTIR analysis from both concentration. We've got hydroxyl and also we've got hydroxyl and also okay. And then it means that all of our results is meet the standard that hydroxyapatite must have bond between OH as hydroxyl and the phosphate bonds. And then the last analysis is CA/PrP ratio analysis by EDX. As we know that the higher volume we use in this study, the higher volume of orthophosphoric acid will lower Ca per P ratio. And if we look into this graphic, we've got the bigger concentration of orthophosphoric acid we use will having the lower Ca per P ratio. From this study, we can conclude that the concentration of orthophosphoric acid titration and volume significantly influence the mass of hydroxyapatite produced. And the FTIR analysis of orthophosphoric acids 1.25 molar and 1.4 molar in each volume, 25 until 100 milli, consistently identifies the phosphate and hydroxyl groups within the hydroxyapatite structure, confirming the successful synthesis. And then the last analysis is IDX is report that Ca per P ratio of our study with concentration of orthophosphoric acid is 1.4 molar with volume 100 milli. Orthophosphoric acid report that the Ca per P is equal to standard. There is 1.75, which is close to the ISO standard. Okay, this is several reference we use into this research. And thank you for your attention. I really open for any discussion if there is, and I really open for any discussion. Thank you.
Thank you so much. Give a round of applause for Ms. Philia. Is there any questions? Okay, I'm gonna go there first. Wait a second.
Thank you, moderator. My name is Vito Binang Saputra from 11 Maret University. I want to ask, in the FTIR spectra, we can see a similar trend at the 25 till 75 milli, but at the 100 milli, there is a new peaks appears. Can you explain why that the phenomena can happen and what the vibration was? Thank you.
Okay, thank you for the question. Okay, as we see on both diagrams, for orthophosphoric acid 1.25 molar and also 1.4 molar, it shows that in the volume 100 milli and in here is the same, but if in volume from 25 until 75, why the result of the peaks is not the same? Yeah, based on our study literature and why the peaks is a lot of peaks here rather than the other volumes variable, it causes the volume of orthophosphoric acid we use will be affect the result the peak, because and also in phosphate bond in about 100 until 1,000 and 100,000, there is a lot of vibration, like asymmetric vibration from phosphate functional groups. Okay.
Okay, thank you so much. Is there any questions left for the people in here? From the people? From the floor? Okay, I do think that it is concluded. You can close the presentation itself.
Okay, thank you so much for your attention. I'm Philia. Thank you.
Thank you so much. Once again, one applause for Ms. Philia. And we got with the speech from, with the presentation from the Ms. Philia itself. Here's a conclude our parallel sessions for today. Thank you so much for attending the parallel sessions on day one of the 6th Isoc 2024. Just want a reminder that we're gonna start tomorrow by a second day in the plenary sessions. We're gonna start from 8:30 a.m. in the morning, and for the keynote session itself, gonna start at 11:30 a.m. in the morning, in the afternoon. So please do not late because we are going to start according to the schedule itself that is already been sent to you in the email by our committee and in the newest file of the program book that has already been emailed to you guys. I think this is all coming from me. Thank you so much for attending and thank you so much for your attentions. You are allowed to leave the room right now. Thank you so much.